Ladies and gentlemen, good morning and Assalamu alaikum. Welcome to the first session of the third day of the STEM Education Training Program funded by British Council. Let's start the session with the name of Allah Almighty.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الله الذي خلق السماوات والأرض وأنزل من السماء ماء فأخرج به من الثمرات رزقا لكم وسخر لكم الفلك لتجري في البحر بأمره وسخر لكم الأنهار صدق الله العلي العظيم.
Jazakallah. Ladies and gentlemen, today we have with us Dr. Sadan Sahur. She completed her PhD in biotechnology in 2013 from Government College University, Lahore. She worked for 3 years as teaching assistant and lecturer in Government College University, Lahore. Currently, she is working as assistant professor and in charge Department of Biotechnology at Virtual University of Pakistan. Her research interests are enzymology and molecular biology. She has more than 20 research publications in peer-reviewed national and international journals. Today, she will deliver her talk on Thermophiles, biotechnological potential and utilization. Welcome, ma'am. Please proceed for your presentation.
Thank you, Ms. Samar. Assalamu alaikum. Today, the topic of my talk is Thermophiles, biotechnological potential and utilization. It is said that there is no vacant space on the earth with respect to life. Life exists in everywhere where the conditions are not favorable. For example, there are many groups that live in extreme conditions, and this group of microorganisms is known as extremophiles. For example, there are many different types of extremophiles. The cyclophiles that grow at low temperature, heterophiles that grow at high salt concentration, metallophiles that live at high metal concentration, acidophiles and alkalophiles. These are the group of microorganisms that live at very low and very high pH respectively. Radiophiles that love to grow at high radiation in the presence of high radiations. Barophiles survive in high pressures, and the group that I am focusing today is thermophiles. These are the microorganisms that grow at high temperature. Thermophiles is derived from 2 Latin words, and these are basically the group of microorganisms that love to grow at high temperature. All groups of organisms can be thermophiles, but mostly the thermophiles are archaea, and some of the thermophiles belong to the group bacteria and eukarya. If we talk about the details of the thermophiles are further divided into different groups. There are moderate thermophiles, extreme thermophiles, and hyperthermophiles, having the range of moderate thermophiles 45 to 60, extreme thermophiles 60 to 80, and hyperthermophiles 80 to 110. Talking about the habitat of the thermophiles exist in very vast different conditions or environments. Some of the habitats are natural and some of the habitats are man-made. In the natural habitats, we have hot springs, deep ocean basin cores, hydrothermal vents, and petroleum reservoirs. While the man-made include drainage mines, biological waste, fermented compost. Recently, there is a new habitat that is reported for the growth of thermophiles. This is a Pilosphere. This is basically the area of plant that is above the ground. A study reported that when the 16S RNA of the microorganisms of this plant is done, 4 to 99% of the organisms that were isolated are thermophiles that are having a temperature optimum 55 degrees centigrade. Among all the habitats of the thermophiles, hot springs have taken most advantage or the most importance, because there are various hot springs, and it is very easy to explore or isolate and work on the microflora of the hot spring as compared to the other localities. In addition to the high temperature of the hot springs, there are some additional factors or additional conditions that make the survival of thermophiles even challenging. That is, the hot springs are often low in organic matter. They contain high dissolved solids, contain sulfur, and they are exposed to high light intensity. If we talk about the occurrence of the hot springs are spread all around the world, from Turkey to China, Canada, Jordan, Malaysia, Egypt, United States, and Saudi Arabia. Pakistan is also blessed with many hot springs. There are many hot springs that are located at Chitral, Azad Kashmir, Murtazabad, Hunza, Gilgit, Karachi. And these hot springs are explored more with reference to their physical chemical properties and less with respect to their microbes. Now, what are the mechanisms or the adaptations that organism must have to adopt in order to become able to survive in extreme condition or extreme high temperature? Because all of the microorganisms cannot survive at high temperature. It has been seen that the thermophiles, they have high GC content. They have cell membrane that is containing more saturated fatty acids. They have reverse DNA gyrase. They have increased frequencies of pyrimidine and purine composition, histones and histone resembling proteins, post-translational modifications, methylation, and specialized proteins, chaperones. Now, focusing on the thermozymes, thermozymes are the enzymes that are produced by the thermophilic bacteria. Thermozymes have certain properties that that make them able to work at high temperature. They are thermostable and oppose denaturation and proteolysis. They have improved electrostatic and hydrophobic interactions. They have few thermolabile amino acids that are exposed to the surface. They have substitution of the exposed thermolabile amino acids and increased numbers of disulfide bonds. If we compare the thermophilic proteins with the mesophilic proteins, as we all know that the three-dimensional structure of the proteins is stabilized by different interactions, that is the ion pairs, hydrostatic interactions, hydrophobic interactions, disulfide bridges, and hydrogen bonds. So talking about the thermozymes, they have more ion pairs or more electrostatic interactions in their proteins. They have hydrophobic amino acid residues buried in their core, and they have more disulfide linkages and more hydrogen bonds that make them stable at high temperature and resist to denaturation. When we talk about the habitat of thermophiles, it is not usual. There are many additional pressures. There are additional things that make them difficult to survive in the lab conditions. So when we want to grow the thermophiles in the lab conditions, we need complex nutritional requirements, appropriate medium enclosing carbon or additional carbon sources or energy sources. In addition, if we talk about the anaerobic microbes, they need additional many cofactors and many microelements that are without which they cannot grow. If we want to utilize the thermophiles in our work or work used to, we want to work on any of the metabolites of the thermophiles or their enzymes, the first step is a sample collection that can be done from any of the sources having high temperature. Then is the isolation that can be done by different techniques, spread plate method, pour plate method, or streaking. Then is the characterization. Characterization depends on different factors, or it is done by different ways. Morphological characterization, biochemical and molecular. In morphological characterization, simply the colonies characteristics are studies. In biochemical characterization, catalase tests, oxidase tests, and then different screening tests are done on the basis of biochemical assays or biochemical tests, for which different types of kits can be used, such as API, REML, or QTS-20 test strips. Then all these characterization give an idea of the group of the organism that is present in that hot spring or that locality. But for the accurate identification, we need to go to the molecular analysis or molecular investigations. Typical steps in the molecular identification or molecular characterization, there is the isolation of DNA, then PCR, specific gene amplifications, then sequencing and phylogenetic analysis that give us idea about the microbe that is present in our sample. Like other ways, the DNA isolation of thermophiles is also not an easy task, as it has a cell membrane that is different from the other mesophiles. So in order to isolate the DNA from the thermophiles, we need additional modifications in the listed protocols. We have to give them more incubation at high temperatures in order to disrupt the cells. And then we have to check its purity. As the yields of DNA from the thermophile is less as compared to other methods or other microbes. When we want to culture the microorganisms or isolate the microorganisms from the thermophilic environments, only a percentage of the microorganisms, these are isolated. Not all of the microbes cannot be cultured in the lab conditions. So if we talk about, if there are 100 microbes, maybe 20 to 30 different microbes we are able to isolate in our lab. So we don't have the idea of the complete biodiversity of that region or that habitat. In order to do so, if we want to know about the complete biodiversity of that habitat, we need to go for metagenomic approach. That is the total DNA isolation and then working on that total DNA isolation independent of the culturing. And then after metagenomic and DNA isolation, we go for the NGS approaches to check for the thermophiles. Regarding the diversity of thermophiles belong to all the groups. There are archaea, there are bacteria, there are different algae, fungi, all these different groups, they belong to thermophiles. Mostly these are archaea and bacteria that are reported. And in case of Pakistan, mostly the hot springs of Pakistan, they are rich in geobacillus strains. And regarding the anaerobic, there are thermococcus strains reported in Pakistan. As we know that the growth of the thermophilic organism is very difficult, and it's very difficult to maintain the thermophilic microorganism in the lab conditions till the production of our desired metabolites. Though they produce the metabolites, when we grow the thermophiles in our lab conditions, they need longer time to grow and longer time to produce our desired metabolites as compared to mesophiles. So if we are going to work on an isolate that has an optimum temperature of 70 or 80, so there are chances of that the microorganism or in which the medium the microorganism is growing, it will dry out. during such a long time. Secondly, while working with the thermophiles, we have to repeatedly reculture the microorganism in order to maintain it in the culture. If we are not going to maintain it in the growing condition, it will become dormant and it's very difficult to reculture that microorganism. So one approach is to, if we want to utilize some metabolites or the enzymes of the thermophilic microorganisms, one easiest way is to clone the gene of interest into some mesophilic host. If we are going to clone the thermophilic gene in the mesophilic host, we will able to get the desired results at the mesophilic conditions and at high level. So if the steps that are including in the expression of a thermophilic gene is a mesophilic host, first there are two steps. First is the cloning in a cloning vector, then cloning in an expression vector, and then checking of the expression. So if we want to clone that gene, first of all, we isolate the DNA from the thermophile, then we amplify our gene of interest. While designing the primers for cloning, we use a sequence CAT-ATG at the start of the vector, and a GC clamp in order to protect it. The reason for adding a CAT-ATG sequence is that when it is a restriction site for ND1 enzyme, when the ND1 enzyme is later used to cut the DNA, it will give a ATG, that is the starting codon. So it will help in the gene expression. Second is after the amplification of the gene is ligated into specialized vector. One such vector, specialized cloning vectors, one such vector is PTZ57R/D. The purpose or the benefit of using this vector is that it gives easy screening. It is a linearized vector that is having a T overhang. When we do the PCR of the gene, there is a overhang at the 3' end. So by using this vector, the A overhang and the T overhang then combine and facilitate the ligation of the gene into the vector. Secondly, this vector also helps in the easy screening, that is the blue-white screening. In the blue-white screening, only the colonies that are containing the recombinant plasmid or the gene, they are white in color, while the colonies that are not containing the gene, they are blue in color. We can have an idea of that the white colonies are having the gene, but in order to further confirm it, whether it is having the gene or not, we confirm it through the colony PCR and double restriction. That gives the idea whether the gene size is obtained on the gel or not. Cloning in the cloning vector helps to save our gene and to increase the copy number of the gene. But the gene cannot be expressed in the cloning vector. As we want to work on the expression of the gene, because our ultimate target is to use that gene product into our future work, in research work or in our industries. So we clone the gene into an expression vector. First of all, the cloning vector is double restricted. The gene is then again ligated into another vector, that is FAT22B plus or some other vector. These are also specialized vectors that have some additions, some deletions to facilitate the expression of the gene. Then the ligation of the purified gene with the help of T4DNA ligase, transformation into E. coli. Now at this stage, we have to check with the help of activity assay, with the help of colony PCR or double restriction, because the expression vectors till now do not allow us to rapid screening like the addition of dyes, as is in case of cloning vectors. Once the gene is cloned successfully in the expression vectors, we need to further confirm whether the product of the gene is functional or not. As we are changing the host, as we are changing the mechanism of the gene expression, there are chances that there is protein, but it is not in the functional form. So in order to check this, the recombinant cells are grown in a medium with ampicillin. Ampicillin acts as a marker that does not allow the growth of the cells that are not having the plasmid. Then, as the plasmids work under IPTG inducer, so these are induced with IPTG, cell harvesting is done. And then after the cell harvesting, there are two fractions. One is the intracellular fraction and then the extracellular fraction. In most of the expression studies, the enzyme or the metabolite that is expressed, it is in the periplasmic space or is in the intracellular fraction. But both of the fractions are checked with the help of STS Phage or activity assay. Once it is confirmed that the gene has been obtained, the partial purification of the enzyme is done with the help of ammonium sulfate precipitation, and then final or the complete purification is done with the help of chromatographic techniques. After chromatographic techniques, the enzyme assays are carried out to check the expression of the desired enzyme. Once the expression is obtained, then it is optimized with respect to different factors such as the concentration of IPTG, time duration, effect of metal lines in order to get the maximum production of the desired enzyme. Because of the benefits of the thermophilic nature of the microorganisms and their thermozymes, these are often used in industry and preferred in industries for their different functions or different phenomena. Some of the factors that make them important or facilitate their use in the industry include the robust and fast-growing microorganisms. They have high growth rates. These enzymes are thermostable. Because of most of the chemical reactions in the industries are done at high temperature. So there are chances if we use mesophilic enzymes, they become denatured. So using the thermophilic enzymes, they facilitate and they accelerate the reaction. And when we use the thermophilic enzyme, there is a low risk of contamination because at high temperature, normal microorganisms or mesophilic microorganisms don't grow, and only those organisms will grow that are thermophilic in nature. They promote the stability and efficient mixing of the substrate and facilitate the downstream product recovery. When we have to downstream or we have to purify our product from the cell medium, if we are having a thermozyme or a thermophilic product, then we simply need to heat that mixture at high temperature. When we are heating that mixture at high temperature, all of the mesophilic components or mesophilic metabolites, they will be destroyed and only thermozymes will be left. Despite of the advantages, these are not very much commonly used because of certain disadvantages, as the genetic makeup and the metabolism of thermophilic microorganisms is not completely known. And there are bioengineering tools that are less efficiently used in case of thermophiles as compared to mesophiles. Infrastructure and fermentation expertise, these are built mostly around the mesophiles. And these are not suitable for all the heterologous protein infection expressions. Sometimes the expression is not at the level that we want. Regarding the applications, the foremost application of the thermozyme is in the polymerase chain reaction. We all know that the tag is obtained from Thermus aquaticus that is used in PCR, and PCR is used in different diagnostics. In addition to tag polymerase, there are other polymerases that are also obtained from the thermophilic microorganisms and routinely used in PCR. Similarly, there are certain pharmaceutical applications of these enzymes. Thermostable proteases are used in pharmaceutical applications. Aspergillus stereus is a thermophilic fungi that produces pterine, and pterine has a cytotoxic activity against the cells having colorectal carcinoma. When we talk about the extremophiles, it is not only the one factor that is present in that extremophile. Sometimes more than one factors is combined with the other factor, and they make a combination of 2 or 3 different extremozymes. For example, the metal tolerant thermophiles. These metal tolerant thermophiles they grow at high temperature, then are also resistant to high metals. And these are used in biosensing, biomining, and bioremediations. Similarly, the biotechnological applications of thermophilic microalgae and cyanobacteria includes the production of bioethanol, biodiesel, wastewater management, pigment production, dye removal, antimicrobial agents, and antioxidant effects. Thermophilic fungi has been used to improve the soil fertility for the production of different enzymes, for antimicrobial production, biotransformation and bioremediation studies, composting, and biofuel production. There's a list of enzymes that can be obtained from thermophiles and used in industries. Some of the common ones, such as cellulase, routinely used in paper and pulp industry, food industry, textile, fuel, beer industry, agriculture industry, and waste management. Microbial lipases have been used in textile industry, cosmetics, paper and pulp, pharmaceutical industry, biofuel, leather industry, and detergent industry. Thank you. And if there's any question. जी सर.
I just want to know आपने दो वैक्टर्स का जिक्र किया है 57 RD और 7 एक एक्सप्रेशन वेक्टर है और दूसरा आपने कहा इसके अलावा जो इसकी रिक्वायरमेंट है आप समझते हैं कि ये हम जो आपने जो शुरू वाला कहा कि इसमें हैंगिंग जो है वो ई है और जिसकी वजह से हम इसको यूज कर सकते हैं फॉर क्लोनिंग प्रोसेस. तो अब मैंने ये यूज नहीं किया, मैं अपने मिशन के लिए क्या ये टी अगर आप बात करें टर्मिनेशन का कोई रोल तो नहीं प्ले करता अगर ये हैंगिंग ट्रीज हैं. Just fermentation ये ऐसा. दूसरा जो आपने कहा ये प्लस है ये आप यूज करते हैं कि थर्मोफाइल्स या सर ये बेसिकली थर्मोफाइल की चीज हमने ये लेने के बाद उसको आपने जब पीटीसी में डाल दिया पीटीसी के बाद आप सारा जो काम है वो मीजोफिलिक कोर्स में तो ये उसमें नहीं थर्मोफाइल में अब आप नहीं करते ये दोनों चीज मिल जाती है कि आप थर्मोफाइल को हैंडल करना बहुत मुश्किल है और अगर आपने उसको लास्ट स्टेज में करना है तो उसके लिए तो आपको अगर आप ओके हमने जो शुरू में एक्सट्रैक्ट किया था थर्मोफाइल आप उसको एज सच लेके नहीं चलेंगे बल्कि आप उसमें से जो डिजायर्ड जीन है जीन ऑफ इंटरेस्ट है वो आप लेके क्लोन करेंगे और उसको फिर आगे आप एक मेरा क्वेश्चन है आपने कहा कि थर्मोफाइल्स जो है उनका एग्रीकल्चर में भी जो है वो यूज है लेकिन ये थोड़ा सा हम क्लियर करें कि ये एग्रीकल्चर के हवाले से हम क्या कोई ऐसी रिलेशनशिप बिल्डअप करें जो स्ट्रेसेस को या हीट टॉलरेंट के लिए कर देंगे या आप किस कॉन्सेप्ट में ये बता रहे हैं एग्रीकल्चर में हम कहते हैं एग्रीकल्चर का जो बेस होता है उसको बेसिकली यूज करते हैं केमिकल एंजाइम्स सप्लाई करते हैं उससे हम प्रोडक्ट्स दे सकते हैं जो आप बात करें ना उसमें 0.5 स्ट्रेस के साथ एप्लो वाटर लेवल के साथ प्लांट्स के साथ बाइंड करने के उसको स्ट्रेस आप सिंपली उसको डिग्रेड करके एग्रीकल्चर प्रोड्यूस है उसको डिग्रेड करके इनके प्रोडक्ट्स बनाएं तो मैम इस तरह हम यूज वेस्ट भी यूज कर सकते हैं कि जैसे मास या डिफरेंट हाई टेंपरेचर वाले हैं हम इन माइक्रोब्स को वहां एज एक्सपेरिमेंटल भी क्या यूज किया जा सकता है फिर तो मतलब है कोई और प्लेनेट जिसका ज्यादा हो तो क्या हम इन लिविंग ऑर्गेनिज्म एनारोब्स को वहां पर ट्रायल ले सकते हैं तो आप ले सकते हैं वहां पे एक आपको कॉन्सेप्ट है ना थर्मोफाइल्स का तो वो उसकी एप्लीकेशंस के लिए आप कुछ एसिड एप्लीकेबल और सकता है लेकिन थर्मोफाइल वहां पे कुछ आप वहां पे लेके जाके उसको क्या करते हो वहां पे तो लाइफ नहीं है ना वहां पे कुछ है कंडीशंस को मेंटेन करें लेकिन सर्वाइवल जो है सर्वाइवल तो होगी हाइपर प्रेशर में सर्वाइवल तो होगी चांस है
Although the gene is present, we are getting the result from the STS page, but it is not expressing because the change of the conditions has affected the structure of the protein. So we have to reconsider our vector, reconsider our host, reconsider our conditions, reconsider our the inducers in order to get our optimum results or desired results. Yes. In every experiment, we have to run the control. First, we optimize the conditions at the thermophilic level in the lab scale. Then we transfer the gene into mesophilic host, optimize it, and then compare its effect. One of the purpose to do the cloning experiment, because it is not an easy task. It takes many retakes in order to get a successful result or a successful experiment. So one purpose is to improve the expression of the gene or the expression of our desired product in the mesophilic conditions. We need to change the vectors, we need to change the conditions, optimize the conditions, check the effects of different ions, different metals, different cofactors. And then when we get the optimum results, we then save that conditions and that particular vector for future studies. हम नॉर्मली जितने भी एक्सपेरिमेंट्स करते हैं, जितने भी हम एक्सपेरिमेंट्स करवाते हैं स्टूडेंट्स को, हम मीजोफाइल्स चूज करते हैं इसलिए क्योंकि मीजोफाइल्स को हैंडल करना इजी होता है. जितना लिटरेचर मोस्टली अवेलेबल है वो भी मीजोफाइल्स पे है. तो मीजोफाइल्स की जो कंडीशंस है वो हमें रेडिली अवेलेबल होती है. अच्छा ये करेंगे तो हमारे पास ऑप्टिमम रिजल्ट आ जाएंगे इस ऑर्गेनिज्म के साथ. लेकिन जब आप थर्मोफाइल्स की तरफ जाते हैं तो थर्मोफाइल्स का क्योंकि हैबिटेट बिल्कुल डिफरेंट होता है, उसको आप वहाँ उस हैबिटेट को आपको लैब में मेंटेन करना बहुत मुश्किल होता है. मीजोफाइल के लिए तो एक सिंपल मीडियम है, आप जो मीडियम देंगे वो उसमें ग्रो करेगा. थर्मोफाइल्स के लिए एडिशनल फैक्टर्स चाहिए होते हैं, एडिशनल को फैक्टर्स चाहिए होते हैं, एडिशनल अब सम मोस्टली जो थर्मोफाइल्स होते हैं क्योंकि वो हाई टेम्परेचर पे होते हैं, वहाँ पे ऑक्सीजन लेवल कम होता है. तो जब आप उन्हें लैब में लेके आते हैं तो वो ग्रो नहीं करते हैं. ऑल्दो वो एनारोब्स नहीं होते हैं लेकिन वो ग्रो नहीं करते हैं. फिर उसके लिए आपको ऑप्टिमाइजेशन स्टडीज करनी पड़ती हैं कि आपको उसको चेक करना पड़ता है ये फैकल्टेटिव है या ऑब्लिगेट है. अगर फैकल्टेटिव है तो उसके लिए फिर आपको डिफरेंट एक्सपेरिमेंट्स करने पड़ते हैं. या तो आप पोर प्लेट मेथड यूज कर लेते हैं या ऊपर आप उसके एगर की लेयर पोर कर देते हैं जब ताकि उसको ऑक्सीजन लेवल जो है वो रूटीन से या नॉर्मल एटमॉस्फेरिक लेवल से थोड़ा कम हो जाए. तो ये सारी जो कंडीशंस है ये मोस्टली ना पेपर्स में रिपोर्टेड होती हैं और ना इस पे ज़्यादा काम हुआ होता. तो जब आप कोई थर्मोफाइल या किसी भी एक्सट्रीमोफाइल पे काम करते हैं तो उनकी कंडीशंस आपको खुद से ऑप्टिमाइज करनी पड़ती हैं जिसमें काफ़ी टाइम लग जाता है. जी मेरा जो पीएचडी का काम था वो मैंने जो आज़ाद कश्मीर पे हॉट स्प्रिंग्स है तत्ता पानी उनके माइक्रोब्स की आइसोलेशन, कैरेक्टराइजेशन और बायोटेक्नोलॉजिकल पोटेंशियल पे है. और वहाँ पे मोस्टली जो रोबिक माइक्रोब्स हैं वो थर्मो जियोबैसिलस के डिफरेंट स्ट्रेन्स हैं और जो एनारोब्स हैं उसमें थर्मोकोकस के स्ट्रेन्स हैं. नहीं उसका नहीं है. उन जो हॉट स्प्रिंग्स का हाईएस्ट टेम्परेचर है वो भी रेंज करता है 70 to 75, 80 तक. और जो एंजाइम्स का ऑप्टिमम टेम्परेचर था वो भी जो जियोबैसिलस के स्ट्रेन्स हैं उसमें 70 to 75 की रेंज में उनकी ऑप्टिमम ग्रोथ है और जो थर्मोकोकस है उसकी 80. लेकिन एक्वाटिकस के लिए तो जो नहीं, टैग पॉलीमरेज एंजाइम के लिए आपको ना स्ट्रेन चाहिए, थर्मस एक्वाटिकस चाहिए या प्यूरोकोकस चाहिए, जिसको फिर आप पायरोकोकस चाहिए, जिसको आप यूज करके फिर आगे से उसकी जीन को एक्सप्रेस करा सकें। अगर आपके पास सोर्स जीन हो तो एक्सप्रेसो हो सकती है, एक्सप्रेसो उसने मेसोफिलिक में ही होना है। आई थिंक कैम जो है, कैम अपना टैग को जनरेट करता है। हाँ, तो उनके स्टूडेंट्स यूज करते हैं, पॉसिबल है। आप जो स्ट्रेन है वो कमर्शियली भी अवेलेबल है, स्ट्रेन को कमर्शियली परचेज करके उसकी जीन को एक्सप्रेस करके अपनी लैब में सेव करके यूज किया जा सकता है। क्योंकि मैंने जितना भी लिटरेचर पढ़ा है, या इसके अलावा 2-3 और हॉट स्प्रिंग्स पे भी थोड़ा सा काम किया है, यहाँ पे पाकिस्तान में मोस्टली जो स्ट्रेन है वो जियोबैसिलस की हैं। ये जो थर्मो टैग के लिए या इनके जो हाई टेम्परेचर 90+ या वो नहीं है। Initially, when I isolated, there were about 35 aerobic and 7 anaerobic strains. But when I further characterized on the basis of morphology, physiology, and biochemical molecular basis, there are basically 5 different strains of Geobacillus and 2 different strains of Thermococcus in Azad Kashmir. Thank you.
Assalamu alaikum. Can you hear me? Hello?
Assalamu alaikum, sir. This is Samar here.
Samar, assalamu alaikum, finally. I'm so sorry. I think this issue has been there due to my, I think some issues were there. So anyway, so I'm back now. So did you manage to open up my my talk?
Sir, it's on. So here's up. Sir, your presentation was will tomorrow, I think.
Was it today? On the 16th? Yeah, Hashmi Saab just talking to because I was on the 16th.
Yes. Yes, sir. According to schedule, your presentation is tomorrow.
Ah, okay. Okay, then maybe it's wrong. So sorry about that. I think I was just, I got a message from Hashmi Saab that we are not able to open this up.
Yes, sir. He told me.
Yeah.
You will connect tomorrow at 10 a.m. or 10:15.
Okay, but that's fine then. Please then continue. Sorry, I got interruption. So please do look at what I have sent you as a recording. So then maybe I'll have a separate chat with you. So my colleague Rabia is getting in touch with you. So because I'll be in the airport, so are sort of a bit of a travel. So let's see if you can see if that is working now. Check it on your computer.
It's okay,
please.
sir. We were downloading the recording you have sent, but I think it's any problem with it. So
No, there's a YouTube link that we shared to you.
Yes. So it's not working as well.
Did you respond to the okay, maybe I think you're talking in front of other people. Maybe if you just drop me a text, and then my colleague who's helping on this is Rabia. So Rabia will be in touch with you.
Yes, sir. Rabia was in touch with me, and I told her that it's not working. So it is requested tomorrow if you come online at 10:15 or 10 a.m.
Okay, thank you. We'll do that, inshallah. So I think was in my diary is the 16th. So this is pretty sure. So I think maybe something has crossed the wires.
So your lecture is of 1 hour?
Yeah.
Let's start right now because our next speaker is a bit late. So let's start today.
Okay. Okay then. Thank you. So sorry about this whole episode and really appreciate what I can say. When you are travel, things are not in your control. It's always a bit of trick. Then
It's
how
okay,
you-
sir. No problem.
So everybody set? Yes, start. I don't know why. Okay, but anyway, I think I'll just quickly say thank you so much, Virtual University, for giving me a chance to present. And again, a bit of my introduction, I would say, and thank you to the British Council putting investment into this program. And I represent here UpSign as well, which is the UK Pakistan Science Innovation Network. So based here in the UK, but we work in Pakistan. Most of you know another program which I'm engaged is a program called Savi. So Savi is a digital platform that links the research translation to the farmers. And thank you so much again for sparing time, and I appreciate it isn't a best it's not a best practice to deliver things online, but I think we are now used to this due to COVID, so things have moved on, so which is giving us a much more efficient as well. So as I am traveling at the moment, I'm in Saudi, so so this is all beauty of the technology and the virtual world, so that we are connected. So connection makes us a much more, I would say, efficient and much more reliable as well. So I will go in more details about the innovation and enterprise, why it is important in a research setting, in a university, or in part of curriculum. So I have a few slides here, which I will just run through with you, and then maybe come up with some questions. Then, yeah.
Excuse me, sir.
Can you hear me?
No, sir, we can hear you, but video is not audible.
Okay, okay. So video is not audible. I think there's one option here, share sound as well. So now you will be able to this could play an important role. Can you hear now? Taking forward the challenges that can you hear the audio? Humanity is facing. So I have 5 components to cover today in my talk. So first of all, I'll try to define what's innovation, and why is the need for innovation there, and what sort of innovation processes are, and then technology readiness level, we call this TRL, and then I'll just jump on to the business plan at the end. So any question you have, you can ask me afterward. I think it's just getting crazy, so maybe I'll use my own sound. So this is the recorded one is making. So I think the discussion we'll have more. How many of you know innovation? So what does innovation mean? Please raise your hands. I just want to see how many of you know about innovation. Okay, quite a few of you know about innovation. So innovation is basically
new idea, a
Yeah,
new
please.
thought, new idea, innovation.
Thank you. Next person, please. Yes, I'm maybe just let me make bigger so that I can see everybody. Okay. Yes, please. Yeah. So the next person, please. So when we say new idea, new thought, so ma'am.
Sir, actually, new idea with an applicable process or you can create some application or something which is doable actually.
Okay, thank you. That's very interesting because doable. So I think the word doable is important. So you could think something could be imaginative, but putting them into practice is the important bit. This is where, okay, there's somebody on the back here raised hand, I think in blue. Yes, please. Green. So,
I think
okay.
an idea which is doable and sustainable too.
Oh, this is very helpful because sustainable is the important bit. So sustainable mean where it could be. So maybe just to define what is sustainable. So please, maybe because you have opened up a quite interesting
Sustainable is something which can survive, which can stay for a while, or it does not just go away after some time. It's not temporary. It's there to stay permanent.
Okay, that's
For the
quite
long
interesting.
run.
So you use this word permanent. Permanent is debatable, I would say. There isn't anything permanent in this universe. Things change as well. So it's maybe sustainability have different definitions. If a corporate firm, like maybe I'm just giving example from Pakistan context, Nestle wanted to be sustainable, so sustainable in their profits. So what does it mean? So maybe just elaborate a bit more. So sustainable mean not only what sort of sustainability are. So there are usually three components of sustainability. Anybody know what are the three components of sustainability? Anybody know here?
No, sir.
Sustainability, I mean, always we look at, I just give you example of maybe food, or we give example of clothing. So sustainable is three components are always there. First is always the key thing is what benefits it's going to bring to you. So if it's a food benefit is in terms of health. So, but if it's I would say important element is economics. So is it economically sustainable? So maybe a solution we have today, I just give example of maybe the water we have available in Pakistan. It's a limited resource. But if you are looking at the same water in a place where it's in the desert, so it's a quite expensive bit to have that water in desert conditions. But also its effect on the environment as well. So I think the important element these days, sustainability is much more linked with the environment. So how it is sustainable. The operations that you are using is sustainable for the planet in terms of protecting the environment. So are we using a lot of energy, a lot of water, carbon to address a challenge or a solution that we are bringing in? So I think this is where sustainability is if you go in future, whenever we look at innovation, it has to be sustainable as well. Sustainable not only in terms of economics, sustainable in terms of environment, sustainable in terms of society. So these are the three important pillars of sustainability. So which is it's like a triangle. So how to make a balance between society, between economics, and between environment. So economics, environment, and society. So three pillars, three corners of a triangle. So it's basically innovation is always looking at those three pillars, three corners of the triangle. Anybody want to add what does mean by environment here? Anyone in volunteer just want to explain from Pakistan perspective what environment mean for sustainability? Ma'am, I just saw somebody was putting their hand and they just put that down as well.
Assalamu alaikum, Dr. Fana Lalaro here. According to, I think environment is the surrounding in which we live.
Yeah, and
Our habitat.
Okay, yeah, that's really interesting. And what sort of measures are taken? So when you look at environment, when you are looking at innovation, so what should it make to the environment?
There are many abiotic and biotic factors
Okay.
which constitutes our environment and interactions among all those factors.
Okay, ma'am, just that's very interesting, and I think that's a quite scientific way to explain biotic and abiotic. So, what does it mean for a common person, ma'am? I just want to again go back to you, please. Bring the mic to the ma'am. So, what does it mean for a common, for a layman? So, that environment, what does it bring to me?
Surroundings with which we interact.
Sorry, आपकी आवाज नहीं आ रही. Sorry, there was maybe mic off.
It is the surrounding with which we interact.
Mm-hmm. So, okay, so if you are a farmer, so what is the environment mean for you? So maybe just give a bit of example here. So my innovation is going to bring something new to you, but what will be a benefit for you as a farmer? Maybe just elaborate a bit more, please.
As you have explained earlier, it means all the
Sorry, ma'am, आपका mic off है.
As you have told earlier, all these three components means a farmer, his crops, and the factors affecting the crop yield, and then selling of the product. All those is important for the farmer. Again,
Okay.
as you said, environment, economics, and society, everything in it for a farmer.
Okay, okay. I think I think I just will maybe elaborate a bit more. I think if I'm a farmer, for me environment mean जिसका मतलब मुझे फायदा होगा, मेरी फसल की health कैसी है, मेरी फसल का जो है health is affected by again things which are coming out of environment. So these pest diseases, humidity, temperature, because that have effect. So if my technology or my innovation is going to address a challenge around the farm, so environment is a more like a public property. So we call it as a public good. But sometimes farmers are researchers. We are a bit we take a blind view to that these things are for granted. So we do not take those consideration. So I think important element, because you guys are from the social from the STEM subject. So I think my innovation have to protect my environment as well. So this is the key that we always use in the UK as a yardstick. So is it environment friendly? So if I'm suggesting a technology which is going to use tons of chemical to control one problem, so where is this chemical going to end up? Of course, in the environment. So it's going in the air, it's going in the soil. When it's going in the soil, it's affecting the microbiome in the soil. When it's going up in the air, it's affecting the air quality. When it's going, I would say, with the water, so it affects the water as well. So which is an important resource. I have actually another slide which I just want to show, because I was just rambling about the subject of sustainability. So this is more sort of bigger view. So economy is profit, which is always meant for a farmer, but which is like a micro view. So I would like, if I'm a farmer, I just like to see what benefit is to me, so increasing production, making more money out of it. But the benefit for the environment is more like a public good, which is of course it gives benefit to the farmer, but it's a holistic. The farmer has to think about it, as somebody mentioned, permanent. So keeping or conserving things. So if I say, okay, I want to leave for my next generation a lot of assets. We always think about properties, always think about cars, but we never, if I'm a farmer, I would never think about the resources, which is water. And you will be surprised to hear that Pakistan is one of the water scarce countries in the world. Now, I'm actually at the moment I'm in Saudi Arabia. I mean, Saudi Arabia has a worst water crisis. I mean, these places are some places they don't have rain. So Pakistan is blessed with water, but I think the amount of water that we are abstracting from ground. So this is going to be a quite challenging thing. And then the benefit for the society. Society is looking at food quality and something benefit in the. I mean, there are places. I mean, even I think I'm not sure in Pakistan, but in the UK, if you buy a meat from a certain region, it's a high price. I'm sure in Pakistan there is a like basmati. अगर आप कहते हो गुजरावाला की बासमती है तो उसकी प्राइस कुछ और होगी बजाय इसके वो जो बासमती पैदा हो मुल्तान में वो मुल्तान वाले भी बासमती होगी लेकिन गुजरावाले की तो वो सोसाइटी का एक बेनिफिट है तो again something with very specific to the society. So I just mentioned here climate change, water, salinity, health, food benefit, pest disease threats. So this is more like a bigger challenge that society is facing. So, but price fluctuation. I mean, जो आप पाकिस्तान में हैं, हम पूरी दुनिया में देखते हैं तो अगर पाकिस्तान में price बढ़ती है, तो लोग सड़कों पे आ जाते हैं. It's a global thing because again, if the farmer who's if your innovation is going to help the farmer to produce crop more expensive, is this mean it's going to be more expensive for the society? But if your innovation is going to help the farmer to produce more crop but cheaper, high in its quality. So I think quality should not be compromised. I think this is where the biggest question is, and how much chemicals are used there. So that's something have to be kept in mind as well. So I think whenever we're looking at any innovation around the farming sector, or in the food sector, or I would say in the life sciences. So these three things are paramount. So we have to look at them for their efficacy. So I'm just gonna stop this one and come back to the original slide, which I was talking earlier. So and I think the other element which is important in this debate of innovation is this doable? Is something we can do? I think one of your colleagues mentioned earlier. Here in the institute where I work, we have developed a technology, but that requires it's something is about nitrogen. So what is innovation? So innovation happened. So nitrogen inhibitor technology, but that would require 2 tons of that chemical. That chemical is basically a plant-based product that you can use. But that's something is in scale-wise, it's not doable. I'm going to the next slide. Thank you. When someone improve, the next slide. Okay. So various sayings from eminent people, especially Thomas Edison, who you all know is one of the innovators who done interesting innovation. He said, there is a way to do it better, find it. So there's always, it's a continuous journey to make improvement the way we do our. So I think the whole, and we all know the famous the last one was, there's always room to improve it. Do things in a different perspective. So I give you example of Phage was quite popular many years ago. Phage tha aapka jo camera aap lete the, Sony was popular. But I think these guys did not do much innovation. They would thought we have done one product is sufficient to go forever. It's not the case. So Phage is nowhere now in this camera technology. So or maybe they are now going into different perspective. Motorola, these companies, Ericsson. So these guys were just wiped out because the new product has taken their place. And that product has again a shelf life. We call this each product, each innovation has a shelf life. So shelf life is how long it can continue. The one thing in the scientific world, we use this patents. So you can patent your product, but I think this is again, patent cannot be granted forever. So they're only granted for 20 years, but it costs you money to maintain them. So once the patent is filed, you need to exploit it and get to the market. Otherwise, after a few years, I mean, I think there's a lot of pressure now. So companies are saying let's bring it to 10 years or maybe short duration. When you file a patent, it takes you 3-4 years before it's been granted. So first 50, so if it's 20 years, 5 years are already gone. So you have only got 15 years. So now there's a lot of pressure that we should reduce it to the next level so that the new companies could jump in. So this means you cannot protect yourself. So this is a continuous thing. We all know that Sir Albert Einstein, he said we cannot solve our problems with the same thinking we used. So the previous thinking or the mindset cannot continue. So we have to create something new. So we have to keep on looking at the new explorations. And we all know the famous saying from, I think I'm just going to the next one here. And the last bit here is, we all know Steve Jobs, who was the innovator of, who invented, or I would say, I would use the terminology, was innovator, who helped us to develop the Apple phone, Apple Watch, and all these Apple, I would say. So he said innovation is the ability to see changes as an opportunity, not a threat. So it could be a new opportunity that could help to solve the problem in a way. I'm just going to maybe stop for a moment, maybe ask again the group. So sorry. Just maybe. So some in. Sorry, I'm just going. So innovation. Okay, I think I'm not going to use this recorded version because it's just annoying me. So just want to ask here the group. Maybe you guys could appreciate me. So maybe give me one example of an innovation in Pakistan which was just on the top and then it's just gone to ground. Anybody can give an example, please? Yes, please. Anyone have any example, please? Gigi, please. Any story for Pakistan or any
Different type of cameras, different type
Gigi?
of microscopes. These are the ideas. Yeah.
Sorry, please.
Okay. Okay. I'm just saying about that anything which was top and gone down.
Okay.
Pakistan. Sorry.
शूज हैं जो एक स्टूडेंट ने बनाए ब्लाइंड्स के लिए, उसमें सेंसर्स हैं, डिटेक्ट कर सकते हैं, उसको राइट पाथ पे गाइड कर सकते हैं, लेकिन ये है कि गोइंग अप एंड डाउन, आई डोंट नो अबाउट दैट. इन्वेंशंस हैं, प्रॉब्लम्स का सॉल्यूशन है फॉर द ब्लाइंड पीपल.
Okay, okay. That's very interesting, Gigi. But I think what do you just an innovation किया था again? Innovation का जो एक Gigi, please. Yes, ma'am.
I want to add something about BT cotton. You must have heard about it because you know that Pakistan is an agricultural country. So our economy mostly depends upon the products which are we get from the agriculture. So BT cotton, I think this is an innovation in Pakistan. Still, the farmers, they are growing the BT cotton in different areas where cotton is growing. Hello.
I think BT is interesting, but I think that's again we call it, you go to you set up a quite high milestone. So we missed something in between as well. So now there isn't any further improvement going from here to above. So the BT was I would say, but I think this is where the things are now progressing as well. I think that's an interesting example, but every innovation has a peak. So it goes, .com has a peak, and then it's came down as well. So I think the point which I'm trying to say, and again, in the research is there's always a gap. So we cannot simply say, okay, this one, okay, Apple is end of the world. No, Apple is not end of the world. So there are things which are still coming on. And again, small companies, they're not making. So there's terminology we use, open innovation. Have you known anybody open innovation? What does it mean, open innovation? Anybody want to say open innovation? Yeah. Okay. Ma'am, anybody else? And this concept is now used by the companies. The large companies, multi-million companies, Google, Facebook. So they do not say, okay, I mean, how many people they can employ. There's always a limit. So you can have maybe 1,000 people, 5,000 people. But in a daily job, your mind is doing things which are you asked to do. So you are confined. But if you're looking something outside the box, so it's always, this is where these innovation companies are saying, giving their employees these a lot of open and flexibility. Workspace, you go and sit in a cafe type environment, relaxing environment, where your mind would start thinking about those ideas. But again, there's a limit. So they say, okay, instead of us investing on innovation, let's let the public to come up with ideas. So the companies say, okay, we would be open to take ideas from outside. So they organize events, they organize workshops, they organize those opportunities and engage with the public. Public means researchers, with the universities, which are not on their payroll. And their innovation, they can pick it up. So now, if you look at Apple may be doing its own things around 40 to 50%. 60% they're outsourcing stuff from others. So this outsourcing thing itself is called open innovation. So which you have opened the doors for others to contribute and bring it together. So it's like a funnel thing. So you let ideas, shift them around, and then bring it to something which is useful. So we where I work in this institute, we I think 3 years ago we arranged a big event, open innovations. We get, and again, this is a bit of a marketing word. So there is not any word which is open. So open is always coming with somebody who owns it. So this is where I think the concept which universities and academia could promote is problem solving. So when the problem solving could be done by the students, I mean, this is where in the Western world, we use a lot of time feedback. So when you get a feedback is basically taking those ideas and then you fine tune your product. So the open innovation is basically this taking this feedback approach, getting others to contribute into your product's attribute. So which is again a good way to look at innovation that could make a difference as well. So I'm here actually at the moment spending some time in Saudi Arabia. One of the big universities called KAUST. So KAUST is a university which is established by the kingdom back in 2009 or 8 or 9, 10, but it was operational only 2013. But in less than 10 years, this university has become the world's second largest or second top tier university in terms of ranking. And what they did, this open innovation, they just let the word to come. So they, okay, you have any idea, you come to us. So I think that's a fantastic example. KAUST is an example of open innovation. So they got people from Japan, from the US, from the UK, Germany. So they pick them up, bring them together. And then when these minds sit together, the ideas generate. Another example I just want to give you is a bottle of water. So I think we use this terminology. I don't know how many of you know interdisciplinary. So can somebody say what does interdisciplinary mean? Anybody would like to volunteer? Interdisciplinary research or the ma'am with a black, please. Sorry, my color recognition is just gone dim.
When more
Yes.
than one departments are cooperated and in the collaboration they have to work, then it is called interdisciplinary.
Excellent, excellent. Any example you want to mention?
I think interdisciplinary research means the integration of two different disciplines into a single research purpose is known as integrated one. The example is, let's say, the scaffolds that are the replacement for the knee cartilage. These scaffolds are made by some engineers, and then they are biologically tested and biologically filled with the cells by some biologists. So this type of research is known as interdisciplinary research, I think.
Excellent. Anybody else?
One of the example of interdisciplinary is the stethoscope. Some physicists actually invented them, and in medicine, we are using the stethoscope.
Okay, that's very interesting. Yeah. Anybody else? So I think you have explained. So we just give you example, this water. Water is a discipline itself because water is inside. I would say maybe just simple water without using this bottle, I would say. So, and then the next thing is, I would say, maybe water is not a good example. I'm just gonna, okay, I'm gonna, is apple. Apple is a discipline, one discipline. So you chop this apple and you cut this into a fruit and maybe mix some other fruit like maybe, I would say orange. So this has become a bit of we Pakistan में हम इसको क्या कहते हैं fruit chaat कहते हैं fruit chaat बन गया ना fruit chaat बन गया so fruit chaat means it's much tasty, it's more appealing as well rather than just apple, so it's more appealing. So when you bring two disciplines, so things get quite interesting. So when you take to the next level, so ये apple था ये orange के साथ आपने banana mix किया, you make a milkshake, ठीक है जिसको या smoothie कह लें. The smoothie is something you can carry along. So you can put into the bottle and you can drink it when you need it. So उसकी क्या होगी कि ये apple की जो value थी वो increase होके gone to the next level. तो उसमें क्या होगा कि now this the apple and orange and the banana, these three things have come together to provide a solution to one problem, which is thirst or energy, or if you are going to do a marathon, something you want to keep with you. So that something is interdisciplinary. So you bring various disciplines together to come up with a solution, which is a smoothie or a juice. So I think the science where it's going at the moment is interdisciplinary or transdisciplinary. So where you bring sciences together. So otherwise, the guy who's working in a computer science department, they were just doing at maybe modeling. But now these computer science people are working with a biologist, and they are working with a mathematician. So they are coming up with a solution that is transdisciplinary, which provides solution to the next level. And I think this is where the world is going. And if you now do any funding proposals in the UK, you need to have always people from different disciplines. Not only science, you need to have people from the social science. So you have done a fantastic work. Your product is fantastic, but it's not being communicated. It's not coming to the means to the public. So you need to have sociologists. You need to have economists. It's cost effective. I think this is where things are going. So okay, I come back to again my talk. So I'm just getting distracted, guys. So please, if you have any questions, just raise your hand. Any comments? Anybody want to say anything here on this subject, ma'am?
Hello, Assalamu alaikum. Sir, I want to add on that I think applied sciences or applied research is interdisciplinary. I mean, if you want to apply an idea and you have gained some product, then obviously without the involvement or even for the formation of the project, without involvement of other people, you can't complete it. For example, if there is a waste management system or waste treatment system, you need engineers, you need environmentalists, you need the study of the hydrology and the water sciences. So multiple people can work together to make something effective. So this is again interdisciplinary. This is applicable to any applied area. So if the innovation, the thought is converting into the wealth in terms of the applied product, then we can say that everything we are producing is interdisciplinary, because without the involvement of other people, we can't complete it from the other disciplines. I have worked on the hydrogen-based membrane biofilm reactors for the waste management, and then we were having the mathematicians and then engineers and then obviously environmentalists and microbiologists. So these four sciences, they work together to make something effective.
And now I have also produced one PhD student, and she has been working on the bioflocs. So in the zoology, we have connected the microbiology and the aquaculture, and we have make a sustainable technology. Biofloc is well established in the Malaysia, Indonesia, and other countries, but in Pakistan, it is getting fame during this decade. So we have also reduced the water loss. We have managed the toxicity in the aquaculture. So with the least consumption of the water, and by using the cheaper wastes as a food for the fish as well as the microbes, we are making the fish. We are growing the fish. So these were my comments with respect to the innovation and interdisciplinary sciences. Thank you.
That's wonderful. I think I love it. I think this is could be a case study. So bringing people of different disciplines, and again, when you bring these people, you bring expertise, you bring the human side into it. So we use this terminology in the UK, and I think something is quite famous in the development sector. Human-centric approach. So when a HDC, so when you are human development centric approach, whenever you are developing a solution or a technology, it's good that you test with the people who are the end users. So see what would be their view. So what benefits it's going to bring to them, which pains it's going to resolve, the challenges it's going to resolve, what would be their convenience as well. So product की जो availability है, कौन से problem को resolve कर रही है, this is fantastic. So I think this is where the innovation have to bring that focus in mind. And I think 10 years ago, people were just looking at silos. People are looking within the department, people are not talking to each other, even within the department. And I think now it's more that you guys talk to mathematicians, you talk to physicists, you talk to people in the social. I think you need to have did you bring ma'am a sociologist as well into this project?
I think I don't remember. I was working with the scientists, and they weren't included any sociologists that time. And even in my PhD student, I think we haven't involved any sociology, but ultimately if it is going to affect the society and the end users, obviously. So जब हम इसको patent करेंगे, तो I think sociologists will also be needed.
I think this is important bit. I think जो sociologist वाला role है, sociologist वाला बड़ा critical role है. So yes. I think that something must be built in.
The society accept it or
exactly. Okay. Perfect. Thank you. So I'll just quickly go back to my. So we call it this innovation could be in anything. There are few examples there in a process. So for example, enabling process, core processes, technology-wise, in terms of its offerings, its performance, product system, services, and if it's a delivery, so how you deliver your product. So something is delivered through distributors, something delivered through online, something delivered through, I would say, peer-to-peer. So like these lectures, could be innovation as well. So instead of having a physical, you have virtual session. This is a new way of delivering things. In terms of finances as well, so is this innovation going to be creating a new business model? Who's going to pay for it? We always use this terminology, big data. Big data is an interesting example. So Google knows now what we need to do. So Google knows, I mean, sometimes I park my car and says, this will take how many minutes to get to home. So because it takes your location and send you next information. So it's a new model. That information is providing some kind of maybe when my location is shared, maybe it's going to the Tesco or my local store. And then when I'm opening up my computer or my app, it's showing me some, I would say, offers from Tesco store because I'm quite close to Tesco. This is a new business model. So new way the things have started moving in. And then we call this networking as well. So I just give you some examples. Okay, so next. So innovation journey. How does it work basically? So innovation basically is a mix of creativity and you adds on commercialization. So when these two things come together. Okay, so I think we touched on this already. So we just need to skip to the next one. So together. So some examples of innovation in the past, you say how inventions have helped to deliver innovation. So we all know this wheel thing. This was many centuries ago used for grinding grain to make bread. So now that's been used by everything in this world, I would say, in terms of that runs on wheels, by cars, by aeroplanes, by turbines. So that wheel concept is used in a variety of functions. Here is the processor, microchip, which was maybe originally developed for a computer, but it's now used by processors all the way from sensors and opening up keys, a car, using your sensor fob as well, so which has got I would say this motherboard. And use of laser light, it's not only sort of used for fancy reasons, but it could be used to a number of things, from printing all the way to doing cutting edge, I would say, the work in the manufacturing. Okay, so I think we have. So innovation took many years, sometime, yeah. So we all know this is example of mobile phone. So I think I need to skip this one as well. So how and when and where the innovation start? So it's always start from the conception, so from an idea. So what is the need someone is trying to help? And once that idea has been tested, idea is generated, it's been worked out, planned through, then you are thinking about how you would like to implement it. So in terms of product development, product testing, we call this prototype testing, prototype development, and prototype application, pilot application, and then testing. So one is tested, then you like to take to the next level and bring it to the market. So when you are looking to bring in the market, it will again require production, and the market would be tested again and see it's maybe it's local product starting in one particular area and then take it to the next area and then maybe countries nationwide and international as well. So 3 steps there: conception. So anything you want to add here, guys? Gigi, any comments, any suggestions on this slide? No? Okay, then we continue. Because I'm just conscious of the fact I think we need to, there's new lecture starting soon, so I need to wrap this up for you guys. Okay, so the next one is the process. So sorry about this, is quite complex chart here, but just the whole idea is to just to say it always start big. So the new product idea always have many things that you would like to bring in, but then it's been cut short in the product concept stage, and then it's been tested in a next level, and when it's tested and it's just been going through a bit of a funnel approach. So coming down to when this idea is when idea is new, it's always the early adopters who adopt it. It's not everyone. So okay. So I think so. This is a bit more detailed. I would say innovation journey itself. So where does it start? We always say it's like a funnel approach. It always starts big. So you're looking at sort of the bigger market, where the idea would go, where the innovation would go, and then you trim it down to the next level, and then you go through the process. Okay, I cannot do this, I cannot do this, maybe bringing it to the next, I would say, precise level, and then maybe you original idea was maybe 10, 15 different attributes, but then when you bring it to the, I would say, the final stage, it's focusing on a very particular advantage that it will deliver. Sure. So I think the important bit here, which I was going to say, I think because of this video is creating me a bit of, I think one thing I just want to explain here, जब हम टेक्नोलॉजी जब कोई आइडिया बना स्टेज डिफरेंट से गुजर गया तो उसको डिफरेंट स्टेज पे लेके आते हैं तो वी इन साइंटिफिक वर्ड you know what do you know about so just because you guys all work, anybody yeah, so any suggestion about TRL? जी TRL के बारे में कुछ समझते हैं, कुछ बताना चाहते हैं? What does TRL mean? Technology readiness level. जी, raise hand anybody, please. What does it mean technology readiness level? मैम जो हाइड्रोजन की जो बात करी थी, आप बताइए प्लीज जरा.
Okay, sir, you're talking about basic technology research.
It's a technology readiness level.
Okay.
This is something
So obviously, the one thing which is first study at the bench level in the lab scale, and then obviously the pilot scale, and then it goes to the industrial scale. So among these scales, there are different levels. So we we do research, then we make something, and then we apply, and then we take the feedback and see, and then obviously it is ready to go to launch. I think this may be telling about the technology readiness level that is now perfect to go into the market or to get commercialized.
You're right. You're right. I think we call it levels up to 9. So from 1 to 9.
Okay. Yes. So as you mentioned that hydrogen based. So it was the USA lab, and then they were working treatment plants of water, and then we got the sample and we got the problem from there, and we solved in the labs, and then it was the solution were given to the directly to the treatment plant. So I think that was level TRL 9.
Okay, so if it's something which is coming out of the lab, which has been tested, it's been
it was applied, and then there were certain problems. So the problems come to the labs, and then we do the research and get the solution to the problem, and then those solutions were given to the industrialists. So,
so I would say if you are doing some work on a theory level, so if it's something as a still research, master student is doing research, and then a lot of work is done to test maybe certain attributes of a chemicals without knowing its benefit to the bigger problem. So I would keep that, this would be seen as under TRL 4. So anything which is research, primary research, which is, if it's a blue sky research, we call it TRL 1. So you don't have to bother about market. You're just looking at the the scientific side of it. But when you are bringing to the TRL 4, when the idea is tested, this is something is a proven principle, then it's going into this into this staircase. I would say next level, technology development, prototype, technology demonstration, looking at on the next level, subsystems development, and then something which is now a product which has been tested on a small group. It's now available for bigger people. So that would be a place which is still not commercialized, but I think this is, I would say, TRL sits between TRL 8 and 9. Okay. So yeah, so once you have done a product, how you would like to bring it to the market. So we all might have heard this this acronym, or this is a quite common terminology used in the business world. We call it a business plan. So you have an interesting idea. Do you have a business plan? So business plan, what does it mean? So how? Okay, यहाँ पे मैं stop करूँगा. People, you know any business plan? Business plan की terminology को कौन जानता है? जरा हाथ खड़े करें. जी मैम, हाथ खड़े के जो business plan के बारे में जानते हैं. How many of you know about business plan? Please raise your hand. जी जी, business plan के बारे में. In front row, यहाँ पर एक white shirt में डॉक्टर साहब बैठे हुए हैं. मैं इनसे पूछना चाहता हूँ. I'd like to ask you, sir, आप बताइए business plan. What is a business plan? Business plan.
Thank you, Dr. Sahib. I am Dr. Tahir from Punjab University. Actually, you are talking with a business plan. So in scientific terms, as you explained that if we develop something new technology or new inventions, so we test in different levels, and then we transfer into the society or a market, but we need to the some persons which is from the marketing, like economics or business. So they help us because as a scientist, we are not familiar how we do the business. Sometimes we make something in the lab, but unfortunately, due to some hurdles like testing agencies, some government's lessons, and also the big markets which have already working business in the society. So it's very difficult to sell our products in the market. So I think it's different aspects because the economics are a little bit different, but in a as a scientist, it's a different things.
Okay.
So I have
Thank you.
ideas.
Thank you. I think this is the thing. I think the whole idea which we are saying is exactly so. The scientist is good in science, but when this science has to develop a product or a service, then you think about a business plan. So business plan, what sort of components it has? It's always just a second. I think just maybe I'll just yeah. Yeah, so once you have done a product, how you would like to bring it to the market. So we all might have heard this this acronym, or this is a quite common terminology used in the business world. We call it a business plan. So you have an interesting idea. Do you have a business plan? So business plan, what does it mean? So how your idea is now being materialized and it's going to deliver benefits for the society, and what would be its cost, and who's willing to pay for it, and what would be the revenue model, and who would do it, who would deliver it. So it's like a some kind of X-ray of your idea. So it has typically 7 components. So one component is executive summary, so which gives the headline about the business plan and the idea that you are trying to deliver. And then it has market research element into it that covers a bit of research. Second is defining product and service. And then the third element is strategic analysis, we typically call SWOT analysis. So that's carried out. And then marketing plan is worked out. So who's going to benefit from the product and how you're going to sell it. And then operation plan, where the product will be made, where it will be delivered, some kind of distribution plan, and is it something a soft product? Is it something digital platform? Something available on internet or on the app? So those kind of things. So who's going to do it? Team, typically. I'll go in more details in next slides, and then financial plan as well. So I'll go one by one of them so that you can this. So first of all is the executive summary. So executive summary is explaining the key innovation of your product. So or a service that you are trying to bring it to the market. So it should have a bit of mention of what is a USP. USP we call unique selling point. So what is additional feature this product is offering compared to other products, and who are the key customers of this product as well, and maybe a bit of a market, how big is the market. So, and also these days it's important that whenever we are trying to deliver a business, it should have a component of if it's addressing the societal challenges. So it's poverty, is it something, I mean, I'll just go in more details. Sustainable goals, I just have 17 of them here. So we have to see our product is able to benefit the society in the longer run, and this will improve reduce poverty in the area, improve if it's any bioproduct or something which is as a health initiative, something that can help to address the issues around health. If it's a new tool that help to address education gap, so that can help improve literacy or improving the quality of education, improving the gender issues, and important element here is water. So if it's something is around climate change, addressing the use of water, and clean air, clean energy as well. And so these are the 17 goals. I won't go in one by one all of them, but I think this is important that whenever we are thinking about that this product is going to benefit, so what benefit it's going to deliver to the society, to the societal challenges, to the global challenges we call these days. We call this terminology global challenges. It's been used in the UK a lot. So actually in the UK they have a program, Global Challenge Research Fund. So that was basically focusing on the UN Sustainable Goals. So we all know the climate change is happening and the issues around net zero target that we have to achieve. So greenhouse gas emissions, water. So Pakistan has a perfect situation of all these problems at the moment. Again, this is the global problem. Energy prices and air quality and all these things. So we had recently the issue with COVID. So COVID itself was a UN fits in the UN Sustainable Goals. So it was one of the issues that has caused the global challenge. So how we do it? So first of all, in the business plan, the important element is you do the market research. So what is the size of that market? So if I'm going to bring a vaccine for COVID, so I would have to think, okay, what's the bigger market? So 7 billion people, 8 billion people, 9 billion people, and how many will get affected? So if my vaccine is going to be different than others, so what USP it has? So it doesn't give you a fever, it doesn't give you any side effects, all these things. If those benefits are proven scientifically, then you can claim it. So, but I think this is something you have to do a bit of market research. So how big is the market? Our government regulations are there. As a freedom to operate, can you deliver something? Is it something safe to the society, safe to the children, safe to the special segment of the society as well? We call sometimes if you are in a Muslim country, you are delivering something, have to have a halal as well. So that's just one example. So the next one is you define your product and service. So what attributes that product carries in terms of its benefit to the end user, its convenience, it's better than others. So we always look at what is the unique selling point, USP. And of course, when you are looking at this product, so if something you are making it, is it something will not be copied by others? So you look at its IP as well, so intellectual property. So you protect your patent. So usually, if you are publishing something, do not publish if something is new. So you have to do the patent first and then get it published. Otherwise, once it goes to the public domain, it's not be there. And usually the patent is protected for
Dr. Khalid, are you there?
years, I think. Yeah, so it should be, and they should be open afterward as well. So, and there is a cost associated with patent in the European market and the American market, global market, Chinese market, Pakistani market. So I think this is important that you think about that product protection as well. And then you look at SWOT analysis. So you might have heard this terminology is pretty common. So SWOT is strength, weakness, and opportunities and threats. So strengths are always so what strength that product is bringing along. So the business which will give advantage over other companies or other competitors. Weakness is the characteristics of a business which make it a disadvantage. So something could be always a worry for you if there's new innovation gonna come or something. Is not this is always a challenge because one product cannot always offer everything. You just have to just take which are the positive sides of your product. Opportunities, so it's an element in the company's external environment that allows its formulation and the implementation strategy. So basically you're looking at maybe just give you example of if in Pakistan these days a lot of people talk about CPAC, so the CPAC could be an opportunity.
Okay, sorry to interrupt you.
So the fourth element is called marketing plan. So you need to look at.
I think.
Dr. Khalid, if you please wind up it. We are getting short.
I think I'm done. So I'm just going to say quickly. I think I've just most of the things. I think the important bit, which is at the end, is the product has to have the price, marketing, and all these things. So I'm just going to stop this one. I have already shared this on the YouTube as well. You can watch it from there. But any question you want to ask? But it was such a good pleasure talking to you. And thank you so much. Sorry, time was a bit of a issue for me, but we managed. So thank you so much. And with that, big thank you and good luck. Assalamu alaikum.
Thank you so much, Dr. Khalid. Thank you. Ladies and gentlemen, we will take a quick break of 10 minutes and we'll get back at 12, inshallah. Welcome back. Ladies and gentlemen, our next presenter is Dr. Tahir Mahmood. He is Associate Professor of Molecular Biology and Biochemistry at Center of Applied Molecular Biology, University of the Punjab. He got Overseas Merit Scholarship from Higher Education Commission of Pakistan and completed his PhD in Biochemistry and Molecular Cell Biology from Institute of Genetics and Molecular and Cellular Biology, University of Strasbourg, France. He also enlisted as the Productive Scientist of Pakistan in 2017. Today, he will deliver his talk on nutraceuticals, a promising future for health and new drugs, recent development and challenges. Welcome, sir. Please proceed.
Assalamu alaikum. Thank you for a nice introduction. So today I'm going to discuss one of the important topic is the nutraceutical promising future for health and new drugs. What is the recent developments and the current challenges to convert these nutraceuticals as a nutrapharmaceuticals. So I'm hopeful you are familiar about these words, nutraceuticals. Those participants who are from the botany, from zoology, I'm guessing maybe some of from biochemistry or chemistry, you are familiar with this word nutraceuticals. So okay, so if we look the simple definition of nutraceuticals, these are the basically foods or a part of foods that basically provide medical or health benefits, including the prevention and treatment of diseases. These two things are very important to prevent and treatment of diseases. These two things are very important. We can use these nutraceuticals to prevent from diseases, and also if the disease happen in the human being particularly, then we can treat by using these nutraceuticals. Nutraceuticals are basically contains the functional foods. Nutraceuticals is that means they are perform nutraceutical functions due to the functional foods. And what is the functional foods? Actually, the functional food is a food that exerts health properties beyond the traditional nutrients that it contains. So these are the functional foods. So functional food ingredients are responsible to show these nutraceutical properties. Nutraceuticals has many advantages over the medicines because they have less side effect and they are very compatible to the human body because they are lesser or they have no side effects, so they are good for the use of the human beings. With the passage of the time, if we look, the nutrition science has been changed. Now, instead of traditional nutrition science, it is converted into optimal nutrition. So what is the traditional nutrition? It's consume food and nutrients that required for the growth, for the development and maintenance of body. These are called the traditional nutritions. What happens now? It's a move from the traditional to the optimal nutrition. In optimal nutritions, it's also provide the promote overall health, improve physical and mental performance. And the third one is important function of the optimal nutrition is reduce risk of diseases, particularly the cancer, obesity, and cardiovascular diseases. So we can prevent by using the optimal nutritions to prevent from these dangerous or very life-threatening diseases, including the cancer, obesity, and cardiovascular diseases. So if we look at the functions of the functional food, the functional food has basically three major functions in our body. The first one is to provide the nutrition, means supplements for the human beings. The second one is the most important is organoleptic or sociocultural properties, because we are familiar some dishes, some food. So these are the organoleptics that are it's commonly used in our society, like Pakistan. And these two properties are known as a traditional food. And if we look the third one, added value, which is the basically health benefits, these three are collectively known as our functional foods. So functional foods now is important to provide the prevention and treatment from the different diseases. If we look, so many years back, the founder, which is a Greek physician, Hippocrates, he knows the father of the medicine. Several centuries ago, he said, let your food be your medicine. Means कि आप जो अपनी खुराक है उसी को आप दवा के तौर पर, मेडिसिन के तौर पर यूज करें. So this philosophy actually contains the prevention is better than cure, because if we use some specific food, so they can prevent from diseases, and that can prolong the human life, and we are more comfortable, more physically fit and mentally fit if we use the good one or balanced food. So functional foods started particularly in the Japan. Japanese are the responsible to develop these functional foods. In 1984, a Japanese ad hoc research group under the scholarship of Ministry of Education, Science and Technology started a large-scale national project to explore the interface between food and medical sciences. So a category of food means food for specified health uses was established in 1991. So the first time term functional food used in 1993 by the Japanese, and they published paper in a Nature News, and with this headline that Japan explored the boundary between food and medicines. So they have find out what is the components they are act as a food, and what are the components they act as a medicine. So they explore the first time these boundaries that is present between food and medicine. Here in this diagram, you can see the function foods. If we look the function foods, so major source of function foods is the plants and fruits. These are the major or vital source to provide the function foods and nutraceuticals. Here you can see the many examples from the different plants like guava, orange, grapes, and aloe vera, and different plants or fruits are responsible to provide the function foods and nutraceuticals. So here, if we look a bigger class of the compounds that are isolated from the different sources, and they are the plant sources, vegetable and including the fruit sources. Here you can see the class of compounds. For example, is alpha carotenes. It is obtained from carrots, and its function is to neutralize the free radicals which may cause damage to the cell. So this beta alpha carotene come from the carrots. Like other compounds, the beta carotene, these are also the source from the color fruits, pigments, orange and yellow color fruits, vegetables, and this also provides to neutralize free radicals and which can cause that prevent to damage the cell. The other compound is a lutein. This obtained from the green vegetable, reduce risk of prostate cancers, reduced risk of muscular degenerations. And another compound is a lycopene, which obtained from tomato products, and it also reduced the risk of prostate cancers. So another compound class of compounds, which is a phytosterols and also stenols, esters, they are obtained from different sources. You can see from corn, wheat, vegetables, and they are also responsible to lower the blood cholesterol levels. So another class of compounds which can obtain the omega-6 and omega-3 fatty acids, DHA and docosahexaenoic fatty acid. These are obtained from different seeds like flax seeds, it's hemp, canola oil, and some other fish. They contain the omega-3 and omega-6 fatty acids, and these are the responsible to reduce the cardiovascular diseases particularly, because they can reduce the cholesterol levels and also the bad fatty acids which is present in the blood. So another compound of the class of compounds, which is the conjugated linoleic acid, that's obtained from the meat, cheese products, and these are also responsible to reduce the cancer. So these are compound of classes which can obtain from the plant sources, from fruits, from vegetables. So these compounds are very useful to prevent from the diseases, and if disease happen, so we can take in a large amount, they can use as a treatment. So here you can see another class of compounds is tocols, is tocopherols and tocotinols. These are the big class of the compounds which can be isolated from seed oils such as the canola and sunflower and some other cotton. So these are the responsible to prevent the oxidation process that happening in our body. They work as an antioxidant and increase the efficiency to work for the heart and good for the skin. So another compounds, a big one compounds class is the phenolics and polyphenols. They are also present in a large amount of plants, vegetables, and in different sources, particularly the nature sources is the plants, fruits, vegetables, leaves. So they contain the polyphenols And these polyphenols are some of polyphenols are mentioned as caffeic acid, fuluric acid, camaric acid, vanillic acid. These all are the polyphenols that obtained from different sources like tea, wine, olives, chocolates, berry fruits, etc. And these polyphenols particularly act as antioxidants, and they are good to prevent from the cancer, and they are also good for the heart diseases, good for immune systems, and they act as an antimicrobial activity. These are reported in our literature. These polyphenols are working efficiently. So another compound is lignans and tannins. These are also obtained from different seeds like flaxseed, cereals, soybean, cabbage, and these contain the class of these compounds, and it can also act as antioxidants, and their functions is possible to prevent from the cancers and act as a radical scavengers which are generated in our cells. They can bind free radicals and prevent cell damaging. So another class of compound which is biopeptides. These are the basically small chain of amino acids. They are also present in different vegetables like bean, wheat, rice, palm, kernel oil, palm kernel, etc. And these act as antioxidants, and they are good for the immunomodulator activities. So another class of compounds which are isolated from different plants, from vegetables, from fruits, they are the prebiotics and probiotics. These are also extracted from different grains, onions, and fortified foods, and they are also responsible to improve quality of intestinal microflora and gastrointestinal health. So these all are the compounds, different class of compounds which are isolated from different sources. They act as a nutraceuticals. So here, the one slide, I want to explain you how you can extract these bioactive compounds which are present in different sources like vegetables, like plants, like fruits, berries, and different like citrus family fruits. So you can extract the bioactive compounds by using a number of techniques are available. Some are conventional techniques, and nowadays some are modern methods are available to extract these bioactive compounds. So first of all, you select the source from where you want to extract the compounds. Then that source can be identified by the botanist. Either you are having a same species or anything you want to extract. Either it is a right one or it's not, because there are a lot of species are available. So you can identify it from the botanist, and then you dry that materials, and then you can use number of solvent for the extractions. These are the conventional methods, still is a good method, although now it's a little bit creating problems for the environment, because these solvents, when evaporates, goes into the environment, they have a problem. Now we have some other green solvents which are commonly used to extract these bioactive compounds. So you can choose the solvents on the basis of nature of your compounds. What compounds you want to extract, so you can choose the solvent. Either you want to the polar solvent, or you want to non-polar solvents, or maybe the green solvents like carbon dioxide, water, glycerine, or some other methods. Now it's called the hydroponic media is available that you can use and extract the compounds. So choose the solvents. It's depending on the nature of your compounds. Either they are polar, either they are non-polar, like a methanol, ethanol, chloroform, or maybe you can use the N-hexane, pet ether. Number of solvents you can use depending on the nature of your compounds and your interest, what you want to extract basically. So normally you can dry the material, and after dry material you can add the 1:10. You take a 10 gram of your solid grinded materials of plant, and you can add 100 ml the solvent and keep it 6 to 8 hours on the orbital shaker. And after that you can basically pooled samples, you can extract 3 times and pooled sample, and then you can basically evaporate the solvents under the using the rotary evaporator under reduced pressures, and you can recover the solvents and extract the crude extract, and that crude extract you can be identified by the HPLC and some other instruments. You can check either your interest compound is extracted or not, and then you can check the activity. That compounds are biologically active, they have some functions, so you can use the number of techniques which are available to purify your compounds. So purification is also very crucial, time-taking, very laborious work, but nowadays some advanced methods are available like HPLC, LCMS. You can use that advanced method and purify your compounds and identify structure by using number of techniques which are available like HPLC, GCMS, MS, and NMR. You can identify the compounds. So another class of compounds which are isolated as from the different vegetables, from fruits, peas, from flowers is called essential oils. Essential oils are the natural aromatic compounds in the seed, bark, stem, root, flowers, and some other parts of the plants. These essential oil is present in a large number of flower as well, and some plant leaf also contain the essential oil. जिस तरह आप देखते हैं ये धनिया है, पुदीना है, ये जो मुख्तलिफ families हैं, they have some smell and that contain the essential oil. So this is another big class of the compound which are present in the plants, and they act as medicines. So it gives their distinctive smell and provide plants with the protection against predators and disease and play a role in plant pollination. Essential oil जो है वो plants में basically involve है, उनको protect करता है from the insects, from the diseases, and that essential oil we can extract and use for the human beneficial plant. So essential oil are basically non-water based phytochemicals made up of volatile aromatic compounds. So these compounds are the class of compounds which is a terpene, terpenoids, hydrocarbons. These are the essential oil which can be extracted from different sources, and it can be used as a nutraceuticals. So they contain true essence of the plant it was derived from. Essential oil are highly concentrated. It does not contain any artificial substances unlike perfumes and fragrance oil. ये जितने परफ्यूम और फ्रेगरेंस ऑयल हैं इन सब में जो है वो अप्रोक्सीमेटली 1300 केमिकल यूज होते हैं, और उनमें से सिर्फ 4 से 5% जो है वो नेचुरल सोर्सेज हैं, बाकी सारे सिंथेटिक्स हैं. सिंथेटिक केमिस्ट जो है वो यूज करते हैं, लेकिन ये जो एसेंशियल ऑयल होते हैं दे कंटेन ऑल नेचुरल्स, दे डोंट हैव एनी सिंथेटिक थिंग्स बिकॉज वी आइसोलेट फ्रॉम द डिफरेंट सोर्सेज. बहुत अच्छा ऑयल है. हम ये जो ऑरेंज है हमारे यहाँ, जितने भी ऑरेंज की स्पीशीज हैं उनमें बहुत अच्छी यील्ड है. ऑयल जो यूज होता है मुख्तलिफ पर्पज के लिए, कॉस्मेटिक्स के लिए पर्टिकुलरली जो लेडीज हैं वो यूज करती हैं, बहुत सारे अवेलेबल्स हैं, तो एक्सट्रैक्ट किए जाते हैं. दे आर वेरी गुड फॉर द स्किन हेल्थ. तो ऑरेंज पील हमारे यहाँ बहुत ज्यादा प्रोड्यूस होते हैं, तो उनमें बहुत अच्छा सोर्स है कि उससे हम अभी मेथड एक्सप्लेन करते हैं कि हमारे पास मेथड्स हैं उनके थ्रू हम ऑयल जो एक्सट्रैक्ट कर लेते हैं. एंड दे कंटेन अ लार्ज नंबर ऑफ important compounds which act as a medicines, वो ऑर्गेनिक केमिस्ट्री होते हैं, वो बेसिक इंफॉर्मेशन दे ऑब्टेन फ्रॉम द प्लांट सोर्सेज एंड देन दे कैन सिंथेसाइज इन द लेबोरेटरी. पाकिस्तान में सिट्रस ऑयल, ये ऑरेंज ऑयल बहुत कॉमन अवेलेबल है, कॉस्मेटिक्स में इस्तेमाल होता है. फिर उसके अलावा ये इस फॉर्म में भी डाइल्यूटेड फॉर्म में वाटर डाल के वो बेचते हैं. और लेडीज को बहुत लेवेंडर ऑयल और ये जो स्किन को ऑरेंज भी यूज किया जाता है. ऑरेंज ऑयल आल्सो वेरी गुड फॉर हेल्थ. ये आप जो पंसार की दुकानें हैं उनसे मिल जाता है और अक्सर लेडीज उनको मिक्स करके तो मुख्तलिफ रेसिपी. हाँ जी, उसमें इसमें ये भी एक फायदा है कि ये चूंकि हम जो वेस्ट मटेरियल्स हैं, चूंकि पील्स हमारे पास बहुत पील ऑफ होते हैं. हमारा ऑरेंज का आपको पता है कि पाकिस्तान एक ऑल दो बहुत बड़ा प्रोड्यूसर तो नहीं है पाकिस्तान, तो 10वें 11वें नंबर पे हम ऑरेंज प्रोड्यूस करते हैं. लेकिन फिर भी जितने पील्स होते हैं उनसे बहुत सारे एक्सट्रैक्ट करके, बिकॉज दीज आर वेरी एक्सपेंसिव, क्योंकि ये इनकी यील्ड कम होती है, नेचुरल सोर्स है, तो एक्सपेंसिव होते हैं. दे आर वेरी गुड फॉर हेल्थ. इनमें ऐसे कंपाउंड्स होते हैं कि जो हेल्थ के अंदर जाके स्किन में जो है वो जो एक्नीज हैं और जो ब्लैक स्पॉट्स आते हैं उनको रिकवर करते हैं. तो नेचुरल जो है वो थेरेपी है. इनको कॉस्मेटिक में बहुत सारे यूज किया जाता है, क्रीम्स में और बहुत सारे जो रेसिपीज उनमें यूज किया जाता है. तो जितने एसेंशियल ऑयल्स हैं दे आर नेचुरल, दे आर प्योर. अगर कोई ऐड करता है तो वो अलग बात है, लेकिन जब हम एक्सट्रैक्ट करते हैं तो दे आर हाईली प्योरिफाइड एंड नेचुरल. दे हैव नॉट कंटेन एनी सिंथेटिक कंपाउंड्स. जो परफ्यूम है हमारे यहाँ जो अब यूज करते हैं पाकिस्तान में भी और वन ऑफ द फेमस कंट्रीज फ्रांस, तो वो जो है उसमें सिंथेटिक जो है वो सारे यूज किए जाते हैं, एस्टर्स होते हैं और उसमें सारे केमिकल जो है मोस्टली सिंथेटिक यूज किए जाते हैं. So essential oils are a mixture of organic compounds. The terpenes particularly are a key component of all essential oils. ये terpenes जो है ये तमाम essential oil हम जब characterize करते हैं तो ये terpene जो family है, जो class of compounds की, ये सब में मौजूद होता है. So the distinctive character of essential oil can be attributed the functional group that is present in its की मॉलिक्यूल्स. तो बहुत सारे और भी जो है वो कंपाउंड्स मौजूद है, लाइक एस्टर, ईथर्स, एल्डिहाइड्स, लैक्टोन, कीटोन्स, अल्कोहल. दीज ऑल आर द फंक्शनल ग्रुप्स व्हिच आर प्रेजेंट, द कंपाउंड्स दैट आर आइसोलेटेड फ्रॉम द एसेंशियल ऑयल. If we look the methods how it can be extracted, so there are so many methods are available. If you can read any review article, a large number of methods, some are conventional, some are advanced methods are obtained to achieve or extract these essential oil. So if I want to discuss here the two most important methods which are using from many years in past, still it is famous in the laboratory, we can isolate these essential oils. So that is called hydrodistillation. This is the one method. What happens in the hydrodistillations? You can take the material from where you want to extract the essential oil. Either you from flowers like jasmine, like a rose, and some other flowers. If you want to extract from lavender, want to extract from a citrus family, जो उसमें lemon है, ऑरेंज है, रेड ब्लड है, बहुत सारी स्पीशीज हैं जो सिट्रस की हमारे यहाँ प्रोड्यूस होती हैं. यू कैन टेक द मटेरियल एंड जस्ट यू कैन here you can see put it the material inside the flask and you can add the water, dip the material in a water and then you can give the heat and when it evaporated, the water is evaporated, it can also evaporate the essential oil from those peels or the their just materials and it can be condensed. जो केमिस्ट्री के लोग हैं, बायोकेमिस्ट्री कंडेंसेशन प्रोसेस, इनको कंडेंस किया जाता है. तो कंडेंस के रिजल्ट्स में जो है वो जब कंडेंसेशन होती है यहाँ पे इन द प्रेजेंस ऑफ वाटर, इट कैन बी सेपरेटेड. जो कि एसेंशियल ऑयल है उनका जो डेंसिटी बहुत कम होती है तो वो वाटर नीचे बैठना शुरू हो जाता है और जो एसेंशियल ऑयल है वो ऊपर उसकी लेयर आए तो क्लियर लेयर आपको नजर आती है. You can extract by using this methods and remove the water, and you can collect the essential oil that is present on the surface of the water. So this is one of the simple methods: soaking, heating, and then you can volatile and condensation, and at the last step you can separate and you can characterize by using the number of techniques, and particularly the good one technique is GCMS. You can characterize the compounds, which can give you the idea about the compounds, which the nature of compound are present in the essential oil. There is another method which is also used for the extraction of the essential oil. This is called the steam distillation. In this methods, instead of dipping the sample in water, you can produce the steam. You can produce the steam, and this steam can pass the material in which you want to extract the essential oil, and that can be passed on to the condenser. Condenser always cool down the evaporated vapors and convert into liquid. And here you can see is the oil is collected here, and the down is the water. You can remove the water, and finally you can collect the essential oil in the steam distillation process. In this process, you cannot dip the material, just you pass on the steam and it can extract the essential oil. So if we look the modern uses of these particularly class of compounds that are extracted in the form of essential oil, here you can see the uses of these essential oil in different sectors. It's involved in cleaning because they have the antibacterial activity. It is also used for the flavor. Some they are good flavor. ये पिज्जा वगैरह में जो है कि यूज किए जाते हैं, they are very good in flavor. They are used in medical medicines. Insect repellent बहुत अच्छे हैं. अगर आप natural sources से लगाते हैं तो ये जो mosquito वगैरह ये उसके smell की वजह से repellent के तौर पर यूज, फिर परफ्यूम में यूज होते हैं. Although it's very expensive, लेकिन जो परफ्यूम afford कर सकते हैं, it can use in perfumes, and also it can use in the dentistry. Dentist ये जो मुख्तलिफ हमारे टूथपेस्ट यूज करते हैं, इन थाइमोल और बहुत सारे कंपाउंड्स जो है वो यूज करते हैं essential oil से. They have good smell and help to clean the material from our teeth. तो जो मुख्तलिफ जो टूथपेस्ट यूज किए जाते हैं, in particularly thymol compound, which is extracted from the thyme, they are used for the making of these things. जी जी, और बहुत सारे हैं इसमें, आप ज्यादा डिटेल में बहुत सारी इसकी लंबी जो डिटेल है, बहुत सारे प्लांट्स जो है मुख्तलिफ पर्पज के लिए यूज हो रहे हैं, कंपाउंड आइसोलेट किए जा रहे हैं और उनको जो है यूज किए जा रहे हैं नेचुरल सोर्स. And the one of the most important sectors of cosmetics, they are using the essential oil for particularly this cosmetics and the other soaps and some other sectors which are not mentioned here. So if we look the classification of the food on the basis of function food, so they are classified into 5 big groups. The first one is known as a conventional or basic food. Second one is a fortified food. ये आजकल आप देख रहे हैं ये बहुत ज़्यादा fortified foods जो मार्केट में अवेलेबल हैं. डालडा जो है वो वनस्पति है और बहुत सारी चीजें आप देखते हैं उनके ऊपर लिखा हुआ है कि fortified हैं. Then enriched foods, altered or genetically modified foods, and the last one is enhanced food. What are these different types of food and how they are classified? If we look, the examples are there, small brief definitions. The conventional foods are, it's also called the basic foods. They contain the only nutrients or bioactive components. They contain some nutrients and they have the bioactive components. They are called the conventional foods or they are called the basic foods. What is fortified food? Fortified food, basically food to which with the nutrients have been added in addition to what have lost in the processing. जब प्रोसेसिंग किया जाता है, मसलन अभी जो डालडा है, घी जो है, ऑयल जब प्रोसेस किया जाता है और फिर जब उसको हाइड्रोजनेशन की जाती है, उसकी ऑयल को घी में कन्वर्ट किया जाता है, ड्यूरिंग दैट हाई टेम्परेचर, ड्यूरिंग दैट प्रोसेसिंग दे हैव लॉस्ट समथिंग, जो उसकी न्यूट्रिशन वैल्यू है वो लॉस्ट हो जाती है, तो उसमें वो एडिशनल चीजें ऐड की जाती हैं, दैट इज कॉल्ड फोर्टिफाइड. मसलन उसमें विटामिन ए, विटामिन ई, विटामिन बी, ये आमतौर पर प्रोसेसिंग के दौरान इसका वैल्यू पहले भी कम होती है, ऑयल में प्रोसेसिंग में लॉस हो जाती है, तो जो सिंथेटिक्स हैं वो इसमें ऐड कर दिए जाते हैं, एंड दैट इज कॉल्ड द फोर्टिफाइड बनस्पति घी, बिकॉज दे हैव सम एडिशनल nutrients which contains. So what is enriched foods? Foods to which new nutrients or components which are not normally present, normally found or added. बहुत सारे फूड्स ऐसे हैं जिनमें बहुत सारी चीजें नहीं होती, तो उनको यूजफुल बनाने के लिए हम कुछ चीजें ऐड करते हैं, दे आर कॉल्ड द एनरिच फूड. एनरिच किया जाता है उनमें. अगर बस जैसे हम अब पाकिस्तान में गंदम है, आटा जो है उसमें आयरन नहीं है या और कुछ अमाइनो एसिड नहीं है, तो गवर्नमेंट जो है वो फोकस कर रही है कि आयरन डेफिशिएंसी को दूर करने के लिए हम उसमें उसको एनरिच करें, उसमें जो आयरन है या जो और अमाइनो एसिड है ज्यादा क्वांटिटी में एनहांस करें. Then altered or genetically modified foods, foods in which some potentially harmful existing components have been replaced with a beneficial components. कुछ foods ऐसी हैं जो जिनमें चीजें हैं बहुत ज्यादा लेकिन यूजफुल नहीं है, कार्बोहाइड्रेट बहुत ज्यादा है, ठीक है, तो कार्बोहाइड्रेट भी हमें चाहिए हमारी बॉडी के लिए, लेकिन हम उसमें कुछ और चीजें ऐड करें, ठीक है, उसको हम अल्टर करें, जेनेटिकली जीन लेवल पे जाकर हम उसके जीन में मॉडिफिकेशन करें. जैसे ये जो सीड्स हैं, जो बॉटनी के लोग बैठे हैं, जो सीड्स हैं, हेम्प है, सनफ्लावर है, कैनोला है, इनमें यील्ड बहुत कम होती है. जब हम एक्सट्रैक्ट करते हैं ऑयल, दे गिव 25 to 30% ऑयल. तो अब इसके जेनेटिक लेवल पे हम जाके ऑयल यील्ड एनहांस करें, ठीक है. फिर जो है वो हमें फैटी एसिड ऐसे चाहिए जो पॉली अनसैचुरेटेड फैटी एसिड हो, तो हम जेनेटिकली उनको मॉडिफाई करें और उसको एनहांस करें, अल्टर करें. दिस इज कॉल्ड द genetically modified or altered food that is contains some special product or special nutrients which is not normally present. The last one is an enhanced food that changing raw commodities to alter nutrient composition. उसमें रॉ कमोडिटीज हैं जो यूजलेस चीजें हैं उनको यूजफुल में कन्वर्ट किया जाए. These are that type of food is called the enhanced food. Here we look the examples of these all five classes of the food. The basic food, which is contain oat, barley, wheat, bran, cereal, soya-based product, green tea, seed, hemp, flaxseed. These all are the basic foods. They contain the nutrients and health benefits. If you look the fortified food, like a calcium fortified milk and beverages, तो ऐसे जो है वो मिल्क आ रहे हैं आजकल मार्केट में कि दे हैव मोर कैल्शियम. नॉर्मली कैल्शियम कम होता है मिल्क में, लेकिन ज्यादा एनहांस करने के लिए उसको फोर्टिफाइड उसमें एडिशन की जाती है ताकि जो है उसको कैल्शियम का लेवल जो है वो हाई रहे. इस तरह बहुत सारे ड्रिंक्स कंट्रीज में आते हैं जिन लोगों को कैल्शियम की डेफिशिएंसी है, उनको ऐसे वाटर अवेलेबल है जिनमें हाई कैल्शियम होता है, दे कैन यूज दैट वाटर. योगर्ट विथ प्रोबायोटिक्स. प्रोबायोटिक्स आपको पता है कि दे आर इम्पोर्टेंट, जो हमारा जो फ्लोरा इंटेस्टाइन उसको इन्हांस करने के लिए उसमें योगर्ट्स में प्रोबायोटिक्स यूज किए जाते हैं ताकि हमारा जो इंटेस्टाइन फ्लोरा है वो ज्यादा हेल्दी हो और जब ज्यादा हेल्दी होगा दे प्रोड्यूस द विटामिन के as well as they could not provide the attachment of the foreign particles. तो जब foreign particle attach नहीं होंगे they will not able to create any disease. Drinks with herbal extract. आजकल आप देखते हैं कि रेस्टोरेंट्स में वो जो पुदीने वाला मिंट उसमें इसलिए दिया जाता है कि उसमें हर्बल डाली जाए ताकि वैसे जो बोतल है दे आर नॉट गुड फॉर हेल्थ जो 7 अप है खासतौर पर 7 अप में यूज किया जाता है लेकिन अगर उसमें आप पुदीना डाल लें तो उसकी टॉक्सिसिटी को वो कम करे और हेल्थ बेनिफिट्स की तरफ ज्यादा जाए इस तरह फोलिक एसिड एंड आयरन फोर्टिफाइड ब्रेड्स एंड सीरियल्स vitamin B fortified margarines and iodized salt these all are the example of fortified food which are nowadays commonly available in the market. Enriched food like vitamins and mineral enriched products like white bread products containing added fiber and symbiotics. तो ये जो है ये enriched है कि इनमें ऐसी चीजें डाली जाएं कि जो और ज्यादा एनरिच हो और उसकी जो क्वालिटी है वो बढ़ जाए और जो एक खास जो उसमें डिमांड और नीड की किसी खास कंपोनेंट की उसको एनरिच किया जाए इस तरह प्रोडक्ट विद एडेड ओमेगा 3 फैटी एसिड ओमेगा 3 फैटी एसिड इज अ इंपॉर्टेंट फैटी एसिड फॉर आवर बॉडी and it cannot synthesize by our body we must obtain from a diet तो ओमेगा 3 फैटी एसिड जो ऐड किए जाए उसमें घी है और जो हम डेली यूज करते हैं उसमें ओमेगा 3 फैटी एसिड यूज उसमें ऐड किया जाए उसको एनरिच किया जाए उसकी क्वांटिटी जो है वो इंक्रीज की जाए कैल्शियम एनरिच फ्रूट जूसेस देन आयरन एनरिच ब्रेकफास्ट सीरियल एंड प्लांट स्टेनोल्स एंड स्टीरोल एस्टर्स एंड margins these are all the examples of the enriched food which are nowadays available in the market. Altered or genetically modified food. अब इसमें जो altered में जो कोई खास कंपोनेंट है उसको एनहांस किया जाए अगर कोई कंपोनेंट टॉक्सिक है उसको रिमूव कर दिया जाए मसलन अब कुछ लोगों को एलर्जी होती है बहुत सारे कंपोनेंट से किसी खास पॉपुलेशन को अगर एलर्जी है तो हम उसके सीड लेवल पे जाकर उसको जेनेटिकली मॉडिफाइड कर दें और वो कंपोनेंट जो रिस्पॉन्सिबल है टू कॉज द एलर्जी उसको हम रिमूव कर दें तो इस तरह बहुत सारे एग्जांपल्स हैं मसलन वेयर यू कैन सी द रिड्यूस फैट एंड फैट फ्री प्रोडक्ट्स फैट ज्यादा ना हो ताकि हमारे लिए प्रॉब्लम क्रिएट प्रोडक्ट्स विद द इंक्रीज्ड फाइबर कंटेंट्स तो फाइबर कंटेंट्स जो इंक्रीज किए जाए दे आर गुड फॉर आवर स्टमक इस तरह शुगर जो है शुगर और साल्ट जो फ्री चीजें हैं वो बनाई जाए तो जो शुगर है वो कम की जाए और जो चीजें जिनमें साल्ट है उनको साल्ट कम की जाए दीज ऑल टाइप्स ऑफ फूड आर कॉल्ड द genetically modified food and it can be level we can alter or knock out that specific gene which is responsible to cause some specific things are produced in a high quantity in any food. Enhanced food like golden rice with enhanced beta carotene contents, enhanced production of vitamin E because this vitamin E is act as antioxidants, so it can be present in different steps in different stuff in a high quantity. Lycopene enhanced tomatoes ऐसे टमाटर जिनमें लाइकोपीन वैसे होता है लेकिन हम उसको और enhance करें क्योंकि they are good for the cure of the or prevention of the cancer or prostate cancer particularly. High lysine corn, fruits and vegetables with enhanced contents of vitamins, carotenoid containing potatoes. तो ये तमाम चीजें जो हैं इनमें enhance की जाए जो नॉर्मल लेवल है उसका लेवल enhance किया जाए ताकि they are good for the health. Here you can see the typical nutraceuticals nowadays. If you visit the market, particularly in advanced countries, in the pharmacies, there are shelf which have the nutraceuticals and vitamin pills, and they are obtained from a natural source. Mineral pills हैं जो आज तो deficiency है अगर वो immune system strong होगा तो face diseases, herbal supplements, oil, gel, caps, these are commonly available in the market. In the pharmacy, particularly in advanced countries, they are moving to the natural sources because synthetics always creates problems. जो पिछली सदी थी 20 उसमें केमिस्ट्री बहुत अरूज पे कई सिंथेटिक चीजें बनाई गई लेकिन सिंथेटिक चीजें are not compatible with our body. So now again shifted on natural sources. Natural sources में चीजें चूंकि थोड़ी प्रोड्यूस होती हैं तो they are expensive. एडवांस कंट्रीज में जो शेल्फ है उनमें सारी चीजें जो हैं वो सिंथेटिक भी मौजूद होती हैं और जो नेचुरल सोर्स हैं वो भी मौजूद होती हैं. तो those people who can easily afford, so they can purchase natural stuffs, organic stuffs, and they are safe for the health. Here you can see the few probiotics function foods. You can see different yogurts, juice, different food stuffs are in market available. They contain some more fiber, some more contains probiotics. Juice, they contain some additional benefits or additional components. So you can purchase these from the market. Here another example, you can see the juice which contain the antioxidants in high amount, like biscuits or other stuffs which are more fiber, and other the small children's different food stuffs, and they also contain more fiber because fiber is good for our health. They are easily digest and good for the stomach. But the most important is the natural source. Always the natural source is the important. You can, instead of focus to the processed material of food stuff, you can use directly the fruits, vegetables, which have the good contents of the nutraceuticals. They have different components and they prevent you, and also they are the responsible if it's happens some disease. You can use high amount of any specific food, and it can be cure. It can be able to cure your disease if it happen. So these are the few vegetables, fruits you can use on daily basis in your food. Here you can see another is a strawberry, is a like a yogurt. It's contain the probiotic fruit drinks, and it also contain the multivitamins. So these drinks or these yogurts contain more probiotics, and it also contains some additional vitamins. Here another company is a Banicol, which has a claim that their products is responsible to lower the cholesterol, because the heart disease is one of the top leading disease which cause the death in the human being particularly. इस वक्त सबसे हाईएस्ट जो रेट है वो हार्ट डिजीज का है. हार्ट के बाद फिर कैंसर है. हार्ट डिजीज जो है वो दुनिया में बहुत ज्यादा लोगों को उनकी, अब हम पाकिस्तान में बहुत कॉमन देखते हैं कि लोगों को हार्ट अटैक बहुत ज्यादा हो रहे हैं. तो ये बैनिकॉल जो कंपनी है, they use the sterols extracted from the pines, from the plant sources, which is more safer, and they use in their cheese, in their butters, and they claim that their product is lowering the cholesterol instead of higher the cholesterol. So all these things are important, but there are certain claims that comes from the nutraceuticals and health claims. What are these claims? Early development and growth. If you use these nutraceuticals, they do the good development and proper growth. Regulation of basic metabolic process, defense against oxidative stress and certain cancers, cardiovascular and gastrointestinal improvements, improve mental health, immune response enhancing effects, and also the optimal defense against infectious diseases. So these are the claims that comes from the nutraceuticals, those pharmacies, those industries who are selling the nutraceuticals. They have common these claims that their function food, their nutraceutical have these properties. So regulatory bodies is important in every country. Unfortunately, in Pakistan, we have less regulatory bodies in different sectors, and also the issue in the function food or nutraceutical. Still, we have problems, but in advanced countries, they have their regulatory bodies. If you can extract something, you can prepare some food stuff, you can get a licensing, you can get some testing from some different regulatory bodies. Here you can see from Brazil, from Canada, from China. They have their regulatory bodies. They can check your claim, either your nutraceutical or functional foods is performing specific functions. After checking to allow you can sold the things in the market. Few advanced countries have their regulatory bodies to check the quality of the nutraceutical and functional foods. Now the important thing is a market over here. In Pakistan, there is no statistical data is available that how much things or how much money we generate by using this nutraceuticals or functional foods. But here you can see some advanced countries data are available. This is a very good market because in the coming years, people are focusing to use the functional food or nutraceuticals. So in each year, the production in terms of finance is increasing. Here you can see it's claimed that its business was in 2017, 299 billion US dollar business of these functional food industries. Now it is increasing, and they are projected, they are hopeful that it will reach approximately 450 billion US dollars by the end of this year, 2022. So we have a lot of opportunity to develop now some foodstuffs and sold in the market, and this is a good chance that because people now are convinced these nutraceutical and functional foods are very good for our health, so they are purchasing these stuff from the pharmacy or from the other like big markets. So we have good gaps to because each sector is the money is important matter. So we can develop some good food stuff to adding some new nutrients or useful nutrients, and we can earn billion. So here you can see in some China, some Europe or North America or Asia Pacific, you can see these are already earning a large amount of $1 billion by using these nutraceuticals and functional foods. So now we come to the problems, because it's look like a simple. In fact, it's not simple. It's challenging, and many problems are still we face to develop these nutraceutical and functional foods. The first, if we look at delivery, many bioactives are unstable. ये बहुत इशू है कि जो हम आइसोलेट करते हैं they are unstable. Chemically instability in oxidation हो जाती है. They can react with the air. Physiologically and physical instability like oxygen and moisture, these two are responsible to damage these bioactives. So we have still problems. We need to overcome these things that we can protect our bioactive compound when we isolate it from different sources. So many bioactives impact negatively or organoleptic of foods and beverages. कुछ बहुत यूजफुल होते हैं, लेकिन उनका जो इम्पैक्ट है वो ऑर्गेनोलैप्टिक्स हमारे जो जुबान है वो एक्सेप्ट नहीं करती, यानी टेस्ट उसका चेंज हो जाता है। आप उसको अगर कहते रहेंगे बहुत अच्छी चीज है तो वो नहीं है, क्योंकि टेस्ट नहीं है। जैसे मेडिसिन में ये हमदर्द वाले वो खून साफ करने के लिए साफी के नाम से एक नीम के दरख्त और पता नहीं क्या, वो इतना कड़वा होता है कि वो पीने को दिल नहीं करता। तब उसके बेनिफिट अच्छे भी हैं, जब पीने को दिल नहीं करेगा तो डेफिनेटली ऑर्गेनोलैप्टिक इज इम्पोर्टेंट। जब आप अल्टर करते हैं फूड स्टफ को, जब आप कंप्लीट फूड बनाने की कोशिश करते हैं तो उससे टेस्ट चेंज हो जाता है। तो वो टेस्ट चेंज जो है वो एक्सेप्ट करना लोगों का वो बड़ा डिफिकल्ट है। तो दिस इज अनदर इशू कि जब आप बहुत सारी चीजें इकट्ठी करते हैं तो उनका जो टेस्ट है वो चेंज हो जाता है। टेस्ट, ऑर्डर, फूड फंक्शनैलिटी, टेक्सचर, माउथ फील। टेक्सचर चेंज हो जाए, फीलिंग माउथ में अच्छी ना हो, आप खाने के बाद कहीं मेरा मुंह का जाता खराब हो गए, तो जितना भी यूजफुल हो ये बड़ा मुश्किल है कि अगर अनसूटेबल हमारे माउथ के लिए तो हम नहीं खाएंगे। Many existing delivery systems unsuitable for wide food, beverages and applications. Expensive है मटेरियल और प्रोसेसिंग इक्विपमेंट, ये जरूरत है बड़े स्केल पर जो है इसको प्रोसेस करना, वो एक्सपेंसिव है, वो अवेलेबल नहीं होगा तो हम उसको सक्सेसफुली जो है वो बेवरेजेस में सक्सेसफुली जो डिफरेंट फूड प्रोडक्ट्स में कन्वर्ट नहीं कर सकेंगे। Then development of suitable food vehicles for target delivery of bioactives. ऐसी स्पेसिफिक व्हीकल्स बनाई जाएं कि जो एक जगह से दूसरी जगह बायोएक्टिव कंपाउंड्स को वो ट्रांसफर करें। Delivery the bioactive compounds at desired levels, then acceptability, stability and bioavailability. Bioavailability is also important. तो अगर वो हम ले लेते हैं और अगर हमारे ब्लड में वो अवेलेबल नहीं होता तो फिर भी यूजलेस है। तो बायोअवेलेबिलिटी भी चेक की जाए कि जो हम कंपोनेंट्स ले रहे हैं आइदर दे आर प्रेजेंट इन अवर प्लाज्मा और नॉट, अगर होंगे एब्जॉर्ब होगा लेवल होगा तो देन यूजफुल होगा, वरना अदरवाइज उसकी बायोअवेलेबिलिटी अगर नहीं है, डाइजेस्ट नहीं हो रहा, उतना लेवल अचीव नहीं हो रहा तो हमारा टारगेट जो है वो अचीव नहीं होगा। देन एंटिसिपेट डिमांड फॉर पर्सनलाइज्ड न्यूट्रिशन and the potential role of functional ingredients. हर आदमी के लिए अलग से डाइट बनाना भी मुश्किल है, हर आदमी की डिमांड के लिए। Ensure stability of functional food ingredients during manufacturing and target delivery. तो आप इंश्योर करें क्वालिटी के जो आप चीजें डाल रहे हैं उनकी अमाउंट जो है वो अचीव हो रही हो, ऐसा ना हो कि लेवल कम हो तो जो आपके ऑब्जेक्टिव्स हैं वो अचीव ना हो। Then need to define the bioavailability of functional ingredients. तो ये भी है कि हर नेशन के लिए कितनी हमारे ब्लड में कितना लेवल उसका अवेलेबल होना चाहिए, कितना यूजफुल है और कहाँ टॉक्सिक है, कहाँ हाई लेवल, बहुत हाई लेवल भी होगा तो टॉक्सिक है। अब आयरन अच्छा है, कैल्शियम अच्छा है, लेकिन अगर बहुत हाई क्वांटिटी में लेंगे तो दैट विल क्रिएट प्रॉब्लम। कैल्शियम डिपॉजिट होना शुरू हो जाएगा हमारे किडनी में एंड दे विल कॉज द स्टोन। कैल्शियम सल्फेट, कैल्शियम कार्बोनेट और कैल्शियम के जो साल्ट हैं बनने शुरू होंगे। Then establish dietary reference intake for a wide range of nutrients. जो establish dietary है EDR, ये रेफरेंस ये बनाना होगा कि कितना यूजफुल है और कितना यूजफुल नहीं है। तो ये सारे वो चैलेंजेस स्टिल हैं, हम आइसोलेट कर लेते हैं, करेक्टराइज भी कर लेते हैं, लेकिन इनको कन्वर्ट करना फूड में और जो न्यूट्रास्यूटिकल्स बनाना इट्स डिफिकल्ट। देन अदर रिलेटेड इश्यूज, पब्लिक हेल्थ एजुकेशन। अब लोगों को ट्रेन करना, उनको एजुकेट करना कि these nutraceuticals natural source are good. Investment to bridge research gap, lack of knowledge in the intellectual property protection, then insufficient coordination of research activity. अब ये जो फूड के लोग हैं, फार्मा के लोग हैं और केमिस्ट्री के लोग हैं, ये जब तक आप इसमें मिलकर काम नहीं करेंगे तो डेफिनेटली ये जो डेवलपमेंट है फूड की, न्यूट्रास्यूटिकल्स की, फंक्शनल फूड्स की, ये नहीं होगी। क्योंकि जो फार्मेसी के लोग हैं वो ये कहेंगे कि सिंथेटिक कंपाउंड्स ठीक है, केमिस्ट्री वाले तो कन्विंस नहीं होते। केमिस्ट्री वाले चूंकि सिंथेटिक काम कर रहे हैं तो वो तो कन्विंस नहीं होते, वो इस चीज को नहीं मानते कि जो सिंथेटिक हैं वो डेंजरस हैं और जो नेचुरल हैं वो डेंजरस नहीं है, क्योंकि उनका सबसे बड़ा क्लेम ये जो सिंथेटिक है वो बड़े स्केल पर प्रोडक्शन होती है, नेचुरल जो सोर्स है वो है भी बहुत कम, तो प्रोवाइड ही नहीं कर सकते इतने लोगों को, तो इसलिए केमिस्ट जो है वो कन्विंस नहीं होते कि देयर इज अ डिफरेंस बिटवीन सिंथेटिक्स एंड नेचुरल सोर्स। देन लिमिटेड financial resources. This is another issue which cannot generate to the nutraceuticals, and a small number of large companies dominate and shape market. बड़ी जो कंपनी है उनने मार्केटिंग अपने कब्जे में की हुई है, तो छोटी मार्केट्स को छोटी जो कंपनी है उनको वो नहीं चलने देती। हमारे यहाँ जो मल्टीनेशनल कंपनी है वो नेशनल को इतना जल्दी नहीं चलने, तो वो ज्यादातर फॉर्मूला उनका ठीक नहीं होता, फिर उनको डिस्करेज किया जाता है कि आप मल्टीनेशनल की चीजें नहीं यूज करें। नीड फॉर साइंटिफिकली ट्रेन्ड पर्सनल, लोग जो है वो ट्रेंड हो ताकि उनको पता हो कैसे एक्सट्रैक्ट करना है। एंड नीड फॉर सर्च ऑफ पोटेंशियल इमर्जिंग मार्केट, दिस इज अनदर इशू कि हमें पोटेंशियल देखना है कि कौन से कंपाउंड हमें चाहिए, कौन सा हमें फूड्स चाहिए, तो उनको हम जो है वो बनाएं ताकि हम उनको सेल कर सकें और वो अर्न द मनी। साइंस इज आल्सो द रिस्पॉन्सिबल टू जनरेट मनी, साइंटिस्ट जो है वही बेसिकली रिस्पॉन्सिबल है कि वो कंट्री के लिए पैसे भी जनरेट करे, और हम हमेशा साइंस में 1 रुपया खर्च करें और 10 रुपए की चीज बनाए, ये ना हो कि 10 रुपए खर्च करें और 1 रुपए की चीज बनाए, ये हमारे यहाँ इशू है। इसी वजह से चाइना जो है वो इकोनॉमी में आगे जा रहा है, वो बल्क में चीजें बना रहे हैं, वहाँ हमें सस्ती मिल जाती है, तो फिर हमारे यहाँ कोई बंदा पैसे लगाने नहीं चाहता, वो कहता है आप तो 100 रुपए की चीज बता रहे हैं चाइना से, 1 रुपए की मिल रहा आपसे क्यों है। इंडस्ट्रियलिस्ट जो है वो तो आपसे बिजनेस प्लान पूछता है, आपसे उसको पता चल जाता है कि यू आर जस्ट टॉकिंग और इन फैक्ट यू विल बी एबल टू समथिंग, और चीपर भी हो, important aspect. So, if we conclude our talk, function food containing physiologically active compounds, either from a plant source, from an animal source, and it can improve health and possibly reduce the risk of diseases. Function foods industry is resource intensive, both in terms of financial resources and the time required for basic research, technology development and commercialization. तो ये इसमें सारे एस्पेक्ट्स मौजूद हैं कि हम इसको कमर्शलाइज कर सकते हैं अपने प्रोडक्ट्स को। New promising technologies such as mutagenomics can be used for development of food for target population groups with defined disease such as allergy, diabetes, obesity and cardiovascular disease. तो genetically modified जो foods हैं, sugar भी कम करें, salt भी निकालें, fats भी उसमें कम हो, ताकि जो लोग पेशेंट्स हैं, जो कार्डियोवास्कुलर के पेशेंट्स हैं, जो शुगर के पेशेंट्स हैं, वो वाले भी प्रोडक्ट्स खा सकें। Then the last one conclusion of the talk is, although they possess unique nutritional and health characteristics, functional foods still could not compete with the conventional food for the market share and shelf space in the retail stores. Still जो है वो फेमस नहीं है, क्योंकि एक तो महंगी है, दूसरा लोग इतनी केयर नहीं करते, जो अनपढ़ लोग हैं उनको तो कोई खास गरज नहीं है कि वो सिंथेटिक चीजें खा रहे हैं क्या खा रहे हैं, ठीक है, तो ये एजुकेशन की भी जरूरत है। तो बट स्टिल वी हैव द गैप, इफ वी डेवलप सम good food stuff, we can sell in the market. So thank you for your patience, for your listening. आपको क्वेश्चन आंसर है तो हाँ जी, प्रेजेंटेशन दे देते, मैं भी ले लें, मुझसे भी ले लें, तो मैं दिए इनको दे देंगे। हाँ जी। तो क्वेश्चन पूछें। ठीक है, ये देखें इसमें ये जो क्वेश्चन है ये डेफिनेटली वो लोग ज्यादातर नेचुरल यूज कर रहे हैं, लेकिन उनका नेचुरल बार दफा जो सिंथेटिक यूज करते हैं उस पर उनको पता नहीं है कि ये क्या है। प्रॉब्लम तो सिटी में भी है, क्योंकि सिटी में तो अवेलेबिलिटी नहीं है। सिटी में हमारे यहाँ प्रॉब्लम ये है, हमारे यहाँ तो मिक्सचर है, बहुत ज्यादा चीजों को मिक्स किया जाता है और लोग जो है वो केयर नहीं करते, तो उस वजह से जो कैंसर है और जो बीमारी है मतलब बहुत कॉमन हो रही है। तो एक्चुअली अनपढ़ का मतलब यह है कि जो लोग जिनको नॉलेज नहीं है कि जो न्यूट्रास्यूटिकल्स क्या है या फंक्शनल फूड क्या है, या अगर बस उनको ये पता नहीं है कि अगर अब तो खैर लोग ऐसा नहीं रहा कि अगर उनको पहले ब्लड प्रेशर है तो अगर हाई साल्ट वाली चीजें खा रहे हैं तो उनको पता होना चाहिए कि जो इसकी केयर करनी है हमने। इस तरह अगर उनको हार्ट का मसला है तो चर्बी खा रहे हैं, ठीक है, गोश्त की, या ऐसे फैटी फूड यूज कर रहे हैं, घी ज्यादा यूज कर रहे हैं। अब लोग मतलब अब चीजें चेंज हो रही हैं, कन्विंस हो रहे हैं लोग अब। तो वो नो डाउट के जो विलेजेस में लोग रहते हैं जिनकी एजुकेशन कम है वो डेफिनेटली वो चीजें ज्यादातर तो नेचुरल सोर्सेज से यूज कर रहे हैं, लेकिन फिर भी बहुत सारी चीजें मार्केट में चूंकि अवेलेबल हैं तो सिटी से भी बहुत सारी चीजें वो लेते हैं, तो कंटेमिनेशन होती है, सिंथेटिक चीजें डाली जाती हैं और वो सिंथेटिक चीजें यूजफुल नहीं है। ठीक है, मसलन अब सिंथेटिक चीजें जो हम मीठे चावल खाते हैं तो उसमें वो जर्दे का कलर यूज करते हैं, मतलब 6-7 रंग के कलर होते हैं, और वो सिंथेटिक 10 रुपए की पुड़ी आ जाती है, वो सस्ते होती है। तो अब उसको नेचुरल कलर भी अवेलेबल थे, यानी जामुन से और मैंगो से और इन मुख्तलिफ सोर्सेस से, लेकिन वो महंगा बहुत ज्यादा। वो इतनी सी पुड़ी वो जो 10 रुपए वाली वो 1000 रुपए की आती है या उसका 1500 की, तो वो तो कोई नहीं लेगा। वो फिर अब ये इसलिए भी है कि बाद लोगों के पास पैसे भी हैं, लेकिन वो कहते हैं ठीक है 10 रुपए की दर लग रही है तो मेहमानों को खिलाने की क्या जरूरत है 1000 रुपए की। तो वो रिकमेंडेड भी है कि आप वाइट चावल बना लें, वो सिंथेटिक कलर जो है वो बहुत डेंजरस है रहा है। ठीक है, तो दोनों साइड पे इशू है, मतलब विलेजेस में भी प्रॉब्लम है और जो है वो सिटी में भी प्रॉब्लम है। तो लोगों को एजुकेट करना है कि जो हमारा जो पिछले जो एक सदी पूरी केमिस्ट्री वालों ने इसको होल्ड रहा है, केमिस्ट्री सिंथेटिक चीजों पे रहा है सारा, अब सिंथेटिक चीजें जो डिक्लाइन हो रही हैं, क्योंकि सिंथेटिक चीजें जो है प्रॉब्लम्स क्रिएट कर रही है। कैंसर बहुत ज्यादा बढ़ गया है दुनिया में, हर चौथा आदमी कैंसर से गुजर रहा है, हर चौथा आदमी, टू मच। और हमारे यहाँ तो चूंकि कोई केयर नहीं की जाती। मसलन मैंने एक देखा कि बंदा जो है वो जो चिकन पीस है वो बेच रहा है, तो उसने जो है वो ही इज एमफिल फ्रॉम एग्रीकल्चर यूनिवर्सिटी फैसलाबाद एग्रोनॉमी। मैं सर्वेदा का वाक्य बता रहा हूँ, मुझे किसी ने बताया कि एमफिल है, जॉब नहीं मिली तो अपनी इसलिए। अब वो क्या करता है कि वो जो प्रेस से न्यूज पेपर निकलते हैं, जो शुरू वाले जब वो ट्रेसर लगाते हैं तो शुरू वाले जो 15-20 पेजेस निकलते हैं वो इंतहाई ब्लैक होते हैं, बिल्कुल उनके ऊपर कच्ची स्याही होती है। तो वो वहां से फ्री ले आता है, जिनको वो बना के लपेट के दे रहा है, वो जब आप घर जाके खोलेंगे आपके हाथ भी काले हो जाएंगे डाई से, और वो जो चिकन वाला पीस लिया आपने वो भी सारा काला होगा। ठीक है, तो वो डाइस जो है काफी हो जाएंगे। फिर बाजार में हम जो ऑयल है ओनली वंस अ टाइम, मैक्सिमम 2 टाइम रिकमेंडेड फॉर फ्राइंग पर्पज, मैक्सिमम। वो भी बहुत हालात खराब हैं, जैसे हमारे यहाँ कंट्री के हालात बहुत खराब हो गए हैं, तो आप एक दफे पकौड़े बनाए तो उसमें दूसरी दफे बना के उसको थ्रो कर दें। हमारे यहाँ देख लें ये जो शहरों में जो फ्राई कर रहे थे चीजें बना रहे हैं समोसे, वो तो खानदानी बिजनेस होता है, अगर कोई चेंज करें और बता दी, वरना कम हो रहा है तो उस पर थोड़ा और डाल दें। ठीक है, अच्छा वो प्लास्टिक का जो पैकेट है वो उन्होंने पुड़ियाँ बन रही हैं और साथ प्लास्टिक का घी का पैकेट एक साइड में रखा होता है, वो उस पर से प्लास्टिक भी मेल्ट होकर अंदर जा रहा होता है। प्लास्टिक का मेल्टिंग पॉइंट जो है वो 430 डिग्री सेंटीग्रेड, और जो ड्राई हीट हम यूज कर रहे हैं, ये जो बर्तन को लग रही होती है, पानी के अंदर पका रहे हैं उसका तो टेम्परेचर 100 से ऊपर नहीं जाता है, मिसाल में बना रहे होते हैं घी का टेम्परेचर जो डालेंगे वो 500 से भी ऊपर होता है डिग्री सेंटीग्रेड, और वो जो ड्राई हीट है उसका 500 डिग्री सेंटीग्रेड, तो वो उस कढ़ाई के ऊपर उसने रखा हुआ है साइड पे वो प्लास्टिक, उससे मेल्ट होकर प्लास्टिक सारा बन जाएगा। प्लास्टिक, पॉलीथीन, ये जो प्लास्टिक्स हैं ये बहुत बड़ी वजह है कार्सिनोजेनिक एक्टिविटीज। अब ये देखें कि हम केयर नहीं करते, डिटर्जेंट्स, कलर्स, फूड कलर्स, ड्रिंक्स कलर वाले, सिंथेटिक्स, बोतल्स, ये जो ब्लैक कलर, ये सारी, इट्स नॉट गुड फॉर हेल्थ, इट्स अ वेरी देयर वेरी बैड इम्पैक्ट ऑन आवर हेल्थ। वो कुछ टाइम हम बर्दाश्त कर देते हैं कि हमारा इम्यून रिस्पॉन्स करता है, लेकिन जब फिर एज 40-45 होती है तो फिर सारी बीमारियां जो है वो आ जाती हैं। हाँ जी। स्टे देखें दोनों क्योंकि सॉल्युबल हैं, उन्होंने जो सिंथेटिक बनाया है वो भी, उसका स्ट्रक्चर भी वही होता है जो नेचुरल कंपाउंड होता है। स्टे दोनों करेंगे, दोनों वाटर सॉल्युबल विटामिन है, विटामिन सी का एग्जांपल है, एक्सेस भी ले लें कुछ नहीं होता, जो एक्सेस होती है वो हमारी बॉडी निकाल लेती है यूरिन के थ्रू, और जितनी हमें चाहिए डिमांड वो बॉडी में सस्टेन रहता है। तो अब अवेलेबिलिटी दोनों की है, उसका कोई इशू नहीं है, लेकिन नेचुरल सोर्सेज में बार दफा हम चीजें नहीं ले पाते, बार दफा डॉक्टर फील करता है कि आपकी बॉडी में काफी डेफिशिएंसी हो गई है। नेचुरल सोर्स ऑरेंज वगैरह लें, उसमें भी ज्यादा है, फिर गावा में उसके बाद ज्यादा है, सबसे ज्यादा ऑरेंज, गावा में ऑरेंज से भी ज्यादा है, फिर उसके बाद जो ऑरेंज फैमिली इसमें ज्यादा है विटामिन सी। तो नेचुरल सोर्स से हम जब देखते हैं कि फील हो रहा है डॉक्टर को प्रॉब्लम आ रही है, बीमारी आ रही है, कोई हमारे गम्स जो है वो काम सही नहीं कर रहे, हमारे स्किन के ऊपर, तो उसमें वो समझते हैं कि विटामिन सी की डेफिशिएंसी, उस सूरत में सिंथेटिक चीजें दी जाती हैं, तो यूज कर सकते हैं सिंथेटिक, लेकिन रिकमेंडेड यही है फॉर गुड हेल्थ कि आप नेचुरल सोर्सेज को जो है, अब हम बिल्कुल सडनली तो मेरी टॉक के बाद आप ये तो नहीं कर सकते कि आप सिंथेटिक चीजें छोड़ दें सारी की सारी, ठीक है, वो तो नहीं पॉसिबल, लेकिन ये कि हम आहिस्ता आहिस्ता हम ट्रेंड बनाएं कि हम नेचुरल सोर्सेज की तरफ जाएं। नहीं, ये ऐसा नहीं कुछ, क्योंकि देखो वो तो बॉडी हमारा एब्जॉर्ब हो जाएगा ब्लड से, जब एब्जॉर्ब हो जाएगी तो उसके बाद ब्लड में उसका लेवल जो है वो रहेगा, वो बायोअवेलेबिलिटी है, ये खास लेवल अचीव हो जाएगा, ठीक है। तो ये इसमें तो कोई खास डिफरेंस नहीं है, दोनों चीजें स्टे करेंगी, लेकिन जो नेचुरल का स्ट्रक्चर है, नेचुरल जो है चूंकि हम यूज कर रहे थे, हमारी बॉडी की कंपैटिबिलिटी ऐसी होती है तो उसका केमिस्ट नहीं मानते, इसलिए कि स्ट्रक्चर तो सेम होते हैं। अब विटामिन जो सी है वो उसका सेम स्ट्रक्चर है, बॉन्ड लेंथ में क्या डिफरेंस है, या हम जो नेचुरल चीजें लेते हैं वो हम प्योर नहीं लेते, मसलन विटामिन सी लेते हैं वो प्योर नहीं होता, उसमें और भी 7 चीजें होती हैं, वो और 7 चीजें उसका जो असर है वो नहीं होने देती, नेगेटिव नहीं। हाँ जी, उसमें फाइबर है और विटामिन भी होते हैं, उसके अलावा ज़ाहिर बात है आप मिनरल्स भी ले रहे होते हैं, तो वो बाज़ दफ़ा आपको यूजफुल जो है वो करता है। हाँ जी, जबकि सिंथेटिक जो है वो तो प्योर आपने विटामिन सी की टैबलेट्स प्रेस हुई हैं, वो कंपाउंड बनाया हुआ है, जो उसका स्ट्रक्चर अस्कॉर्बिक एसिड बेसिकली उसका स्ट्रक्चर है वो बनाया हुआ, सिंथेसाइज किया हुआ उन्होंने, वो प्योर है और अवेलेबल तो होगा, इम्पैक्ट तो करता है, वैसे नहीं कि इम्पैक्ट उसका नहीं हो, लेकिन लॉन्ग जो लाइफ है उसको प्रोलॉन्ग करने की बजाय जो है वो रिड्यूस कर रहे हैं। अच्छा, जो विटामिन है उनकी 2 मेजर क्लासेस हैं, एक वाटर सॉल्युबल है और एक जो है फैट सॉल्युबल है, जो बाकी हैं वो फैट सॉल्युबल है जो आप बात करें, तो फैट सॉल्युबल जो विटामिन हैं वो हमारी बॉडी में लॉन्ग टाइम तक रहते हैं, यानी जो विटामिन सी है वाटर सॉल्युबल वो तो 1 दिन 2 दिन बाद हमारी बॉडी से उसकी डेफिशिएंसी होनी शुरू हो जाती है, फैट वाले जो हैं वो 20 दिन 25 दिन रहते हैं हमारी बॉडी में, तो फैट सॉल्युबल जो विटामिन हैं वो चूंकि फैट में सॉल्युबिलिटी होती है तो वो हम उसका भी नेचुरल सोर्स जो है, जिस तरह ऑयल है, हम डेली घर में सुबह पराठे खाते हैं, ऑयल इज द सोर्स ऑफ विटामिन ए, ई एंड बी। इस तरह जो है विटामिन बी जो है वो और भी मुख्तलिफ जो है वो फिश में है जो इस तरह। तो वो जो फैट सॉल्युबल विटामिन हैं वो बेसिकली जो है वो ओके, फैट सॉल्युबल जो विटामिन हैं वो हमारी बॉडी में रहते हैं। एक्सेस का ज्यादा एक्सेस खतरनाक है, अच्छे नहीं है, लेकिन नॉर्मली लेवल उनका हमें थोड़ा चाहिए होता है तो हम नेचुरल सोर्स से लेते हैं। नेचुरल सोर्स में फिश है हमारे पास, ऑयल्स हैं, सीड्स हैं, ये जो हम मुख्तलिफ सर्दियों में ड्राई फ्रूट्स खाते हैं, मूंगफली है, अखरोट है, बादाम, दीज ऑल आर कंटेन्ड विटामिन, फैट सॉल्युबल विटामिन ए, बी, ई, के। चलें, टाइम क्या नहीं ज्यादा होगा, मैं मीटिंग में पहुंचने को मैं जी। हाँ जी, कुछ अच्छा, ग्रीन सॉल्वेंट साथ है। ग्रीन सॉल्वेंट में जो ग्लाइसरीन is a green solvent, water is a green solvent, कार्बन डाइऑक्साइड उसको ऐसे प्रोसेस से गुजारा जाता है, दे आर आल्सो ग्रीन सॉल्वेंट। आजकल हाइड्रोपोनिक मीडिया आ गया। हाइड्रोपोनिक मीडिया ऐसा मीडिया है कि उसमें ये फ्रेंच एक साइंटिस्ट है जो फ्रेड चबाउट के नाम से वो साइंटिस्ट, जो इज वेरी फेमस साइंटिस्ट, उन्होंने ग्रो प्लांट को किया हुआ है मीडिया के अंदर, ठीक है, मीडिया के अंदर ग्रो किया है, और वो मीडिया के अंदर ऐसा ग्रो करते हैं प्लांट्स, और उसी के अंदर जो है वो कंपाउंड जो है वो आते रहते हैं, दैट इज कॉल्ड द हाइड्रोपोनिक मीडिया। तो ये जो इस तरह के मीडिया हैं जो सॉल्वेंट्स हैं वो ग्रीन सॉल्वेंट्स कहलाते हैं, जो लेस हैजर्ड्स हों, जिसकी लेस टॉक्सिसिटी हो फॉर द एनवायरमेंट। इसकी निस्बत एनहेक्सेन, मेथनॉल, एथनॉल, क्लोरोफॉर्म एंड पेटिथर, ये सारे वो सॉल्वेंट्स, लॉन्ग लिस्ट सॉल्वेंट्स की, वो एनवायरमेंट के लिए डेंजरस है। कोशिश तो की जाती है कि उनको उस तरह एनवायरमेंट में ना फेंका जाए, लेकिन जो हमारे जो लोग काम कर रहे हैं, रोटरी ऑपरेटर होता नहीं है, तो हम ये करते हैं कि चलो रख दो, सुबह आएंगे तो उड़ जाएगा, तो वो एनवायरमेंट में जाएगा। दूसरा, हम ये समझते हैं कि हमारे दो बोतलों से कौन सा एनवायरमेंट खराब हो रहा है, वो दो बोतलें करते हुए हमारे एनवायरमेंट का, अब इस वक्त सबसे हॉट इशू जो है वो एनवायरमेंट है। एनवायरमेंट इस वक्त बहुत ज्यादा प्रॉब्लम है जी। अच्छा, इसमें यह कि जो वो बटर इस्तेमाल करते हैं, मैंने बताया उसमें स्टेनोल और वो जो कोलेस्ट्रॉल है वो एक्सट्रैक्ट करते हैं प्लांट सोर्स से, ठीक है, जिसमें शायद एक नाम आपने देखा होगा पाइनएप्पल और इस तरह सोर्सेज हैं। तो ये बटर वगैरह में डालते हैं, उससे ये होता है कि जो अनसैचुरेटेड फैटी एसिड्स है, ठीक है, हमारी बॉडी में जो बैड कोलेस्ट्रॉल है वो कम होना चाहिए, और जो HDL है, हाई कोलेस्ट्रॉल लेवल जो है वो ज्यादा होना चाहिए, जिसके साथ प्रोटीन्स अटैच हो, ताकि जो उसकी जो ड्रॉपलेट्स हैं वो ना बने, और हमारे हार्ट में से जब गुजरे तो वो ड्रॉपलेट्स हमारे हार्ट की आर्टरीज को ब्रेक नहीं करें, ब्लॉक ना करें, ठीक है, क्योंकि उसमें मैकेनिज्म उनका ये है कि वो जो प्लांट सोर्सेज है जो डालते हैं स्टेनोल्स, वो जो है वो उनको हेल्प करते हैं डाइजेस्ट करने में, और ड्रॉपलेट्स नहीं बनने देते, वो अटैचमेंट होती है विद द प्रोटीन, हाई डेंसिटी लिपोप्रोटीन में कन्वर्ट होता है, और हाई डेंसिटी लिपोप्रोटीन जितनी ज्यादा होगी उतना ही वो गुड होगा हमारे लिए, जो लिपिड्स हैं, जो फैट्स हैं, ताकि हमारे ब्लड में वो ड्रॉपलेट्स ना हो, और अगर ड्रॉपलेट्स होंगे तो जब ब्लड गुजरेगा हार्ट की आर्टरी से, तो सबसे स्मॉल आर्टरीज होती हैं हार्ट की होती हैं, वो ब्लॉक हो जाती हैं, उनको ब्लॉकिंग की वजह से वो हार्ट काम नहीं करता। तो अल्टीमेटली हार्ट अटैक, हार्ट की मुख्तलिफ डिजीज और भी हैं हार्ट अटैक के अलावा, तो वो जो है वो अपीयर होती है। इसलिए वो जो कंपनी है वो ये कहती है कि अगर आप हमारा बटर, हमारा आप घी इस्तेमाल करेंगे तो आप प्रिवेंट रहेंगे, मतलब आप सेफ रहेंगे, कि इंस्टेड ऑफ लोकल आप कोई चीज यूज कर लें और वो आपके लिए अच्छी ना हो, यानी हार्ट को प्रॉब्लम क्रिएट करें। अब मैकेनिज्म ज्यादा, तो पहला मैकेनिज्म इसमें यही है कि ड्रॉपलेट्स नहीं बनते, ट्राइग्लिसराइड जो सैचुरेटेड है वो कम हो, अनसैचुरेटेड हम ज्यादा इस्तेमाल करें। तो अनसैचुरेटेड जब हम ज्यादा इस्तेमाल करेंगे तो वो सॉलिड नहीं बनते रूम के अंदर। ये घी वगैरह अच्छी पहचान ये होती है, जो दिल दिल किरण और गनी और असलम और सबक के नाम से जो घी आ रहे हैं, ये इनका चेक करने का यही तरीकाकार होता है कि आप थोड़ा सा घी निकाल के अपने थैली पे रखें, 1 मिनट में, सर्दियों में खास तौर पर, और अगर आप टेम्परेचर एसी में इस वक्त 25 टेम्परेचर है, अगर रूम टेम्परेचर पे आप उसको रखें, अगर वो देखते हैं कि 1 मिनट के बाद अगर तो वो मेल्ट होना शुरू हो गया, आपका जो सॉलिड है वो लिक्विड में बनना शुरू हो गया, तो वो घी गुड क्वालिटी वो आप यूज कर सकते हैं, क्योंकि हमारी बॉडी में वो मेल्ट हो गए। और अगर वो देखा जाए वो जनाब रख के बैठे हैं आप, उस तरह पत्थर बना हुआ है, मेल्ट नहीं हो रहा है, तो बैड क्वालिटी की है, वो ना खाएं, क्योंकि वो जो है ये जितने खैर से सस्ते थोड़े होते हैं, जो प्रोसेसिंग उनकी सस्ती होती है, ये जितने हमारे होटल हैं ये सारे के सारे बैड क्वालिटी यूज करते हैं, गुड क्वालिटी यूज नहीं, थोड़े से पैसे बचाते हैं अपने। तो चलो मैं आप क्वेश्चन या तो मेरे ईमेल पे कर दें, क्योंकि मैंने भी मीटिंग में जाना है और मैडम भी शायद कर रही है। चलो थैंक यू। आप लोग जो शुरू में शायद मैं था ईमेल कर सकते हैं, ईमेल लेसन, मैंने भी एक मीटिंग यूनिवर्सिटी में जाना है और मैडम जो ऑर्गेनाइजर्स वो भी चाह रहे हैं। थैंक यू। बस कुछ समझ आ गया आप लोगों को शायद इसमें कुछ बीच में एक अच्छी समझ आया तो बंदा कहता अच्छा हो रहा है, और अगर कोई समझ ना आ रहा तो चलिए थैंक यू। चलिए थैंक यू, बहुत शुक्रिया आपका। थैंक यू मैडम।
Thank you so much, sir. It was very knowledgeable and very interesting session with you. And ladies and gentlemen, this brings us to end the first session of the day. Let's take lunch break and resume the session at 2 p.m. It will be 30 minutes break now.
Ali. Hi, Martha. Martha, I think, okay.
I muted myself, sorry. Hello.
Okay. Okay, here is a lunch break, and we were a bit late in our first session. That's why people are coming back to auditorium. It may take 4 to 5 minutes, so
No problem.
we will resume at that time.
Okay, let me know when you're ready.
Okay. Okay. Madam बुला लें. Martha, this is my university auditorium.
Lovely. How many people are we expecting in there?
Sorry?
How many people will you be expecting? How many students are on site?
No student, majority of participants are the faculty members from our university and from other universities.
Oh, I see.
But they are mostly Lahore based. I think the number is around 35 to 40.
Because I can see 26 online as well.
Yeah, it may increase in a few minutes because it was lunch break, so people will join us again. Okay. So I think about 40 online and 40 in person here.
Okay. And I realize it's a sort of mixture of talks, something about education, but also science. Looking at the schedule, today they just had genomics lecture, is that right?
Yeah, it was a mixture, yeah. Okay, thank you.
Okay.
Here is Miss Summer. She is hosting this event.
Okay.
Hi, Dr. Martha, how are you?
Hello, I'm good, thank you. It's early morning here. Well, not early, but 9 o'clock. So early start in a way in comparison to you.
We are just starting in 1 or 2 minutes.
That's okay. So what city are you actually based? Because it's a virtual one. So I suppose people can join from wherever they are, but the auditorium, where is it physically?
We are actually in Lahore, Pakistan, our head office, Virtual University of Pakistan. And the participants are also from Virtual University and other universities.
Okay.
And some participants, those are from outstation, they are on Zoom online.
Yes.
Ladies and gentlemen, welcome back. We will now resume our second session of the day. The speaker of this session is Dr. Martha Potema. She is working as lecturer in Cardiovascular Science, Cardiology Research Center, Molecular and Clinical Science Research Institute, St. George's, University of London. Before that, she was serving as Senior Research Associate at Center for Heart Muscle Disease, Institute of Cardiovascular Science, University College London. Dr. Potema is reviewed for numerous journals and a member of Genomics England Cardiovascular Domain of the 1 Lakh Genomes Project. She is finalist of the Young Investigator Award competition and active member of the ICS Research and Academic Networking Group. Today, she will deliver her talk on genomics of familial hypercholesterolemia from diagnosis to management. Welcome, Dr. Potema, on behalf of Virtual University of Pakistan. Please proceed for your presentation.
Thank you very much for this kind invitation and an introduction. It's a pleasure to join your event and to see many attendants in the audience and online. So yes, it's early morning here, and I'm kind of full of energy, but I suppose you just had your lunch break, so you might be in slightly different state, but I hope I won't be presenting too much of hard science. I want to today demonstrate to you based on the disease, familial hypercholesterolemia, how you can utilize genomics, how you can improve patients' identification, treatment, and most of all, prevent cardiovascular disease. Let me just find my slides so that I can share my screen with you. Can you see that? Can now see my screen?
Yes,
Okay.
yes, Dr. Fatima.
I'll do full screen. Right. Yes, so I am a lecturer at St. George's University, but I still have close links with UCL, where I did my PhD and 2 postdoc positions. Tomorrow, I believe you will have a presentation from my ex-PhD supervisor, Professor Steve Humphreys. So we really enjoyed working together, and we still continue some work on familial hypercholesterolemia. I will call familial hypercholesterolemia as FH, which is a little bit easier to pronounce. And today I will talk to you about the clinical characteristics of the disease. It is a disease of high LDL cholesterol, and so we will cover the basics of that pathway, how that cholesterol is metabolized. Phage is a genetic disease, so we will cover the genetic causes of it and look at the genetic epidemiology of phage. So how common it is, what mutation cause it all around the world. We will look at the diagnosis, from clinical to genetic diagnosis, and screening approaches for phage. And in the second part of my lecture, I want to show you something more on genomics. So comparing monogenic versus polygenic causes of high cholesterol, and how you can utilize genomics knowledge to identify novel causes of phage, and from that, how you can go to novel treatment as well. So it will be genomics heavy. I don't know what exactly you just had on your previous lecture, but I hope this is not going to be too much, and perhaps you can relate. This would be a good example for you, how you can use your previous knowledge. Yes, so hopefully that will be, we will be able to cover that all today. So what is familial hypercholesterolemia? What are the clinical characteristics? Well, it says a lot in its name. It is a disease that affects families, and the hyper part of the name means too much cholesterol, and emia is from a Greek word that means in blood. So in its name, we can understand that it is a disease that affects families due to high cholesterol levels in the blood. And indeed, if we're talking about familial disease, I wanted to bring you an example of a family that is affected by phage. I didn't want to use the real picture, so this is just a sketch of a family, young family. We've got mom, Jeanette, who is 33-year-old, dad, Mike, 35-year-old, and they've got 2 sons. There is Daniel, who just turned 5, and a toddler, Matthew, who is 1-year-old. And so they are living a busy family, young family life, both working parents. And suddenly at the age of 35, Mike unfortunately suffers from myocardial infarction, so a heart attack. And obviously this is tragic news for young family. It's not only a health issue for Mike, but also puts loads of pressure and stress on the family. Therefore, I want to just highlight that as a familial disease, even if a person is not affected, there will still be affected in other ways, such as stress, or that they will need to start looking after member of their family. So one of the first things that doctors do when they diagnose patient with myocardial infarction is they look at their cholesterol levels, and specifically LDL cholesterol. This is the bad cholesterol. So they did that for the whole family in Mike's case, because they were already, I think, suspecting familial hypercholesterolemia, as the heart attack occurred very early on. And indeed, Mike's cholesterol levels, LDL cholesterol, was 6.8 millimole per liter. If you compare him to the cholesterol levels of his wife, it is much higher. And they both have similar diets, so you kind of think you could exclude any dietary issues here. The doctors decided to look at the children, and I will expand on that later. Why would they be looking into children? But it would be mainly because of you might be able to prevent such event in the future. So let's just bear in mind that Daniel has got LDL cholesterol slightly lower than his mum, and he's a 5-year-old, whereas Matthew has cholesterol level of 2.5 millimole per liter. So we will come back to that later. So clinical characteristics of a phage include the fact that it is an autosomal dominant disease. What that means is that 50% of your first degree relatives would inherit a mutation. So your children or your parents would also be affected. Therefore, when talking about a phage, it is very important to analyze the family history of either early myocardial infarction or very high cholesterol levels. Having 50% of your first degree relatives really strongly suggests that this is a phage, that there is something that is strongly inherited. And it is a disease of high LDL cholesterol, so the bad cholesterol, and we will talk a lot about LDL a little bit more later. But that cholesterol levels is people with a phage, they are exposed to that high cholesterol levels really from birth. And although perhaps in children this isn't visible, it's kind of a silent risk factor. But when the cholesterol level accumulates over time with age, that leads to a very high risk of premature heart attack. And the Phage Foundation estimated that 1 in 5 heart attacks under the age of 45 are due to familial hypercholesterolemia. So pretty serious issue. So let's think about Mike again and think how or what caused this heart attack in his case. So to talk about heart attack, we always think of cardiovascular system. So here is a picture of an artery, a healthy artery. We've got the lumen, the arterial wall. And so we know that Mike had hyperlipidemia or high cholesterol levels. So clearly, when you have high cholesterol level over time, that leads to a buildup of that fatty substance in the arterial wall, a process called atherosclerosis. There is a sort of over time plaque that forms, there is an inflammation going on. So loads of bad things are happening, but this is still kind of okay until the moment when this plaque ruptures. And when it ruptures, it will form a clot. And the clot is really the very dangerous part of that atherosclerosis and a plaque formation. So the clot may block an artery, and that will prevent obviously the blood flow, oxygen flow to organs. And if it happens near heart, we will have a muscle damage, and that is the myocardial infarction. So really, if you think about the cause in terms of Mike's case, we would probably need to start thinking about why he's got high cholesterol rather than looking at the downstream events. So we want to go back to this point, why there is high LDL cholesterol in this person, and could this then downstream events be prevented? So I think to answer the question why a person may have high cholesterol, we would need to look at how is actually cholesterol cleared in our body. So at least here in Western Europe, we get loads of bad press for cholesterol. It's kind of, we know we should not be eating fatty diet, and also our environment is full of fatty food. So the cholesterol itself, LDL cholesterol, gets very bad press in a way. But it is a very important component in our body. So we must remember that every single cell in our body is composed, needs cholesterol for its membranes. Hormones are synthesized from cholesterol, bile acids, as well as it's an important precursor for vitamin D, which plays many important roles. And the main organ in our body that does the job of metabolizing cholesterol is the liver. So really about 20% of the cholesterol that we have in our body actually comes from the diet. And we as humans developed the process of synthesizing our own cholesterol in the body. So clearly that highlights the fact that it is a very important component, and humans simply cannot live without it. We need it. And as I said, we have a process in which we can synthesize the cholesterol ourselves, and that happens in the liver. So LDL cholesterol is the main type of cholesterol in human body. Of course, you might have heard of HDL cholesterol. This is the good cholesterol. So also VLDL cholesterol, very low density lipoprotein. Triglycerides, this is the dietary cholesterols. So there are several different types, but a majority of cholesterol in human body is transported in the form of LDL cholesterol. So it's a particle. It's kind of a packed particle with loads of cholesterol inside, and it is wrapped around by a protein called ApoB. So now if you imagine that it is a fatty substance, so if you drop a little bit of oil in a glass of water, you will see that it sort of floats on the surface of the water. It will not easily dissolve in a aqueous solution. So if you think that cholesterol needs to be transported in circulation, in blood, and blood is mainly made of water, this cholesterol needs to be made slightly more hydrophilic. So it needs to like the water. And that's the role of ApoB. ApoB, as a large protein, wraps around that fatty substance, that cholesterol, and makes it easier to be transported in blood. And liver and many other types of cells, but mainly liver, has or expresses LDL receptors. And these are sort of like a 7-fingered hands that are sitting on the surface of a cell, and they would wait, their function is to bind the cholesterol from circulation. And so it is the ApoB part of the cholesterol. So I've marked it here like this green color. And this is the ApoB wrapping around the cholesterol. It is the ApoB that binds to LDL receptor, and that triggers endocytosis. So this cholesterol goes inside the cell where it is metabolized. And next, the LDL receptors can be recycled back to the surface of the cell, or can be degraded. And the process of the recycling and degradation of LDL receptors is controlled by an enzyme called PCSK9. So hopefully you can see there's 3 major players here in that pathway. We have the LDL receptors, we have ApoB that binds to LDL receptors, and then we have PCSK9 that degrades LDL receptors. And that work was mainly done by Professors Brown and Goldstein, who received a Nobel Prize back in 1985 for their work on cholesterol metabolism. They were the first Phage mutations actually came from their lab as well, LDLR mutations. When I look at this picture here, I think they drawn here actually sort of a symbolic LDL receptor. So this is kind of like that hand that is sticking out, waiting to bind ApoB. So if you're more interested into that, you could search for their names, and this is the sort of pioneering work on LDL receptor and LDL cholesterol. So a phage is due to the defective clearance of plasma LDL cholesterol, and that obviously then leads to high cholesterol in the circulation. So thinking about the previous slide, the cartoon, you could conclude that perhaps this faulty clearance of cholesterol could be because of dysfunctional LDL receptors that cannot bind the cholesterol. Perhaps the binding between the ApoB and LDL receptors is insufficient, and that also leads to increased circulation concentration of LDL cholesterol. Or maybe there is an enhanced degradation of the LDL receptors because of the PCSK9 function. And indeed, mutations in these three players in LDL receptor pathway are known to cause a phage. So we've got mutations in LDL receptor gene that lead to either lack of receptors or dysfunctional receptors. We know that they cause familial hypercholesterolemia. Mutations in the ApoB protein decrease affinity of the ApoB to the receptor, and they also lead to a phage. And mutations, these are gain of function mutations, so they improve the function of PCSK9. And so PCSK9 starts degrading LDL receptors at a higher rate, and that leads to less receptors being available on the cell surface to bind LDL cholesterol. So these are the three important players in a phage. There are novel genes coming up, and I will cover a little bit more about that later. Probably most convincing recent mutation was reported in ApoE gene. There is one single variant. It's a deletion of leucine at position 167 that has been suggested to lead to a phage. There was a nice family co-segregation study. But looking at our population data, recent ones, we think this variant might be slightly milder than the known phage mutations. There is another form of a phage. It's a recessive familial hypercholesterolemia, which I will not be covering today, but I want you to be aware of it. This is due to mutations in LDLR adapted protein 1, and that protein is responsible for formation of those coated pits which are involved in the endocytosis of LDL receptors. But this is a recessive phage, so slightly different inheritance pattern, and we will not be talking much about it today. So if we look at the genetic causes overall, about 80 to 90% of all FH mutations are in the LDL receptor gene. So this is the most common cause of FH. ApoB, it's a very large gene, mostly understudied. I think now with next generation sequencing data, we're learning more and more about the gene. But there is one very common mutation that affects the binding of LDL receptor to ApoB, to LDL cholesterol. It affects about 6 to 10% of all FH mutations. It accounts for 6 to 10% of all FH mutations, but leads to slightly milder phenotype. And again, we will look at that a little bit later. Gain of function mutation in the PCSK9. Again, this gene is difficult to study because you need to distinguish between gain of function and loss of function, so that requires functional work, studies, cell work, for example. but that mutation is very rare, affects about 2% of FH cases, and however leads to most severe cause of hypercholesterolemia and highest risk of premature coronary heart disease. So important to look for it. So this is kind of the proportions of these three genes in terms of the mutations. Mutations in the ones that lead to recessive FH are relatively rare. So again, we will not be covering that. ApoE mutations, so this is the new gene. Here, the frequency overall, it's not known yet. It's something that we are working on at the moment. We think it's higher than the mutation in PCSK9, but again, the phenotype is milder, so it will be something more like the ApoB mutation. So there's plenty of different variants that cause a phage. Last time I looked, there was over 1,700 different monogenic causes of the phage reported worldwide. Here are two main mutation databases for a phage. I will show you a little bit more about that later. But the bottom line is that the cause of a phage is highly heterogeneous, so many different causes. There are some populations, however, where just few mutations account for the majority of a phage, and for example, in Greece, Finland, or French Canadians. It is important to know that if you want to design an effective genetic testing for a phage, and so that you know how to, what methods to use, how to adapt it. I think I have a question here, but okay, maybe that's, sorry, that's not me. Okay. So how frequent is a phage? I think 10 years ago, if someone asked me how frequent is a phage, we would classify a phage as a rare disease under the definition that we use here in Europe. It was the old sort of estimated frequency of heterozygote mutations was 1 in 500. So 1 in 500 individuals in a population would inherit a mutation that leads to a phage. But recent studies, genetic studies based on next generation sequencing methodologies, discovered or confirmed more and more variants. And so the frequency now we estimate is about 1 in 250 or 1 in 300. So suddenly the frequency kind of doubled. And this is because we have much better methods now to test the mutations. And in the previous era, let's say, it was only an estimation. So how about homozygotophage? So this is when both parents are affected. They both have an FH mutation. And so when they have children, they have 25% chance that their offspring will inherit both of those faulty variants. And so that will result in homozygotophage. Homozygotophage, it's a severe case. It's children at the age of 5, 10-year-olds develop cardiovascular problems. They require very specific treatment, and I'll show you a little bit in the next part of my talk about that. So it's in a way a tragic event. And we used to think that it affects 1 in a million individuals, but again, genetic studies suggest that actually it is much more common. It's more like 1 in 300,000 individuals who have homozygotophage. Therefore, in the UK, we estimated that we've got about 200 cases of homozygotophage children, actually we don't know at the moment where they are, so we're still kind of behind with diagnosing them, which is quite astonishing, because I think that the phenotype is very, very obvious. but obviously not many people test the cholesterol level in children. We will again discover a little bit more about that later. But yes, so it isn't a rare disease. It affects more individuals than we previously thought. And there are even populations where FH is even more common. 1 in 100 has been estimated to affect French Canadians, Christian Lebanese, or South Afrikaners, or Ashkenazi Jewish individuals. So these are sort of genetic isolates. And it can go the other way. So for example, in Iceland, also a genetically isolated population, a recent report using next generation sequencing data suggests that FH frequency was 1 in 800. So they lucky, they've got much less FH in their population. But I think the bottom line is that FH remains highly underdiagnosed. So this is a figure from 2013 that shows you the percentage of individuals diagnosed with FH in each of those countries where we had data for. So Netherlands are doing very well. They had 71% of FH cases identified. They had very well implemented cascade testing program where you trace families who are affected, and then you identify all the affected individuals, and then you can go and expand your search going through another generation. And so they've been doing very well. But imagine this is actually based on the previous frequency, the historical frequency of 1 in 500. So if you think that actually mutations affect 1 in 250, you will need to divide all those numbers by 2. So we're not doing very well at all, not even the Netherlands. UK as well, our figure was here 12%. I think since 2013, we are getting better. So if you divide that number, we probably have about 7 to 10% of FH cases currently identified. So there's loads to be done still. Those individuals are walking around not knowing that they are affected, and more needs to be done to prevent the heart attack. So then the question arises, how can we systematically identify FH individuals? And why is this very important? Why do we have to put loads of effort into that? And first, to answer that question, I think we need to look at the clinical diagnosis of FH. So how can you see that someone has FH? And the major criteria is cholesterol measurement, of course. So we've got total cholesterol of 7.5 millimole per liter or LDL cholesterol of 4.9 millimole per liter cutoff in adults. This is using the Simon-Brome criteria, which is one of the main defined criteria to identify FH. To look at children, the cutoffs are slightly lower. If you remember on one of my first slides, family history is very important. To analyze family history, there is a clear pattern of autosomal dominant inheritance that strongly suggests it can be familial hypercholesterolemia. So if you have the high cholesterol and family history of either high cholesterol or premature heart disease, then that will classify a patient as possible FH. Now, in addition, if an individual has tendinocyantomas, so this is kind of like a physical deposition of cholesterol in the tissue. It can be observed around ankles. Some people have around eyes. That classifies patient as definite FH. So there's clearly something going on. There is a high burden of cholesterol. Those individuals have been exposed to high cholesterol levels for many years, and so you've got a build-up. Also, genetic testing is in place, and we will talk much more about it in the next few slides. If a patient has a Phage mutation, obviously that gives them definite FH diagnosis. Worldwide, there are two different systems as well. We've got the Dutch scoring system to define FH, and there is in the US MedPed criteria. All of them roughly perform in the same way in terms of identifying patients who carry an FH mutation. And now about 40% of those possible FH cases and 80% of definite FH cases actually have an FH mutation in the known genes. So clearly there is something else going on. We're missing about 60%. And the second part of my talk will be kind of explaining why this is the case. So I think that suggests really that those criteria, although they work not bad, they are not perfect. So there is perhaps other ways that we can we can improve the criteria in order to identify those individuals who have an FH mutation. So we know the clinical diagnosis of FH. So now, why is it important to identify those individuals early? And this is because patients have premature heart disease. So why do they have heart disease so early? And this is because of the LDL cholesterol burden. It has been estimated that 50% of men and 30% of women will get CHD by the age of 55 years. Here is a plot of that burden, so LDL cholesterol exposure in terms of multiplied by the years. So how long individuals have been sort of exposed to high cholesterol levels. And I said earlier to you that it happens from birth, really. FH mutation carriers have higher LDL cholesterol than non-mutation carriers. And so here is a plot of FH individual in blue, in red, non-FH individual. You may say first look, oh, they not really differ that much, but if you think about looking into a 45-year-old individual with a phage, their burden of cholesterol is as much as about 72-year-old healthy individual. So really there is a big difference in terms of age. They are exposed to high cholesterol much earlier, and the burden is much more severe. And there is a need really for aggressive treatment to reduce that burden, and that treatment really should be started early in childhood. The current recommendations say to start starting treatment in FH children by the age of 8 years. So yes, and the early diagnosis and treatment will give you a good chance to prevent CHD. So you may say, okay, well, why don't we just screen using cholesterol levels, check cholesterol level in the whole population, and then we all know who is affected. That would be nice, in a way, a cheap method to do that. But the problem is that there is an overlap in terms of cholesterol distribution. So here is a histogram for non-FH individuals in blue and FH mutation carriers in red for children aged 5 to 15-year-olds. And you can clearly see that those FH individuals have higher median cholesterol levels than those unaffected. But you will also see that overlap here. And that's the problem. We struggle with the diagnosis in those individuals. So using a cutoff somewhere in between here, between those two overlapping curves, would give us 8% of the actual individuals who don't have a mutation being classified as a phage. But I think what is the bigger problem is the false negative rate. So 15% of individuals who have actually a FH mutation would not be identified using just LDL cholesterol measures. And that is an issue that actually increases with age. So here is the same graph for 45 to 54-year-olds, and you can see that the overlap between affected and unaffected is massive. And that would lead, if we again take the cutoff sort of in the middle, that would lead to very significant false negative. So you would miss basically 46% of affected individuals if you were to use cholesterol levels as the sort of diagnostic tool. So as mean LDL cholesterol rises with age in every single one of us, the non-FH and FH distribution overlaps. And so DNA testing would be a good solution here to say who is actually carrying an FH mutation and who isn't. So let's have a look at the genetic testing. This is something that is very useful tool. It gives you a crucial information for some screening strategies, which we will discuss later. If you really are into this kind of methodologies, we have recently published this review in the Journal of Lipid Research. It's quite a long review where you can find a bit more about the methods that were used in the past, that are used currently, and that perhaps will be applied in the future in terms of identifying FH cases. So let's have a briefly look at those methods. In the past, we had methods that were really not specific, but there was no human genome published. We were not aware of the specific sequence of the genes that were identified to cause FH. So it was, there was loads of information missing. But one of the methods to identify what allele mutation that causes FH may be located was restriction fragment length polymorphism, or RFLP, which is a cutting a fragment of DNA with a site-specific restriction enzyme. So restriction enzymes come from bacteria. They are sort of a preventive mechanism to destroy viral DNA. And so they cut DNA at specific sites, and we know the sites, and we could, if you know of polymorphisms that occur in your gene, you might want to use that kind of technology. So what to apply that methodology in terms of identifying disease, you require rather large family tree and many samples to be tested to decipher who actually carries, who inherits the mutation. So in this case, we have an example of the restriction enzyme called PVU2 that cuts at a specific site. It either cuts if you have the variant, or it doesn't cut it. You get sort of, you can see different size of the bands on Sadan blot. So in terms of this family, you had the proband here. We can, after analyzing, you can see that the phenotype, FH phenotype, co-segregates with the V1 allele. But the specific variant is not identified. So we can then go further. Now we've got the technologies to sequence DNA very quickly and cheaply, but previously that kind of methodology was used. So the index case in this case needs to be heterozygote. So it doesn't work for homozygote cases. It needs to be heterozygote for the restriction enzyme cutting site. It doesn't identify, as I said, specific mutation, but only sort of looks at the inheritance pattern and mode in the family, and it requires rather large family tree. So not very effective method. We then moved to different assays. There was ARMS kit, which you could sort of buy a ready mixed assay that would test for 20 most common FH mutations. It was a nice test that when I was doing my PhD, I actually applied that. It was still in use, but again, it looks at only 20 mutations. It needs to be sort of specific to the population because you're looking at the most common mutations in your population, and that varies between different countries. And it is only 20 mutations. If you remember from one of my previous slides, we know now of more than 1,700 different cases, different mutations that lead to FH. So if you think of 20, it's really pretty bad. We then had HRM, the high resolution melting, and here is a lovely curve of that. I think your Professor Ali Akhtar used that method in our lab when he had when he was doing his PhD. So you may want to ask him for details. But it's a lovely method that based on PCR that you would be looking at different melting curves. So you make your PCR fragment and then you melt it, and because of if a person has a variant, that affects the melting temperature. So here is the temperature on this axis, and you can clearly see these individuals in blue are unaffected, and the red ones actually carry a mutation. So again, it doesn't tell you what exactly the mutation is, but you can see the difference. And then you can take those individuals and sequence the fragment of the DNA to identify the specific variant. It was, as I said, lovely method, but quite laborious actually. You need to divide your DNA into chunks and then do it over and over for every exon of the gene. So yes, there are still some limitations to that. Another assay, MLPA, multiplex ligation dependent probe amplification, important assay for detection of large rearrangements within the gene. Again, this can also be done these days by next generation sequencing. So these methods were definitely more accurate than the RFLP, but they are still very laborious. They can be costly as well, and there's multiple steps that are required. And so if you've got multiple steps in the lab, that leads to higher chances for human error, unfortunately. So some things could be missed. So what is the present really in terms of genetic testing for Phage? It is the next generation sequencing methods, more precisely targeted sequencing. So here is an assay where we targetly, we select fragments of the known genes from the whole genome and genes that are known to associate with a phage, and they are all, the libraries prepared so you pull out all those regions from LDLR, ApoB, PCSK9, you can put in ApoE, you can put any other novel gene that you identify, and they are all sequenced at the same time in one assay, so much less time required. There is less room for an error. And you can also, as I said in the previous slide, detect the large rearrangements within the gene when you plot the coverage, so the read depth of the exons. Here is an example. And again, if you want to read a little bit more, you can go to this paper here. It's an example how you can use the next generation sequencing data to identify, in this case, it's a deletion of exons 3, 4, and 5, and 6. You can see the read depth is much lower than the average, and that suggests strongly that there is a deletion of those exons in this patient in LDL receptor gene. There are still, unfortunately, some limitations to those methods, and these are mainly due to genetic variation interpretation. So we are identifying loads of novel variants, and we are not quite sure how to interpret them. So again, functional studies or family co-segregation is important to prove that a specific variant leads to the phenotype. And it also requires specific bioinformatics skills, which still there seem to be a shortage worldwide. So how about the future? I think next generation sequencing is here to stay for sure. And we are moving, if you have loads of money, we are moving towards whole exome or whole genome sequencing. And in that case, you can look at not only the rare variants, but also common variations, so SNPs, polymorphisms. And that leads me to the polygenic risk scores, and we will discuss the polygenic cause of a phage later on. But that method looks at the whole genome. So it's not only selecting specific genes that cause a phage. You can find all sorts of stuff, to be honest, and that's why, because we're finding so much. One of the biggest issues is that, again, the interpretation of the data. Plus, the methods are still relatively expensive, and you also require high performance computing in addition to the bioinformatics skills that are required. And what else we need to do is to ensure that we have the reference population data that is diverse. There is, unfortunately, in genetic studies, still bias towards individuals of European background. And so most of the databases that we can use as our controls or our reference come from individuals of white European background. Therefore, if we are studying different population, there is a clear discrepancies if you identify a rare variant. You may identify a rare variant that is not present in the Europeans, but perhaps is very common in South Asians. So one may get excited that we identify a novel rare variant in I don't know, in Germany, but then you go and look at a large genetic database in India, for example, and you'll find that that variant is actually quite common in that population, and it doesn't associate with any phenotype. So there is loads of effort at the moment going into that to make the reference genome much more diverse. That's something that we definitely need. Okay, so let's come back to Mike's family. So again, if you remember, Mike had a heart attack at the age of 35. And so after looking at his cholesterol levels, the doctors referred him for genetic testing for a phage. And so his DNA sample was sent for targeted sequencing. So this is the slide where I show you the present state-of-art DNA testing for a phage. So his DNA was sequenced for the Ldl receptor, for ApoB, and PCSK9. And after, I don't know, I think the turnaround is about 3 weeks, the data were analyzed, and what was identified is that Mike has an LDLR mutation. It's a premature stop codon formation. So the star indicates that there is a stop codon. So this is the cysteine 231 amino acid that one base pair leads to premature stop codon. So basically the gene terminates at that position. And this is relatively early in the gene. LDL receptor gene has 862, if I remember well, amino acids. So clearly, if you cut it at 231, you're missing a big chunk of the gene. So it's a null mutation in a way. So the question then was, okay, we did the targeted sequencing on Mike. The question is obviously, do his children, can his children be affected? This is a very important question that family needs to consider. Do they want to know? They should know, I think, in terms of we've got a treatment for the children. There's loads of studies showing that introducing treatment at early age can prevent heart disease. So very important question. And that would be sort of part of cascade testing. These two little people are first-degree relatives of an individual who has an FH mutation. So it is very fair to look at their DNA. Now, because we know Mike's specific mutation, we don't have to then pay for expensive targeted sequencing, but we can only look if they inherit this specific mutation. So a Sanger sequencing of a specific fragment of the gene can be applied for a quick test. And that's what happened. And unfortunately, Daniel has been identified to also inherit the mutation. I think this could have been concluded from the fact that he has a higher lipid level than his mum, and he's relatively young. But if you think about the overlap in the cholesterol distribution, it could go really both ways. So the DNA testing really gives him accurate results here. On the other hand, Matthew tested negative. So here, big sigh, he's relieved he has not inherited the disease. So I suppose this is very positive news for him. And in a way, I think for Daniel, it is positive because now he knows he can adapt his lifestyle, he can have the treatment, and he can prevent the disease. Obviously, mum was not tested because she's not genetically related to Mike, and her cholesterol levels are within normal range for an adult. So yes, this is how you can implement DNA testing to screen family. So how about mutation interpretation? I think I've looked through your program, and I've got a feeling that you might have already talked about it. I have mentioned to you before that this still remains a significant limitation in terms of DNA testing for a phage, and more efforts to prove that a specific genetic variant causes a phage are needed, especially for variants of unknown significance. So since we have the next generation sequencing and we are being screening through all the sequences of the 3 genes, what we identify is that a significant proportion of them, of the variants, we cannot say if they cause or not cause the disease. And this is important from genetic counseling point of view. So imagine that we found a VUS or variant of unknown significance in this family, telling them that we don't have enough information to prove that their variant identified causes a phage. It leaves them sort of in limbo. They don't know if Daniel is affected. They don't know what to do really. So I think having diagnosis is very important for family. It gives them, you can then action basically, and you know what to, in a way, to expect. So sorting out those variants of unknown significance is important. And here is a graph of the CleanVar. So this is another database that collects all variants associated with different phenotypes. So here specifically, we looked at variants that affect that are associated with a phage, and benign are the yellow ones. So these are the not affecting, so not pathogenic variants. The pathogenic are the ones in red, and the VUS is the variants of unknown significance are in blue. There is still another group of unclear, which I think are because of the wrong submission to the CleanVar database. CleanVar is a sort of open access database where anyone who finds a variant can then submit it to that database. And believe me or not, many people would submit things that don't make sense. So they would put, for example, wrong base pair or wrong amino acid number. And we know the standard sequence of LDL receptor, for example. So if someone is submitting something that doesn't make sense, we can't really analyze it. So yes, majority variants in LDLR, I think we're doing very well in terms of interpreting the variants. We know a lot about the gene, about its structure, about its function. And so majority would be classified as pathogenic variants. And we've got only 8% here of LDLR variant being difficult to interpret or uncertain. In terms of ApoB, it's a different story, and it's the same for the PCSK9. So I said these two genes are still understudied, especially ApoB. It's a very large gene with loads of different variants, loads of rare variants. And so majority of the variants that we find in ApoB are variants of unknown significance. So very important to find an assay that can define the effect of those variants on LDL cholesterol. In PCSK9, again, it's a similar issue when you have to distinguish between loss of function and the gain of function variants. So family studies are a good way to analyze the effect of variants. So if the variant clearly co-segregates with the family phenotype, with a phage phenotype in a family, then we can see that that gives you a strong evidence that yes, perhaps that variant really truly causes a phage. Large databases, again, from what's important, diverse populations to establish frequency of those variants is also important. These are, again, the databases that are gene specific and curated. So this is people who are really experts in the function of, for example, LDL receptor. We had Sarah Lee, Mafalda Bourbon, who are people who really sit down and analyze variant by variant and decide which variant is pathogenic and which one is not. So there's loads of different criteria that you need to tick to define a variant as a pathogenic, to avoid sort of false diagnosis. Those criteria have been defined, for example, by the Association for Clinical Genomic Science back in 2020. So you might want to follow those guidelines to classify a variant. If you go to these databases, these guidelines have been already taken into consideration. Not on ClinVar, unfortunately. So ClinVar is kind of, it's great thing to see what people find, but as I said, the interpretation can be a little bit subjective. So you don't know if the person who is actually submitting the variant actually have gone and read those guidelines. So yeah, it's a big job to do, to interpret the variants. You don't want to give a patient false positive diagnosis, for example, if you find a variant, because that's not right. And it's the same with false negative, giving someone a diagnosis saying, oh, you don't have an FH mutation, you can go away and just look after your diet. It's also not right. So this is important part of your genetic diagnosis. So why does it really matter knowing specific mutation? We know that a patient has high cholesterol, we know they've got a mutation. Why do I want to know if they've got mutation LDLR, ApoB, or PCSK9? Well, I think it has some value for sure. First of all, we've observed that the risk of premature coronary heart disease differs by different phenotype or different gene mutated. And the PCSK9 mutation carriers have the highest LDL cholesterol, and they also respond to statin treatment quite poorly, and that, I suppose, leads to the high risk of premature CHD. Whereas ApoB mutation carriers have quite low cholesterol levels to start with in comparison to other mutation carriers, and their risk of CHD is also relatively low. Recently, we looked at children with FH mutations. This is a large database of different registries across Europe. And what we were able then to do is to further divide LDLR variants into different classes. So, for example, variants reported in the promoter, missense variants, splicing variants, nonsense, and large rearrangement within the gene. And what we found is that the LDL are large insertions and deletions, as well as nonsense mutations. So these are the ones that Mike in our example family had. So a variant that leads to premature stop codon. These are one of the most severe ones. So this is when you don't have a functional LDL receptor. So in a way, we are leading towards this kind of precision medicine approach where one can look at a variant and think, all right, I know I need to give this person a more aggressive treatment. I need to, for example, see them more often in the clinic, especially if they have a PCSK9 mutation, because I know that they might be at higher risk of CHD. Obviously, defining those findings requires large number of patients to study, but I think we are getting somewhere with it. So yeah, definitely a step towards some precision medicine approach here. Another question that I get asked is, okay, so we have the FH mutation, we know the phenotype, for example, they've got high cholesterol, but again, we have now looking at population data, there is a significant proportion of people who have an FH mutation, but their cholesterol level is actually okay. It doesn't go over the diagnostic threshold defined by Simon-Brown criteria, for example. And so what do you do with that individual? Do you treat them? Do you see them in the clinic? I mean, they don't have high cholesterol, so they should be right healthy and they can look after themselves. Well, we need to be very careful with such conclusion. There's more studies showing that the risk of cardiovascular disease is always higher in FH mutation carriers, no matter what their cholesterol level is. Here was a study from Kara et al. That showed a difference. So here is a LDL cholesterol, different sort of grouping of LDL cholesterol. This is now given in milligrams per deciliter, so different cutoff than what I presented to you before. But basically, those individuals that definitely don't have high cholesterol, and these guys are kind of normal cholesterol levels. And you can still see in that dark purple, these are the mutation carriers at any level of cholesterol levels, that mutation carriers have significantly higher risk of or odds ratios for coronary heart disease. So yeah, it's I think it's important to know that mutation. And I think what we need to start thinking is that mutation itself, it's in a way an independent risk factor, independent from the cholesterol level. A recent study, very recent, just published in the Circulation Genomics Precision Medicine, again suggests that after adjusting for LDL cholesterol levels, the mutation carriers are still at significantly higher risk of CHD. So yes, again, another evidence that CHD is independent in terms of mutation carriers, independent from the exposure to LDL cholesterol. And of course, knowing the mutation is also a very crucial piece of information for screening strategy. And that's what I want to talk to you about now. What can you do? How can you take all that knowledge on mutations, on LDL cholesterol distribution, on all the overlaps, and how can you utilize that information to effectively identify FH individuals? So screening approaches. Well, we know that early diagnosis and treatment are very beneficial for FH patients. So there is no doubt, I think, that we want to identify those individuals. And of course, preventing cardiovascular events gives significant savings to the national health system through the reduction of those events. They are very costly. If a person ends up with a heart attack in the hospital, that costs much more than prescribing statins from the age of 10-year-old. Statins are relatively cheap. And so there is no doubt that this prevention or early identification before any cardiovascular events is beneficial in terms of money. Now, cascade testing, so this is when you trace families using the DNA information, is very cost effective. So because it's autosomal dominant disease, you're expected to find 50% of your first degree relatives also being affected. And that 50% is relatively high for screening strategy. And so it has been implemented in the UK. And now knowing the family history, the family mutation is a key piece of information to make it cost effective. So if you remember again, Mike's family, the next generation sequencing assay was first applied. But then once the mutation is identified, you can use cheap Sanger sequencing to confirm the variant in his relatives. And it's also very important evidence to start early treatment in children. So once there is a mutation, there is no doubt that that child should start thinking about receiving treatment. But if you think about cascade testing, you still need to identify that initial index case to then trace the families. And it is the issue with identifying index cases that we are way behind with. So again, going back to Mike's family, he had a heart attack. That's too late to identify him. That's how he became the index case. We need to make sure that we find index cases way before that. And so one of the approaches to identify early individuals who have an FH mutation is child-to-parent screening. Now, this is when it's currently being tested actually in the UK. There is a large pilot, and I think it's 30,000 children who come to their general practice for MMR vaccination. And while they're having the vaccination, a blood spot sample is taken and their cholesterol level is measured. And if their cholesterol level is higher than the average, it's I think over a 90th percentile, then they are invited, those children are invited for DNA test. And then the parents as well are being tested. So obviously you expect one of the parents of the affected child to have an FH mutation. So we kind of going backwards from a child to a parent rather than from a parent to a child. The idea is good, I think. We would be identifying those children very early on. There's loads of issues with sort of logistics of it. But as I said, it's currently being tested and it might be at some point implemented. Another way is to opportunistically search through electronic health records that GPs have. So here, you know, test cholesterol levels. In England, we test, or every 40-year-old individual is invited for sort of general health check, and cholesterol levels are then measured at that time. If their cholesterol is above a certain point, then they would be perhaps invited for DNA testing. But this isn't systematic, really. It hasn't been well implemented yet, although the NICE guidelines, so this is sort of a policy for healthcare in England, recommends doing that. It hasn't been going very well. So we still have the issue of underdiagnosis. Majority of FH cases, unfortunately, remain undiagnosed. So why is it not that easy to detect individuals in a population? Very recent work here by my PhD student, Jasmine Grattan. What we did is we used the UK Biobank resource. So this is 500,000 individuals of UK. They are mainly white British, but we've in this study focused only on the white British to make it sort of cleaner. The data for whole exome sequencing is available, and it was available at the time of the study for 140,000 individuals. So we looked into the 140,000 individuals of UK Biobank. We extracted DNA information for LDLR, ApoB, PCSK9. We looked at their mutations, and we identified among those 488 mutations. So the prevalence of phage mutations in the UK Biobank cohort was about 1 in 288, so relatively close to the 1 in 250. And yeah, and now the question is, what cutoff of LDL cholesterol could be used to, for example, screen those individuals, to use that information to screen individuals in a population. So if you were to use the 4.9 millimole per liter cutoff, that's the one that Simon Broom criteria uses, we would find almost 40% of individuals who have a mutation and have the phenotype. But we would miss 60%, so 292 roughly individuals who have a mutation would be missed. So in blue, you've got the cases missed. And obviously, the higher the cholesterol, the more cases you identify. But if you are designing a population screening, you need to, of course, think about all your resources. So what is your point? You will be taking those individuals and sequencing all of them to identify the mutations. So it's really matter of discussion in terms of on country basis. What are your resources? How many people can you screen? How many people can you sequence? What is your budget? So roughly this is, we just showed, we modeled how how many individuals you would be able to identify if you use those specific cutoffs of LDL cholesterol. But it isn't ideal, right? I mean, you're still missing 60%. So on its own, using LDL cholesterol actually it's not giving good results. It's still a poor predictor for FH mutation. So then we, together with Jasmine as well, she applied a machine learning algorithm on the UK, excuse me, the UK Biobank data to identify if there are other predictors that perform better. So, for example, including not only the genetic, but also environmental measures that could together improve the detection rate. And so this is a lasso model. It's a statistical method, machine learning algorithm. And what we found was that actually triglycerides were one of the best predictors. The lower the triglyceride level, the higher the chance that an individual carries an FH mutation. I mean, not on its own, but in combination with other factors. So LDL cholesterol, of course, information on statin use, so relatively obvious things. We also included a polygenic risk score, and I will talk about that a bit more later. But here is the AUC statistics, and you can see the LDL on its own is in blue. It's performing much poorer than the red line, and this is the lasso model. So the lasso model definitely improves the prediction of FH mutation. And so if one has access to large data sets, phenotypes, genetic data, applying a machine learning algorithm can improve the detection of FH mutations using electronic health records. Perhaps we're not there yet. This requires, of course, some infrastructure as well, but I think this will be way forward in the future. Right, but I don't know if you remember now on one of my slides before I said, okay, but FH mutation can be found in about 20–35% of individuals who are diagnosed with possible FH. So clearly there is loads of people with high cholesterol who do not have a FH mutation. And the detection rate is much higher in definite FH cases. So these are individuals with tendon xanthomas, but still there is a percentage of sort of missing inheritance. So what are the reasons for that? Why still we can't explain high cholesterol, high LDL cholesterol in some individuals? And several issues probably could be highlighted here, and I will discuss them in the second part of the lecture. So I'm kind of I just want to leave you with those thoughts now for the break that will happen soon. So again, if you remember from some of my previous slides, majority of the FH cases would have a mutation in LDL receptor, followed by the ApoB. Then about 2% of FH cases are due to PCSK9. What I will talk to you about next is the idea of polygenic hypercholesterolemia. And yet, if we explain some chunk, I would say about at least 13% of definite of possible FH cases having polygenic hypercholesterolemia, although it might be more than that, there is a proportion of individuals who are left without any explanation in terms of genetics. Some of the points highlighted that came up in our studies were that there were still technical reasons why some mutations were missed. Perhaps they are located in the gene regions that haven't been previously screened. So you can sort of relate to the previous old methodologies that were used. They were not data accurate, they were laborious, they were prone to human error, and I will show you examples of that. Perhaps really there are novel genes involved in FH, and I will talk to you about efforts to identify such novel cases. This will include whole exome sequencing, whole genome sequencing. Another issue is perhaps there is overdiagnosis of FH. So if you remember the Simon-Broom criteria, they are mainly based on cholesterol levels, family history. You will be surprised how many individuals that when I analyze the data for don't have a clear family history. And I think that's straight away rings to me a bell. Okay, perhaps that isn't the autosomal dominant FH. So perhaps they have high triglyceride levels. Those things, I think the overall bottom line from that is that the current diagnostic criteria in terms of clinical FH are not very precise. And I hope I just showed you as well that LDL cholesterol isn't a great predictor for an FH mutation. And of course, the polygenic hypercholesterolemia. This is a relatively new idea. Polygenic risk scores are the hot topic currently in clinical genetics. How can we use them? Are they really useful? It still remains a question, but I will show you after the break exactly how, where we are with it and where we are going in terms of polygenic causes. Okay, so I actually finished my first part of the talk. if you would have any questions for now, that would be great. If not, we can break now and then come back after the break. So it's up to you now. I'm here to answer any questions. I really hope that isn't too much of a specific science. I think my bottom line is that I want you to understand that this is a good example disease, and it can be translated to many other genetic diseases. Phage is great because it's relatively common, and so we have loads of data, loads of resources to study it. And I remember when I was starting my PhD, I thought that lots has been done on a phage, and I probably won't have much to add. But actually, the boom in next generation sequencing has enabled us to add much more knowledge to that disease. Okay, so what shall we do now? Can I speak to the organizers?
Thank you, Martha, for such a nice talk and very informative topic, which is very common in, I think, worldwide. I have one question.
Okay.
If somebody visits the lab and he has the result that he has some high levels of cholesterols while doing lipid profile, particularly LDL, and he doesn't know about his genetics. So what is your suggestion or recommendation that he should revisit the lab and should do the lipid profile at how long time interval? Could you suggest?
How long in? Sorry, I don't understand.
I mean, if somebody tested today and he has higher lipid levels, and is there any recommendation from your group or internationally that should do it after a certain time?
The DNA testing?
Not DNA testing. Lipid profile.
Repeat lipid profile. Is that what you mean?
Yeah. So he can do the best for his lifestyle, and he can reduce. He can manage the lipid profile in the normal range.
I see what you mean. Okay. I'm not aware of specific guidelines on that, to be honest. When you're saying that they are not aware of the DNA cause, do you mean that they would receive negative tests, or that wasn't done yet?
Yeah, because I'm just talking particularly related to our country, because we are not very much facilitated with the DNA testing. So most of us might have not been tested for lipid with the higher lipid profiles and not tested for these ApoE, ApoB, and LDR and PCSK9. So we don't know our genetics. But because this lipid profile is cheaper, so one can regularly monitor his or her lipid profile and then can do the best things which are suitable for the lifestyle.
So I think the recommendation, I think the issue, so I'm not a doctor, but I know that the clinicians that I talk to, if they see cholesterol level, especially at the level of clinical diagnosis for a phage, so let's say the 4.9 millimole per liter, that is high. So I know that they will prescribe statin straight away. They will prescribe statin, and they will monitor the response to that statin. So the patient, I think if it's a new patient, it's still, the guidance are not specifically there, but I would say if it's a new patient, that they would be seen probably in about 6 months' time to see how they are doing on that statin, if they're responding well, or if they need the dose to be changed, or even statin itself to be changed. So the lipid profile would be repeated at 6 months. If a patient is responding well, they are managed well, they would be seen in the general practice, I would say, on annual basis. I know that FH patients, if they have a mutation, they are also seen at annual basis if they are responding well to their treatment, but they are seen by a specific clinic, a lipid clinic, so a lipidologist. So I think if cholesterol level is high, there is no need to wait. Triglyceride levels now, if these are high, they suggest that perhaps dietary intervention may work. But still, as far as I know, statins would be prescribed. There is a clear evidence that the earlier you start statins, the better the projection is. So yeah, there is that shouldn't be delayed in a way. Does that answer your question? Does that help in any way? Don't feel bad about the fact that you don't have DNA testing set up yet. I mean, DNA testing for FH in the UK has only been approved about a year ago by the National Health System. Whatever we were doing before was research. So there was loads of sort of research. The charity, the British Heart Foundation, major charity, was sponsoring most of the work before. So it wasn't very systematic. Yes. So yes, I understand that focusing on LDL cholesterol lipid profile is a big part of it.
Hello, Martha, can you listen to me?
Yes.
First of all, I must say that it was a very nice presentation and you elucidated very well. Now coming to the question, my first question is that what I perceive from your presentation is that there is a direct relationship between FH and LDL, which is a bad cholesterol. Whereas VLDL also come under the ambient of bad cholesterol. So what I want to know from you is that any relation between FH and VLDL? And the second question is that you have mentioned multiple testing procedures for FH disease, but I was waiting that might be you will told us some preventive measure or some treatment regarding this disease, because I have read somewhere that some herbs are there which can be very fruitful or useful for the lowering of LDL.
Mm-hmm.
So what's your take on it? Thank you.
So VLDL, we're not really looking into that, to be honest. LDL is definitely more specific to FH. So I can't really tell you in terms of FH how to analyze VLDL. I don't think that's not the risk factor that we are focusing on. And as far as I know, in terms of cardiovascular disease, it hasn't got much value in terms of prediction and being a good risk factor. Your second question, so again, I will be talking about treatment on the next slides, on the second half of my talk, but I am not touching on any in terms of herbal or dietary interventions. This isn't recommended in terms of FH. FH is a very clear that it will lead to cardiovascular disease, even if just, so I mentioned previously that 20% of our cholesterol in the body comes from diet. And here we have a mechanism in terms of FH, is the sort of our body cholesterol production that is, and clearance is not going well. So focusing on diet is not very effective. I mean, diet, you mentioned herbs. To be honest, I don't have knowledge on that. The only studies that I analyze is on statins or novel treatment that I will discuss to you, but these are not based on herbal findings. So sorry, but that's beyond my expertise. Do you have studies that you think are showing good effect in terms of those substances?
Yes, have you ever heard about oats, barley? Definitely you have heard about oats and barley.
Yes.
These are
I
basically
eat
cereals.
myself porridge every day, so yes, there are definitely reasons.
There is a long-chain polypeptide named as beta glucans. They are commonly found in oats and barley, and they are very effective in terms of reducing the level of LDL and VLDL. Even they sometimes, it has been also observed that they enhance the level of HDL as well. So have you ever go through the...
Okay. I apologize, I don't know much about it, but the recommendation is, so for example, in terms of Phage patients, you need to lower the cholesterol level by 50%. So if you can do that with your
Martha, I will share some data with you.
Okay, yes, please do. If the cholesterol can be lowered by 50%, I think that's great. And perhaps we will talk about the polygenic hypercholesterolemia next. And this is a disease where the environmental factors will have a bigger play than the genetic ones. So if we start moving away from monogenic causes where you really can't do much, but try to have sort of aggressive even drug treatment. In terms of polygenic hypercholesterolemia, the dietary or your lifestyle has much bigger effect, and perhaps agents, as you just mentioned, may be more successful.
Okay, thank you.
Okay. I think there is a question. You're welcome. Just getting thank you on the chat. So shall we break now for...
We have one more question, Dr. Martha.
Say that again, sorry.
Hello, ma'am.
Hello.
Good afternoon. I'm Dr. Amina. I want to ask one question. What is the identification criteria for FH patients? Are your universities linked to any tertiary care hospital or anything else?
So identification criteria. So I mentioned to you the clinical diagnostic algorithm or the criteria, the Simon-Broom, the Dutch scoring lipid system. So I think majority of FH cases would come from GPs, so general practice. And I mentioned in England, individuals at the age of 40 are offered a health check, which includes cholesterol measurements. If their cholesterol is above those cutoffs for clinical diagnosis of FH, they would be then referred to lipid clinic. In terms of hospitals, I think these are the cases that would come with a heart attack, myocardial infarction, so already a cardiovascular event. And that's how I presented to you the Mike's family. So it is in a way too late because the disease wasn't prevented, but they would then come that way. So in terms of identifying systematically, the only implemented at the time system is the cascades testing. So once you have an index patient, you would be screening their family members for a specific mutation. And the child parent screening strategy is currently being tested and hopefully will be implemented too. So these are the kind of efforts that we have at the moment in terms of identifying the individuals.
Thank you so much, ma'am.
You're welcome. Okay, shall we have that break? Otherwise we'll miss, and I really need my drink. Sorry, my throat is going dry. I will just pause the sharing.
Okay. Hi, Martha.
Oh, now I can see you. Hello,
There is another question.
Okay.
Good afternoon, Martha. How are you?
Hello, I'm good. Thank you.
All right, I have a question. You just answered Dr. Amna's question that how the patients reach, the samples of the patients, they reach your lab. But in a developing country like Pakistan, where the research centers are absolutely, out of touch from the tertiary care centers or the hospitals, how can we do screening or analysis? Because I'm pretty sure this particular familial condition is very much prevalent in a country like ours, where consanguinity is culturally promoted.
Yes.
Culturally and socially, most of the familial disorders are linked to intermarriages and consanguinity, cousin marriages. So it's very much prevalent in Pakistan, and probably we have a very good pool for this particular disease. I'm pretty sure.
I'm
But
sure
we do
too.
not-
Yeah.
I'm more than sure, because the South Asian population, India, Pakistan, Bangladesh, has the same cultural norms. Well, that's another thing. So what strategies would you suggest in which we
Yes.
can collect the sample and bring them to the labs?
It's a very
Very challenging for the
It's very
research
challenging.
scientists who are sitting in the labs. We want to do work, but we do not have avenues where we get the patient's sample into the labs.
Yeah, I see. I'm just thinking, so I'm in touch on LinkedIn and Twitter with a person. I'm gonna bring his name after the break. As far as I know, he's been putting some efforts in terms of identifying individuals that just very recently started in terms of children in Pakistan. So I will try to find the contact. Have you? So this issue it's big, and I understand it's a problem. And looking at the experience of UK, where we have the resources, in a way, you would think we can do it much quicker. It has been going very slowly still, and not only UK but Europe. I think the Netherlands was doing a great
And
job.
then again, sorry to interrupt you. Then again, we've got conventional methodologies for excluding the genetic variants, like, we've got reflip analysis, or just small regions which we analyze. But we have meager funds for next generation sequencing or things like that, unless we have a grant from some institute. So these are things which are really restricting our energies, and I would say capacity of doing work.
Yeah. Yes, it's unfortunately still not quite there, isn't it? In terms of we have the knowledge, but to implement it in terms of preventing cardiovascular disease, it's another story. And I understand it will involve not only scientists, not only clinicians, but public health specialists, and it's a big logistical operation to implement such thing. I suggest, I mean, the only thing, every country is specific, that's the thing. So you will know exactly what your needs are. I mean, you would need to sit down with others to work out what would be the best place. You could learn from other countries who have started doing such things. So the European Atherosclerosis Society meeting is coming up in May. And as far as I know, so there is a initiative to drive the diagnosis of a phage. So there was a big Lancet paper recently of different countries overview, and how things can be moved forward, really, because we know that FH is underdiagnosed, and we've been talking about it for years now, and not much has been done. So yeah, I understand your in a way frustration, and I just really hope that something can be moved forward. as I said, I will try to put you in touch. I will send you the contact detail of the person who is trying to do something.
Thank you. Thank you.
Yeah, but I understand it's not an easy task.
Thank you, Martha.
You're welcome. So is there more questions now?
No, Martha, I think it's and do you have another session or?
I do, yes. So I do have more slides after that. We will cover what are the reasons for the missing heritability in FH. We will cover some novel treatment and how you go from genetics to novel treatment. So yes, there is still more things to cover if that's okay. So shall we have a break? What was the planned break now? Was it 20 minutes or can you remind me?
It was, but now you can continue if
I see.
you...
Well, I need 5 minutes if that's okay.
Okay.
I need to get myself a drink. My throat has gone very dry, so
okay.
If you give me 5 minutes, I'll be back.
Okay. Thank you.
See you in 5. Hello, everyone. Is everyone kind of back, or should I give a few more minutes?
Welcome back, Dr. Martha. You can continue now.
Okay. So I just want to, I want to share with you my screen to show you the person that I mentioned.
Yes, we can see now.
So Motas al-Sabah, I think he's from Iraq, but he's been posting the news about Pakistan. So I mentioned that there is some sort of effort started quite recently. So this is a month ago when he mentions his colleague Sabine Khan, the director of newly established Islamabad Pediatric Lipid Clinic in Pakistan, diagnosed the first 2 homozygotophage cases based on the FH symposium delivered to them. So I think, yeah, so this is exciting news. I don't know if you are aware of those cohorts, of those efforts to identify FH cases. This is children, so yeah, I think that's amazing. But there is clearly something already starting, so you might want to try to get in touch with this person or with colleague Sabine Khan. And as I said, Islamabad Pediatric Lipid Clinic in Pakistan already diagnosing children, which is wonderful news. So if yes, I'm not exactly sure what the national efforts would be, but that person will be definitely more in place to help. Did you take a note of that? Because I want to now swap. I'm going to go back to my slides. Okay, so we were here. I got my coffee. So just before the break, I mentioned to you that we're still lacking in terms of identifying genetic causes of FH, that only about 20–30% of possible FH cases, which is really the majority of FH patients, have a mutation. So in a way, you might say, well, the genetic testing is kind of, it's not very cost effective, right? If you only confirm the diagnosis in that proportion of patients. Still very important to do it, I think, if you have the resources. And as a colleague in the audience suggested, that perhaps in Pakistan, you might have quite a few, or a higher percentage of homozygote cases. I think it's very important to know the mutation. So let's think about it. What are the reasons for the missing FH mutations? And one of the first ones that came up was technical reasons. And we only learned about it because there were significant advances in sequencing technologies from the previous methodologies. In our study, I think this is back in 2012, where we applied whole exome sequencing, we identified 25 FH mutations out of 125 patients who should be, the mutations should be detected using the previous methods, such as the HRM. So that was quite shocking. It's a quite big proportion of patients that we said that they are mutation negative, but they actually had a variant. It's just the technology perhaps wasn't quite there. Another issue is that we are still not really looking into the deep intronic spaces in LDL receptor. We're only focusing on the coding regions. This is where we can easily interpret, well, not easily, but we can start interpreting the mutations because they affect amino acid. So we kind of can predict what's going to happen when you have a mutation in the coding region. But obviously the intronic regions are actually much larger than the exons, and they might affect the correct splicing of the gene. And there was a study, for example, one of the studies back in 2018 showing the deep intronic region sequence. They sequenced the whole sort of intron here, and specifically between exon 14 and 15 of the gene. They identified there was a cryptic splice site, which meant that when the gene gets translated into a protein, there is a retention of a region of a DNA which shouldn't be there, and that obviously caused an issue. It shifted the whole reading frame of the gene, and there was a then premature stop codon formation just in the exon 15. So these variants, you can see it's quite deep into the intron. Why were we not looking? They do affect small proportion of individuals. They estimated 0.24% of all FH mutation negative patients, but still I think are if we want to pinpoint the missing causes, this is an important part of DNA that we are not looking at. And we were not looking into those regions because of they are very rich in repetitive sequences, specifically ALU sequences, and they are very difficult to sequence technically. So yeah, it's one again reason why perhaps we can't find a mutation. I want to talk about the overdiagnosis, and maybe our discussion here was kind of leading to that. Clinically, the clinical diagnosis of FH are mainly based on LDL cholesterol. But of course, if you see a patient, you will see loads of individuals with high cholesterol. Now, the question is how high, and what are the other cholesterol fractions like? So, for example, triglycerides. Triglycerides are the indicator of, in a way, dietary cholesterol. And when you look at typical FH patient, they are actually quite lean. They wouldn't be having many issues with diet. So their triglyceride levels are actually low. And I did the study this is a sample of about 300 individuals from a clinic around Oxford area in England. And if you take, so we were looking at the mutation detection and how you can improve the sort of diagnosing of individuals who actually have a mutation. And please don't take me for this. I'm not saying that those individuals who have high cholesterol but don't have a mutation are not important or should not be treated. But as I presented to you before, is that having a mutation is an additional risk factor. That's why I'm focusing on those individuals. So additional risk factor to plus the LDL cholesterol, that's why it leads to very high risk of premature heart disease. Nevertheless, all patients who have high cholesterol should be treated. So you can, what we showed in that study, we achieved 100% mutation detection rate in individuals who had total cholesterol, in this case, total cholesterol levels in the top quartile, and triglyceride level on the other side, on the bottom quartile. So if you have very low triglyceride level and a very high total cholesterol, we were able to achieve 100% mutation detection rate in those individuals. So there is a suggestion that perhaps taking the triglyceride levels into consideration when clinically defining FH will improve the diagnosis or detection rate of FH mutations. And of course, the family history, I said that now probably 3 times, but looking into that pattern of 50% first degree relatives being affected in an autosomal dominant manner is again a strong indicator that that individual carries a mutation. So yes, there is a probably still issue of the overdiagnosis, or that our clinical diagnosis is not quite accurate. So let's have a look now into the polygenic cause. And this is a cause that majority of your patients will probably have, or this is majority of those who have high cholesterol level that you might see in your clinic. So how did we come about defining that something has a polygenic cause? This was enabled because of the genome-wide association studies for lipids. And back in 2010, there was then the largest study of over 100,000 individuals where they had genotyped SNPs. So here we're talking about SNPs. These are single nucleotide polymorphisms, not mutations. So I always want to separate those two. Mutation is a rare variant that has a strong effect on the phenotype, in this case, LDL cholesterol. So on its own can cause disease. Whereas you've got SNPs, these are polymorphisms. So you and me will have millions of SNPs in our DNA, and that's why we differ. And these variants are common. They are, in one population, they might be more common than in another, but they are relatively common. And on their own, they will never lead to a disease. If you have one SNP here that affects LDL cholesterol, it doesn't have a much of an effect. But if you imagine taking them together, they might have an effect. And so that's what we wanted to test. How many SNPs affect LDL cholesterol, and what happens if you inherit more than average of those SNPs. So we did take those polymorphisms from that GWAS study. Now, bear in mind, this is actually quite old now. There was a very recent GWAS study also from the Global Lipids Genetics Consortium. There's a Nature paper that just came out at the end of last year. But back then in 2010, there was 95 SNPs significantly associated with cholesterol. And each SNP had a modest effect, right? This is the beta effect. So that's when you do the statistics on the GWAS study, you will get beta effect. And that shows you how much a specific variant modifies the LDL cholesterol in this case. So, however, in combination, the total set of those variants explained a considerable part of the variance in LDL cholesterol, and that was 12%. So we can use those SNP in combination to calculate polygenic risk score, and that's how polygenic risk scores, that's what they are really. So what happens is you genotype an individual. So we've got an individual here for several different SNPs. In this case, I'm only showing you an example on 2 SNPs. So we've got SNP1 and SNP2. From the GWAS study, we know that the A allele in SNP1 is associated with higher LDL cholesterol. So we genotype it. We've got a homozygote for the A allele. And then we've got a heterozygote, another individual heterozygote for the A allele and then C allele. And then there is a homozygote, another individual for the alternate allele. And we do this for a number of SNPs. So SNP number 2 in this case is the T allele that is associated with higher LDL cholesterol. And these were the genotypes for the 3 individuals we looked at. So if you then want to translate it into numbers, you would say the A allele is the lipid rising SNP. So if a person is a homozygote for that SNP, they would get 2 points. So 1 for one A, 1 for another A. if you are heterozygote, you'll get 1 point for 1A. And if you are homozygote for the different allele that doesn't raise LDL cholesterol, then you get 0 points. And you do the same for SNP2, where the T is the risk allele, as we say it. So if you are heterozygote, you get 1. This person gets 1 point as well. And this one hasn't got any T's, so they get 0. So based on those two SNPs, we can calculate a score. This is very simplified, but you can basically add the number of risk scores, of risk alleles, sorry. So for individual 1, we add 2 plus 1 is 3, that's their gene score, and so on. But that, it's very simple calculation, doesn't assume, or assumes really, that every SNP has the same effect, which isn't true. So from the GWAS studies, we know that every SNP has a slightly different effect on the LDL cholesterol. So then we generated a weighted gene score, where you multiply the number of risk alleles. So from that simplified score, you can then multiply them by the specific beta effect that the GWAS studies, the statistics analysis provides. So if you multiply by that, you will get a slightly different number. You again sum up the number and you end up with the weighted score. So that's the simple idea of polygenic risk scores. I've been saying gene score, SNP score, so you can see that there's a different vocabulary here, but it really means the same thing. I think these days people don't want to refer to polygenic risk score if the score is based only on, I don't know, 20 SNPs. If you look at all the Nature papers, they these days use millions of SNPs, and that's when they kind of reserve that vocabulary of polygenic risk score to when you have hundreds, thousands of millions of SNPs together to generate one polygenic risk score. So I will be saying either SNP score or gene score in this case, but it really means the same thing. It's just a matter of numbers. So we, from that GWAS study, the original GWAS study, we took SNPs that have the biggest effect, so the beta effect on LDL cholesterol, and SNPs that are the most common. And we identified 12 SNPs. We then calculated the gene score based on those 12 SNPs, as shown on the previous slide. And then we plotted that, that score. Now this one is for the not weighted one. This is just the number of risk alleles. And you can see that's the distribution in the cohort of Whitehall-2 population. So this is a kind of it's a cohort of civil servants from England who donated their DNA, who donated their lipid levels. 3,000, just over 3,000 individuals. We managed to do the score, and what we shown was that the score strongly correlates with the cholesterol level. So here's the deciles of the gene score. So the higher the score, the higher the LDL cholesterol. So it isn't binary thing. It's not like having a mutation and no mutation. This is continuous. It's almost like the distribution of LDL cholesterol, right, in a population, and it nicely correlates with the LDL cholesterol. I want you to bear this in mind. It explains only about 10% variance. That means it's not, again, very diagnostic, but it's a tool that shows you that those individuals who are in a top decile of the score have much higher, significantly higher cholesterol levels than those in the lower deciles. So clearly there is some association. And we call this a genetic instrument. So this gene score was kind of a genetic instrument that allowed us to look into individuals with FH. So this is all done on the sort of healthy, normal, let's say, population. When I say healthy, obviously, not many individuals are healthy in the general population, but just an average sample. So we had that control sample from the Whitehall-2 cohort, and what we then decided to do is to genotype for the same gene score individuals with a phage who don't have a mutation. So they have a clinical phenotype of a phage, but we couldn't find any mutation in their genome using that was, I think, the targeted sequencing method. And what we found was that individuals with a phage but no mutation had significantly higher gene score in comparison to the control. Again, you can see very big overlap. But there are some individuals who are really at the extreme of the gene score. And that really suggested that there is a polygenic component that plays a significant role in terms of individuals with clinical FH but no mutation. And so this information can be also used in terms of identifying novel variants for FH. We think those individuals who have very high polygenic risk score, we can kind of explain them and say, your cholesterol is due to those SNPs. You probably don't have a mutation. Otherwise, if you have mutation and polygenic risk score high, then your phenotype will be very extreme. We can also use that information and say, have a look at those individuals here. They've got very low gene score, even lower than the average healthy population, but they don't have a mutation identified. So perhaps whole genome sequencing, searching for novel genes in those individuals can be a good idea. And another conclusion from this was that testing, a cascade testing, so when you trace within family individuals in the polygenic hypercholesterolemia, will not be that effective, because the inheritance pattern of polygenic cause is different than monogenic. Monogenic, it's clear, you've got 50% chance of inheriting your mutation from affected parent. Whereas with polygenic, when you've got, in this case, 12 SNPs, inheriting them, they are all independently inherited. Inheriting 50% of those, the chance is much lower. So we think that about 30% of polygenic FH cases would be having would be passing on that disease into their first degree relatives. And on those bases, that's why it isn't cost effective. So compared to the Whitehall-2 population, the control, in the FH mutation negative group, 54% had a score in the top 3 decile, in comparison to only, in the top 3 deciles, and only 11% in the lowest 3 deciles. So very different from the control normal distribution. Okay, we then replicated those findings of the gene score in further cohorts, including children. In this study, we had a Dutch children, Netherlands, and Greece, and they actually had the highest polygenic risk score. And I think this is interesting because that's suggests if you take children population, the effect of environmental factors is not as pronounced as in adults. And so they are kind of more pure cohort. And it makes sense that they have the highest genetic reason, polygenic reason for their hypercholesterolemia. They don't have mutation, but they have very high polygenic risk score, and they do have the phenotype. So those studies on polygenic score in familial hypercholesterolemia have provided some evidence again for the health policy, for the NICE guidelines. Since then, the cascade testing resources are really recommended only for the monogenic, for the mutation carriers, based really on the cost effectiveness of it. And Another guideline for research, future research, is to focus on those who have very low polygenic risk score and don't have a mutation. So they are more likely to carry a novel gene. So again, it's in a way an example of precision medicine here, how the different genotype has a consequence as well on the patient management. So patients with monogenic, we reserve much more resources on the monogenic cases, and I will show you on the diagram later. These guys, they get more attention in terms of management in the lipid clinics by specialized lipidologists, whereas individuals who have polygenic cause, and they are more likely as well to respond to treatment much better, easier. They could be managed by general practitioners, which is a cheaper way. So let's have a look how you can go on patient stratification using the genotype. So if you have a patient, an FH patient comes to clinic, I mean, so you don't know at that point that they are FH, but they have LDL cholesterol of 4.9 millimole per liter. They have a strong family history and they have tendinxantomas. So in the UK, what we would do, you would send that sample for the next generation sequencing test. And why my screen is okay. And if the mutation is detected in one of those known genes, then that patient is referred to an FH nurse. So we've got now FH nurses who are very good at performing the cascade testing. So they would contact all first-degree relatives. They have from this test the information of the mutation, and they would send the DNA of the relatives for confirmation if they inherited the cause of FH. But in most cases, the FH patient will not have a mutation detected. And so what happens then? We would determine the 12 SNP gene score that is actually included, not officially, but it is included in that DNA test. And if they have a high gene score, then there is a high probability of them having a polygenic cause. We do not cascade test those individuals because there is a lower detection of affected individuals. Remember, 50% here chance, here probably around 30% chance. So it is not recommended. Someone has calculated the cost and said this is not cost effective. If that's the right choice or not, it's not for me to judge here, but that's how the guidelines are at the moment. So how about if an individual hasn't got the high gene score, but they've got low gene score? So now you're thinking, oh gosh, they haven't got a monogenic cause, they haven't got a polygenic cause. So you could refer those individuals for research to look for other novel causes of FH. And this is where the 100,000 Genomes project in the UK comes in, and I will talk about it a little bit more later. But it is really looking for novel causes in that population is likely to be more effective. But the bottom line is treat the phenotype and you cancel the genotype. So still, goal is to lower the cholesterol level of those individuals, no matter if they're monogenic or polygenic. So what could be the novel causes of FH then? And why do I bother about it? I will tell you after showing you some of the studies how you can go from identifying novel gene to a novel exciting treatment. So our first go at looking for novel causes of FH was being involved in the UK 10K study using whole exome sequencing. We had the chance to sequence 125 definite FH cases. And that is when we found out that 25 of those individuals had actually mutation in the known genes. And that gave us some lesson there, that our current methods were not performing very well. We then analyzed the genetic score, the gene score in those individuals, and further 29 patients had a high polygenic score. So we removed those from the further analysis. We then ended up with loads of variants still. You can imagine whole exome sequencing generates loads of data. So we had to filter all those down. We ended up comparing all genetic variants in 71 unexplained FH cases versus almost 2,000 controls who were sequenced as part of that project. And we identified some interesting genes. The top genes were CH25H and INSIC2. But I have to tell you that the statistical suggestion is still that the significance is not quite high yet to be certain that those genes are causal of a phage. So it is still work in progress. And unfortunately, those sequencing 125 individuals, it's not enough if you want to identify novel cause. So we need to enhance the power of the detection by building bigger cohort, probably more diverse as well. So yes, we then moved to the 100,000 genomes project. So 10 times bigger. And it is genomes now, so we're not looking into only the coding regions, but here into the whole genome, so all the intronic spaces, all those untranslated regions. And this is part of a big initiative that was paid partially by the government in the UK, where we invested into genomics research. Many startup companies since then appeared on the market in the UK. Loads of people were doing whole genome, whole exome sequencing, etc. So it was a big change back then. And we do have a cohort of FH patients again in that project, over almost 500 really, I think now, individuals. And they are part of the cardiovascular disease domain. So there are several other rare cardiovascular diseases being sequenced at the same time. And we had an initial look at the data in the FH cohort. And unfortunately, again, we found some known mutations. So bear in mind, those individuals that were referred to Genomics England should have previously had the DNA testing for FH mutations in known genes. But for some reason, some of them were missed. And so in total, we explained 15% of those submitted cases as to who had a mutation in the known FH gene. This is first time we also managed to look at the ApoE mutation that was quite recent. And it was interesting to see that there were 10 of those individuals had the ApoE mutation. In brackets, you've got a number of different variants. So obviously mutations in LDL are most common. And so there were 54 cases due to 38 different mutations. Having the whole genome sequencing, we also looked at the 12 SNP score. And if you take the 75th percentile cutoff, we identified polygenic cause in 29% of the recruited patients. So again, quite a substantial proportion of the cases. So the next, so we would assume that the monogenic in known genes are explained. Those polygenic 29% cases again are explained. So we would focus really on those with lower gene score, no mutation, to do a burden test analysis. So here we've got, being part of the big project, we have a chance of comparing variants in our FH cohort to those other cohorts within the project, who are all sequenced in the same way. So at the moment, what I was looking at is that GWAS hits. So these are good candidates for novel cause of FH. So these are SNP genes that were shown to be associated with LDL cholesterol in the large GWAS study. So as I said, there was a recent large GWAS study published end of last year. So I'm currently running those analysis. We have looked at, there is a recent paper suggesting RNA processing playing a role in terms of LDL receptor gene metabolism. And again, I'm looking into those specific genes and then looking at any other gene in the genome. But I think, again, the bottom line is that novel cause of FH is likely to be very rare and possibly unique to a specific mutated family. And so the success of such studies really depends on the sample size. The bigger, the better. And the more diverse, the better. Although novel gene will not significantly improve the diagnosis yield, because we will not increase the percentage of detection rate by a drastic measure, but it might lead to a new drug target for LDL lowering. And that leads me really to the next slide. Actually, no, here is, sorry, just a summary as well of what I've mentioned to you about the RNA processing. So you might want to look at that paper. It's a very, a lot of science, to be honest, packed into this one report. We did that in collaboration with colleagues from University of Zurich. So another method of identifying novel causes of FH, it's the approach here was to do genome-wide RNA interference screen. So this is for genes that limit the uptake of LDL cholesterol in cells. And what happened with the results shown that 54 genes were significantly lower. So maybe, sorry, maybe I'm going too fast, but this RNA interference screens means that you knocking down every single gene in the cell and see if that knockdown will have an effect on the uptake of LDL cholesterol. And indeed, 54 genes, when you knock them down, they had a significant impact on the LDL cholesterol uptake, or clearance, if you like, from plasma. And 15 of those 54 genes encode for components of U2 spliceosome. So this is kind of like a machinery that controls correct splicing of genes. Knocking down any one of 11 of those 15 genes resulted in a selective retention of intron in LDL receptor. So clearly something went wrong if you knock down some of the spliceosome genes, and then the whole machinery was not able to correctly splice LDL receptor. And so there was a retention of gene regions that shouldn't be translated. This is, in a way, the junk DNA that should not be there. It should be spliced out, cut out. But it led to that, and that had effect on the LDL receptor function. Now, RBM25 was one of those genes, and that gene actually came up associated as well with FH phenotype in my UK10 case study, so the whole exome sequencing study. So this was quite exciting, because that's another sort of evidence from having those initial genetic findings now to this sort of functional study, showing that perhaps that gene is important. Again, more studies are needed to confirm that. Because it's very rare. We only have 3 patients with that, with variants in that gene, and saying that those variants are actually truly pathogenic, it's another challenge. So from those rare novel findings, how can we go through to effective treatment? And I will get to that point, but first to talk about treatment, we need to cover what is the current treatment available. And initially, it was the fat-restricted diet that we had perhaps a little bit touched on during the discussion. But that reduced cholesterol only by about 10 to 15%. So that wasn't enough for an FH patient. The first drugs licensed were cholesterol binding resins, which were really not tolerated well. And again, the cholesterol reduction wasn't very effective. In 1990s, the powerful and probably the most prescribed drug statins was discovered by a Japanese biochemist. And the first statins would lower cholesterol from 25 to 55%. So that was a big difference from the previous efforts. And although studies, we do know that they have some side effects and some patients do not tolerate them, they do have significant impact on the survival. And this is on Phage patients. Cumulative event-free survival, when we're talking about event, is the CHD event. No statin treatment is the red line, and blue is on statins. And the difference is it's clear, and I think if you have an FH patient, they need to be on statin. They will benefit from that. So let's think about how statins actually work. I hope you would recognize this if you're not a scientist or maybe not a biochemist. But this is sorry, it's not quite working. This is the hepatic synthesis of cholesterol. And so we go from the beta oxidation of fatty acid through to several processes until the cholesterol is actually synthesized. And that process is tightly regulated by rate-limiting enzyme HMG-CoA reductase. Obviously, the cholesterol then goes to play important roles in the body. And statins are actually they are inhibitors of the HMG-CoA reductase enzyme. So they very smartly sort of cancel the function of that enzyme. And so this process of synthesis of cholesterol is reduced. If we knock down that enzyme, then the cholesterol synthesis is lowered. And so what happens if your cell, if your liver doesn't or is unable to synthesize your its cholesterol because its enzyme is inhibited, then the liver will try to express more LDL receptors so that the LDL receptors can pick up cholesterol from the circulation. Okay, so that's how it works. That's how the cholesterol is lowered in the circulation, in the blood. So it clears the cholesterol from blood because it needs it for its own purposes, because its internal or hepatic synthesis of the cholesterol has been inhibited. So it really works through upregulating the LDL receptors. In a way, you remember from the genetic studies. Now, if you remember from my first slides, it is the LDL receptor that needs to be functional in order to clear the cholesterol. So here's a little catch. If you're a homozygote, you clearly don't have the functional LDL receptor because you inherited faulty one from mom and from dad. So you don't have anything to upregulate in a way. But the statins on homozygote cases do not work very well or at all. And that's why homozygote cases needs a completely different treatment, which I will cover. But yeah, I think we need to bear that in mind, that heterozygote cases have 50%, so they've got one copy of a gene that is working well and one copy that is faulty. So we can only upregulate the the good copy of the gene here. So again, you can't go completely normal in a way if you still have one faulty copy of LDL receptor. So now I just mentioned to you the homozygote cases. The heart disease in those children would be evident by about 5 to 10 years of age, which is extremely early. As I said, in a diverse population, you've got frequency of 1 in 300,000 individuals, but perhaps in Pakistan that could be higher, as suggested. Poor response to statin, there is no normal LDL receptors to regulate. So the idea to treat those cases, if they're not responding well to the statins, is to introduce LDL apheresis to extend life. And LDL apheresis, it's kind of like dialysis of blood, where a patient lies down and they've got the tubes connected, their blood comes out to this machine where the LDL cholesterol is cleaned, just like dialysis really, and then the clearer blood comes back. But you can imagine this being very expensive. The patient needs to travel to the center. These numbers are actually from about 7 years ago, so they probably everything has gone up in price. So I suggest that this is underestimate. Patient need to travel, and it needs to be this needs to happen in about every 2 weeks for it to be effective. So it's tragic for those homozygote patients. They really depend on that treatment. They may also require a liver transplant. You would want to, if you want to know a little bit more about homozygotophage, there's this very recent paper, the experience of homozygote phage, retrospective cohort study, a large study that was just came out in the Lancet in February. So some of you might be interested in finding out. But that is the story for homozygotophage at the moment. So that, of course, then drives people to identify new treatments for phage. And one of the ideas was a drug called mipomersen, or Kynamrum, which is a short 20 base pair single-stranded anti-oligo that should bind to the ApoB mRNA. So we, in a way, knocking down ApoB. Now, is it a good idea if you think about it? So this would affect the synthesis of LDL in the liver. So the liver obviously can synthesize its own cholesterol and then lets it out to the circulation. And when it's in the circulation, it is wrapped around with the ApoB. So the idea here was that if you knock down the ApoB, the cholesterol will not be synthesized, or will not be exiting the cell into the circulation and increasing the cholesterol. It has been approved by FDA for use in treating homozygotophage. It has been rejected twice by the European Medicines Agency. And the reason for that is that it is associated with fatty, so here, secretion of VLDL and fatty liver. So you can imagine that if you knock down the ApoB, think the cell, what it does, it still synthesizes its cholesterol, but the cholesterol cannot exit the cell without the ApoB that is knocked down. So it starts accumulating in the liver. Okay, so it isn't a great idea, but it is an option in the US for homozygote cases. There are better drugs on the market that can do a great, actually, LDL lowering. One of those is, well, two of the drugs actually. So there are two different companies develop drugs almost simultaneously. They are fully human monoclonal antibodies against the PCSK9. So PCSK9, if you remember from my initial slides, it's an enzyme that regulates, or that let's keep it simple, that degrades LDL receptors. So if you knock it down, you will not have the degradation of LDL receptors, and there will be basically more receptors on the cell surface available to bind LDL cholesterol. So that's the idea behind PCSK9. It was based on the discovery of PCSK9 mutations in FH patients, gain of function mutations, if you remember, and also on the fact that in a general population, if you do a GWAS study, for example, PCSK9 always comes up as lowering LDL cholesterol. Now I'm confusing you, I think, but loss of function variants in that gene associate with low cholesterol level, whereas gain of function associate with FH phenotype. So in one gene, you can have two different types of variants that lead to the completely opposite phenotype. That, in a way, gives us a good control, genetic natural control. And there are 2 reports of apparently healthy individuals with no circulating PCSK9, as they've got a mutation that removes that gene. And those individuals are healthy, living normal life. And that, in a way, suggests that this is a great control for that drug to go ahead. Because if you knock down in PCSK9, as in those 2 individuals reported, then that will not have any side effects. And indeed, no major side effects using PCSK9, which are both approved since 2015, have been reported. And PCSK9 it's a great target, and more companies, drug developing companies, have picked up on that target. Here we've got a synthetic small interfering RNA made by Novartis. They came up with a drug, again, to inhibit PCSK9, so the same mechanism, slightly different delivery, twice a year. What they're doing is they're having an injection, they call it even a jab. Twice a year, a person would come to the clinic to have an injection with that small interfering RNA agent, which would lower their cholesterol for the next 6 months. And in the UK, this made news. It has been approved in September 2021. Somehow the policymakers made a good deal with the drug company. I'm not involved in, don't know what the deal was, because the drug is still quite expensive, but it's there if it's needed. I think there's, of course, specific guidance who can use the drugs, but it is available. Now, another super exciting thing is a treatment based on gene editing. And I believe Ali Aksa has presented to you something on gene editing. So this is kind of similar philosophy in a way. So you want to modify a gene, and if you do that, obviously that will stay for the lifetime. And that way you could have an effect on LDL cholesterol. So there is a company called Verve Therapeutics based in the US who have published already this in vivo CRISPR-based editing of PCSK9, and that was done in monkeys, in primates, and it lowered cholesterol beautifully in a way, and it lasted for a long time. I think they still got those monkeys, but basically it's a lifetime one-off treatment. So what they're doing, again, they're targeting the PCSK9 gene. They're modifying a single base pair that will lead to a premature stop codon in the PCSK9. And yeah, it's basically knocking down PCSK9. The idea is to do it once in patient's life through the liver. Obviously, there are issues in terms of off-target effects. What if you injected? Are you really sure that it will modify only one base pair in a specific gene? They've got data showing that yes, that is the case. So very exciting. They're planning a clinical trial in human to start soon, as far as I know. So watch the space. That might be a breakthrough soon. So that is where we're heading, and I just wanted you to understand that this is coming from the genetic findings in a disease. So from knowing a very rare mutation in PCSK9 that accounts for about 2% of FH cases. So a rare variant that does not have bigger impact on patients on the mutation detection rate, but provided incredibly important information in terms of identifying novel drug. From the actual genetic finding of PCSK9 to development of the antibodies and having approved the drug was about 10 years time, which is incredibly quick for novel drug development. So genetics more and more has been proving to be aiding the way we develop drugs. And the genetics really play an important role now if you want to test novel drugs. The PCSK9 is a great example. Okay, guys, so I've prepared for you a few quiz questions, and I just want to, for you, obviously I'm not going to mark this, but I want you to kind of try to reflect on what we've learned today. See if you can answer those 4 questions. I guess you can always go back to your slides later. This is very FH specific talk, I realize that, and I know that high cholesterol affects many more, not only FH patients. So yeah, again, I think it's a good model disease that you might be able to translate in the future to other things. So I'll give you 5 minutes, and please think about those questions if you can answer them, just a little exercise, and then we will have a look at the answers. That's okay. I won't be checking individually with you.
Hi, Martha.
Hi.
Thank you for such a long session and management about this disease. And I'm looking at the participants. They have answers.
They do.
Don't need break. Yeah.
Okay, excellent. So I hope you remembered that FH affects 1 in 250 individuals. So the most common Mendelian disease, remember that. There isn't any other ones that I know that is that common. The gain of function mutation, I think I said that so many times now, and it has been evident throughout talking about the treatment. PCSK9 is the gene where gain of function mutation as opposed to loss of function mutations cause FH. What is the most likely cause of FH in mutation negative patients? You could probably think for a while here, but it is the polygenic cause. So the average person that you will see in a clinic as a doctor will have a polygenic cause, a combination of genes, common variants, and a lifestyle. And statins reduce LDL cholesterol by increasing the expression of Ldl receptor. So knocking down the important enzyme in synthesis of cholesterol or drive expression of Ldl receptor. So I hope you got some of those answers right. I'll just leave you this summary slide again. I think we probably summarized quite a bit in the quiz. Yeah, it is an inherited disease of high LDL cholesterol, very strong risk of CHD. Unfortunately, remains underdiagnosed and undertreated. So even if you have diagnosed patients, still some of them are not receiving the treatment, appropriate treatment to lower their cholesterol. As I said, the European guidelines say lower LDL cholesterol by 50% from the baseline. Mutations in genes involved in LDL cholesterol pathway cause a phage, the frequency 1 in 250. Polygenic hypercholesterolemia mimics the monogenic phenotype. Knowing the genetic cause is very important in terms of the monogenic cause, as it helps to have a cost-effective screening strategy. And genetic findings in a phage, I hope I've just demonstrated that, can generate important knowledge for novel drug development. So last but not least, some of my acknowledgements here. Obviously, I've presented many other people's work, hopefully cited them, but yeah, thank you very much for your attention. I will stop screening my, sharing my screen, so thank you. And I'm sorry if I've been talking too much, but it's my favorite subject, so I can talk all day.
Thank you so much, Dr. Martha, for joining us and sparing time for us. I hope participants have learned some knowledge out of it. And thank you so much, participants. This brings to end of the third day of the session. So see you tomorrow, inshallah.
Thank you so much, Samar.
Thank you.
Thank you.
Thank you, Martha. Thank you so much.
Thank you. Take care, everyone.